I see the camera on.
Hello, Dr. Modi. Good morning.
Good morning. Hi, am I audible?
Yes, you
Am
are
I audible?
audible. Yes, you are.
Fantastic. Can I just check my screen once if I can share it?
Sure, you can do that. Students, team SRC, please see that. You can share now. You have your collaborator. You have a right to share.
Yes, sir, it's visible.
It's visible. Just let me check if it's moving.
All the way at the bottom near the Zoom bar, you will be able to see the presentation. Yeah, right there.
Yes,
Near the Zoom bar. Yeah, that's the one.
That's a full screen mode, right?
Yeah, now we see the full screen. Yes, it's a
All right,
presentation.
the slides are moving, right? All right.
Yes,
Okay,
it's
super.
moving. Perfect. Yeah.
And I have a video to play, which is on the browser. Let me just check that as well.
Afrin madam, if you can guide sir about the video, voice, audio.
So when you share it, there'll be an option to include sound. So when you click on
Yes,
share, yes, sir.
that I have done, but my screen is shared now, right? Is this the video now?
Yes, sir.
The lung on a chip is a micro device that
Yes,
has the
sir, it
potential
is.
to replace
Yes,
animals
audible
for
as
the
well.
testing of drugs and toxins.
Fantastic. Okay,
The device, which is lined by human cells,
go on.
Ma'am, if you allow, we start.
Just give me a minute.
Dr. Srinivas Navle? I think we should start. It's 5 minutes over. We should respect time of all our speakers and participants. We can go live on YouTube, are we?
Are we?
Yes.
Yes, ma'am, we are.
Okay, I'm ready.
Dr. Srinivas will take little time. He just dropped a message, but we can start with, no problem. Okay?
Okay, ma'am. Yes.
Sure.
Good morning, one and all. We, Nishka Thakkar,
And that's this side.
are delighted to welcome you all to the day 2 of the 5th Student Research Congress organized by University of Mumbai along with our esteemed industry partners ACG Worldwide and SciTech Center. The pursuit of knowledge has no limits. The more you learn, the more you realize there is so much more to discover.
Welcome to today's special session, where we explore the exciting intersection of science, innovation, and transformative research. In this ever-evolving landscape, knowledge is the key that unlocks new doors, leading us to previously uncharted territories. As we delve into the latest breakthroughs, We will witness how cutting edge discoveries are reshaping our understanding of the world and opening up limitless possibilities for the future.
SV, SVKM's Dr. Bhanuben Nanavati College of Pharmacy offers a platform that is graced by renowned experts, each a pioneer in their field, who will share their groundbreaking work, innovative ideas, and their unique insights. Their research is not just expanding the boundaries of knowledge, but is driving also progress. These distinguished speakers will inspire us to think confidently and push the limits of what we believe is possible.
As we gather today, let us embrace the power of curiosity and collaboration, for it is through these principles that we will continue to transform ideas into reality. Together, we have the opportunity to learn, grow, and most importantly, contribute to a brighter and more innovative future for all.
It is with great pleasure that I extend a warm welcome to our distinguished keynote speaker, Dr. Deepak Modi, Scientist G and Head of the Molecular and Cellular Biology Laboratory at ICMR, National Institute for Research in Reproductive and Child Health. Dr. Modi is an elected fellow of the National Academy of Sciences India and has been ranked among the top 2% of scientists worldwide by Stanford University. With over 150 publications and numerous national awards, his current research areas include mechanisms in embryo implantation, placental development, genetics of infertility, and sexual development. He is also an editorial board member of journals like Fertility Science and Research and Human Reproduction.
Dr. Modi's profound expertise and invaluable insights are a tremendous asset, and we are truly privileged to have him with us today. We are excited to hear the enlightening perspectives he will share, which are sure to inspire and inform us all. Please join me in extending a warm and sincere welcome to Dr. Deepak Modi.
So I take this opportunity to welcome Dr. Modi on behalf of SVKM's Dr. Bhanuben Nanavati College of Pharmacy. Sir, we sincerely appreciate your time today and your thoughts you will be sharing with the young professionals who would be now venturing into research as a profession. And I'm sure your talk will motivate them to take research as a career and also think that research in life sciences has a huge potential to make the difference in society. So thank you very much, sir, and welcome to the virtual platform of SVKM's BNCP. And we thank you for graciously accepting our invitation and spending your time with us today. Thank you very much. Over to you, sir.
Thank you. So very kind invitation, and thank you for such a generous introduction. You have been really very nice host, and I have enjoyed being on this platform. I wish there was a way of doing this meeting physically, but I understand the constraints. But anyways, knowledge can be shared on many platforms, and I hope I'm able to do justice to your expectations. And I take this opportunity to also thank Dr. Sujata, who kindly invited me here. And my students attended the meeting yesterday, and they're also online today, and they were very excited with all the talks that have been happening. So thank you so much for even organizing this. So without spending too much of time, I'll start my presentation. Um, so my screen is visible, right?
Yes, sir,
Yes, sir.
it is.
All right. So what I'm going to talk to you today is about organ on chips. In the current era, it's getting increasingly, things are increasingly changing in the way we are doing science and the way technologies have developed. One of the important milestones, which I believe, is development of these small mini organs on chip. What I'm going to tell you today is about how do we develop them, what are the principles, what are the applications, and over in the latter part of my talk, I'll also cover our experience of making an organ on chip. So essentially, my lab, as spoken about, is interested in understanding how pregnancy is initiated, how the early placenta is formed, how the endometrium and the embryo crosstalk to initiate the entire process of pregnancy, and how things go wrong there to cause implantation failures, recurrent miscarriages, IVF failures, and even some pregnancy complications. In addition, we're also looking at the role of Y chromosome genes in male infertility. So this is a very fundamental research question where my group is looking at stem cells, inflammations, epithelial to mesenchymal transitions, and so on and so forth. In addition, we have also developed assays for genetic diagnosis of male infertility. We use a lot of artificial intelligence tools to predict the outcomes of IVF in male infertility. Both of these products are commercialized and will be in the market soon. We are also developing newer assays to study infections associated with preterm birth. And the talk that I'm going to give today is a part of our endeavor to develop placenta and endometrium on chip. And this work is done in collaboration with IIT Bombay, Professor Abhijit Majumdar. So when we talk about the pharma industry, studies have shown over the last numbers of years that it takes on an average 10 years for a new medicine to complete the journey from an initial discovery to actually come it into the market. And of them, the longest time that's taken is in clinical trials, which may even take 6 to 7 years. And some drugs even take way longer. Now, unfortunately, while this is the longest period of the longest incubation period in a pharma industry for a drug to hit the market, 90% of the drugs that pass the preclinical testing fail in clinical trials, and that's primarily due to unanticipated toxicity. Now, this costs millions because doing clinical trials is way expensive because you have to do phase 1, phase 2, phase 3. Each one requires human volunteers, and in these volunteers, you have to pay them for the compensations, you have to pay for insurance, and a lot of things are involved there. And this is very costly. And this cost can come down if we are able to predict toxicity way in advance, way better than what we are able to predict today. And also, one of the problems is that we do not understand the mechanisms of actions of many drugs. We are unable to predict failure in patient subgroups. Now, it's not that the drug fails in every individual or drug shows toxicity in all the individuals. There are sex-specific, there are pathology-specific, there are age-specific or race-specific effects, which we are unable to predict in these subgroups. And the long-term effect of chemicals, especially in the cosmetic industry, this has been a growing concern that we use so many cosmetics that come into the market. And what are the long-term sequelae of these chemicals in the cosmetic industry are not clearly understood. And one of the major problems, again, is the ethics. We use animals to do preclinical research, and clearly we know that animal research needs to be regulated. It is regulated, it is controlled, and the idea is to reduce the numbers of animals. So, are there any ways of determining the toxicity yes? Are there any ways of minimizing toxicity and predicting the toxicity way better than we are able to do today? Now, when we look at the preclinical scenario, within the pharmaceutical drug development industry. It involves three models. Primarily, one is an in silico model, which begins with where you predict the toxicity of the drug based on its based on its chemical properties, which is in silico prediction. The drugs which clear the in silico prediction get into in vitro and in vivo toxicity, where you also determine the PKPD, the pharmacokinetics and pharmacodynamics of the drug. And the models that we use there are in vitro models, and in vivo we do animal models, which you typically begin with the mouse, and then you need at least one higher species, preferably a primate, but primate research again is highly regulated. So species like dogs or cattle are used by different industries. Now the problem is when we are using these cell lines to determine first, and the first step when a drug is developed and it's clear, you can see that there is a dramatic reduction in the numbers of compounds which are predicted to be toxic in the preclinical screening model, either on cell lines or in the animal model. And about that, only a fraction of that goes actually to the clinical trial. Now, the bottleneck is here because we are trying to predict toxicity here. And when we reach this in the clinical trial, the compounds which re-enter the clinical trial are found to be toxic to humans, which were otherwise deemed to be non-toxic using these in vitro models or in vivo models. Now, when we use these in vitro models, we use cell lines. These are cells which are grown in the lab dish. All of you would have heard about animal tissue culture. And we use these cell lines. Typically, you grow them in the lab dish. They are grown as monolayers. They are often single cell types. They are more often immortalized cell lines, either derived from patients who had cancers, or they're immortalized using a genetic approach, and therefore they're not physiologic in nature, and they do not simulate the organ as a whole or even in a part. Remember, all our organs are three-dimensional, and our organs are comprised of more than one cell type, but the cells which are growing on a lab dish. Now, your lab dish is a very, very stiff substrate. Your body is not. We are on soft substrate. Your lab dish does not contain extracellular matrix. The cells, whereas in the body, the cells experience extracellular matrix. So obviously, you will say that why not we do these studies in animals? And we have been traditionally and conventionally using animals. However, the animal genomes and the human genomes are very different. However, we may convince ourselves the mouse genome and the human genome is not absolutely identical. There are way more differences in physiologies. The metabolic rates in animals versus humans are very much different. The mice has way higher metabolic rates than humans. And let's accept it that mice are just not 25 gram humans just because they have 2 eyes, 2 ears, 2 hands, and 2 legs. Obviously, we say mouse have 4 legs, mice doesn't have hands. So let's not anticipate the things that work in the mouse to work in humans. And that's not surprising because that's why 9 out of the 10 molecules that worked in mice do not work in humans. And therefore, there is a clear need for additional smarter models which can help us to do this preclinical research way better than what we are doing today. Just before I move ahead, anyone who has question can type it into the chat box. I'm unable to see the chat box right now, so I'll try to answer them at the end of the presentation. But if somebody also has a burning question, can interrupt me and I'll be happy to answer that question while online. So do not hesitate in asking questions because I'm here to answer your questions. Don't just keep it to yourself. We can always discuss it and others may learn through that. Moving ahead. So we must understand that like in the lab dish, when we grow cells which are planar and two-dimensional, life is complex. Our tissues are made up of multiple cell types and each cell type talk to each other. It's not that one cell remains in isolation. It's like when in a family, when we stay together, in a college, when you're all together, you all love to talk to each other and you respond to how the others are behaving towards you. And therefore, the way you behave in isolation versus the way you behave together, especially with different people, is way different. In addition, the cells experience soluble factors which are given by the blood and the other cells in the body. As I told you, that there is extracellular matrix which also helps in biochemical signaling into the cells. The extracellular matrix also provides biophysical properties, and it is a place where a lot of soluble factors are in a controlled manner are given to the cells. In addition, there are thermal stress, there are stress and strains which the tissues experience. Like, for example, our lung is not a planar lung. It is constantly shrinking, contracting and expanding so that air comes in and out, which is obviously not simulated in your lab dish. And in cases of neurons, you also the electrical impulses going on. So the idea is that can we simulate one or many of these properties into a lab dish And this has led to the development of the concept of an organ on chip. And it is believed that the organ on chip is going to bridge the gap between the planar 2D cells and the mouse, or directly from the planar 2D cells to the humans, obviating or at least reducing the experiments that need to be done in the mouse. And this leads to the idea of developing organ on chip. So, what are organ on chip? So, these are artificial testing subjects that resemble human organs in some aspects. And usually they are based on the principles of microfluidic, where you have where they can be either static but with some fluidic substances there. They may have a flow or they may even have a gradient. Our cells don't experience everything at a single concentration. Some cells constantly produce factors, some cells utilize them, and there are always gradients, which we call in the concept of developmental biology, the sink and the source and the sink approach. And these gradients may have an important role to play in our tissue physiology. In addition, cells which experience blood at the same time also experience flow or shear stress. When the blood is flowing through our capillaries, the cells of the endothelium experience a flow over them. And biophysical studies have shown that these flows do alter the responses of the cells. And ultimately, I've told you that the 3D architecture that the cells experience in the tissues need to evaluate it. So these organ on chips try to incorporate the neighboring cells, cellular matrix, physical fields, soluble factors, flow and shear stress, 3D architecture, and probably some gradients. I must remind you that organ on chips are not artificial organs. So they're not meant to grow organs in a lab dish for organ replacement. So those of you who might have imagined this, I must correct your thought process that these are not artificial organs. Artificial organs are not organ on chips. That's completely a different field. There's a new field which is emerging. It's called as organoid, mini organs in the lab dish, brain organoids, liver organoids, placental organoids, endometrial organoids, and even blastoids are being developed. These are three-dimensional structures which resemble the human structure, and they can be grown in a lab dish, but they do not fall under the field of organ on chip. Organ on chip is a different entity from these two. So principally, what are the things that an organ on chip must have? Obviously, if we are talking of humans, then they have to contain human cells, probably more than one cell type, but there are many monocellular organ on chip, not a problem as far as you are fine with what you are wanting to do. They preferably should be three-dimensional in nature, but there are exceptions to this. They should capture some of the structural aspects of the organ that you are studying, at least one. It should also capture some of the functional aspect of the organ that you are studying. And it may have a microfluidic flow, which could act like a perfusion or to study shear stress. So why microfluidics? So when you look at organs, our organs are pretty large and you will require volumes and volumes of cells and volumes of media, which are then very difficult to do it in a lab condition, whether when you want to put it in the dish, and especially in industry where high throughput, where you want to test many drugs at many concentrations at many time points in a short span of time, you need things which are high throughput, and therefore microfluidics come in pretty handy. Because they are less in volume, they are portable, you can take it them from one place to another. Because they're made up of material which are typically transparent, it is easy to put them under a microscope and observe them. They can be using the principles of engineering, you can do the scaling there. So by the principles of engineering, if you know the size of the organ and the dimension of the organ, you can apply the principles of engineering and scale them down. So micro scaling is possible to do, and by therefore the microfluidics becomes useful. And surface forces dominate over body forces. So you can use these surface forces to evaluate things which are there inside our body. So microfluidics need a fabrication technology, and the engineering people who are involved in microfluidics use multiple kinds of fabrication technologies. That favorite being microfabrication, which falls under the category of photolithography, micromilling, 3D printing, and injection molding. The other fabrication technologies also include electrospinning. So essentially what you do in a photolithography is you take a silicon wafer. So primarily first you have to imagine your design, how your microfluidic device is going to look like, whether I'm going to grow two cell types, one on top of the other, side by side, how I want the blood flows or the fluid kinetics or the gradients to be formed in my device. A lot of imagination goes where I'm going to add my cells, where I'm going to add my medias, how the media is going to flow through these channels. The designing is done first, and this designing is then put on a computer system, and the computer system is attached to something that is going to create a photolithography kind of a condition where you coat the silicon wafer with your material of choice. Typically, currently, PDMS is used for this kind of thing. So on the silicon wafer where you have coated this material, you kind of do etching over it, and then you prepare your mold. On the mold, then you pour PDMS, where you do the casting, and then you remove it, and you get your chip ready. You can do micro milling, where you can take these blocks and then actually do the cutting over it the way you need the design. You can do a 3D printing. 3D printing is getting in vogue. You would have seen it long ago. It was on sci-fi movies. Now you can practically see them, that you can have your design first in the computer. You can have a ink, which could be plastic or they could be bio inks, and you can actually grow them into the system and then use them as chips. 3D printing is also currently used to make small mini parts of organs, or even they're considered to be part of transplantation processes. In bone, at least 3D printing is being extensively thought to be useful. And then there could be injection molding. I'm not going to go to the details because this is a very, very engineering talk, and an engineer like my collaborator, Professor Abhijit Majumdar, will do a way better justice than me. Anyway, so what are the steps of working with the organ on chip? So primarily a lot of time goes in the design. So typically a biologist or a clinician who has an imagination to do this work collaborates with the engineering department, spends a lot of time, the engineers understand what do they mean by things, what do a biologist mean by things, and trust me, the language between these two groups is very, very different. They do not seem to understand we talk. We do not seem to understand their terminology. So in our experience, at least, me and Abhijit spoke for almost a year or two to even figure out what all are we wanting to do and what will be the final imagination like. Then a lot of time is spent in designing that how are we going to grow the cells? Are we going to grow them, as I said, one on top of other? What is going to be the matrix? How are we going to separate them? What are the media requirements of the cells? What are the flow rates and blood pressures? Now, there are two things here. One is you need to know what are the flow rates and pressures in your actual organ, what is the design of your organ, and then you have to try to simulate them, and in the device engineering has to be changed accordingly. Uh, uh, After that comes a fabrication. So then you decide the material, you choose one of the methods that I've explained you about, and then you decide the extracellular matrix. Then the entire device needs to be sterilized. Then it comes to the biology lab, where it goes into the animal tissue culture room, which is typically a sterile condition. Then you start growing these cell types, and you have to optimize your culture conditions, because the way you grow your cells in a 2 dimension versus the way you grow in a 3 dimension is very different. And then by the end of it, after you have assembled the whole thing, you have to do analysis. And here is the very key point, that this entire choice of the organ on chip device depends on the question that you want to ask. This will get increasingly clear as I run through my presentation. So your analysis, whether you want to just check live dead numbers of cells which died or which are living in the organ on chip, or you want to take some functionality organ on chip, or you want to see how much of drug is passing from one region to another, or how much of drug is metabolized, or how the entire organ, or how the organ is responding to infections. Depending on the question that you have and depending on the organ of choice, all of these can keep changing. So a few examples of an organ on chip. So what all organ on chips can mimic? What all it can do? As of today, organ on chips are largely used to study tissue barrier functions. Most of you would have heard about the blood-brain barrier. The brain is protected from the drugs or the infections that we have by a blood-brain barrier because brain is not as vascularized like our skin or our muscles or our lungs or our heart. So how things in the blood go to the other side and how things from the other side.
Dr. Modi, you're muted. You will have to unmute yourself somehow.
Sorry,
Yeah,
sorry,
you're right.
sorry.
Go ahead.
Okay. Did this slide, could you hear on the slide the barrier functions? Part of it?
Yes, you
Okay.
just kind of went off the mic,
All right.
but then
Okay,
you're
sorry.
back. Yeah.
Sorry, I probably just touched some parts. Okay, anyway, so the barrier functions of the organ is what the organ on chips are currently very well designed for, where you can use these commercially available membranes. You grow epithelial cells on one side. Typically, you put on a membrane, you can grow them. You can put your endothelial cells or the blood vascular cells on the other side. The barrier is formed, and you can test how things move from one side to other. And these devices are typically used to study transport of drugs, biomolecules, viruses, microbes, and hydrogels are used to model this interface. Over and about this, you can simulate the blood vessels because, as I told you, that like for example, a blood-brain barrier or any barrier which is inclusive of the blood, things the blood is always flowing. The other side may or may not be static. And in this, you want the blood to flow at a certain rate, not too high, not too low, so that your cells may experience some shear stress, depending on your organ. In some organs of blood, the endothelial cells don't experience shear stress, and you can have these flow kinetics. For example, in the kidney, the glomerular filtration happens, and there is a constant flow of liquids into the organ, and this can be simulated very well, and the flow kinetics can be regulated there. Sometimes you may want to add pulsation, as I told you, in case of the intestine or in case of the lungs or the heart. There is constant pulsation, stretching and straining, and you can incorporate that in your device. And in case of tumors, where you want to study metastasis, you may want to study cellular migration or invasion. So you cannot have all of this in one device. For each of this that you may want to study, you may have to do a different design. You may have to do different principles of making this device. Different materials may need to be used, and even different cell types may be involved. So I'll give you some examples. So this is one of the beautiful examples, and the video I will share in a minute, which was published by Harvard a couple of years ago, but I still love to show it. In this, they have tried to simulate a lung on a chip. So first of all, we know that lung as a barrier model. The endothelial cells, all of you who have been a little extra reading during COVID would have understood that the viruses come from the air, and your endothelial cells, which are the lung alveolar cells, which are the epithelial cells of the lung, are exposed to the air, where the viruses or things come in there. And then you have these epithelial cells lie on a barrier of extracellular matrix, and there are blood capillaries in which the lung is highly vascularized and things flowing from there. So now your epithelial cells don't have to experience media. Now, when you grow these epithelial cells in 2D, two dimensions, they are all covered and they're submerged inside your media. Now, in a lung, they're not submerged in media, but they're exposed to air. So now you need to have a design where you can grow these lung cells, but they also, after you put them into the device, they need to be exposed to the air. Then you need to, on the other side, you need to have a blood flow in a direction. And now this entire unit needs to contract and expand. And therefore, if you apply vacuums on either sides, pulsatility, you can have these pulsatility simulated, and the contraction and expansion of that small region of the lung which it experiences can be simulated in the lab dish. Here, typically, they have used a thin PDMS layer, a commercial membrane to separate these two cell types, and gels are used as barriers. And in this, they've done epithelial modeling, like they've studied epithelial inflammation when you have inflammation happening in the lung. You have excess of IL-2, and you can increase or decrease IL-2 in the lung and see what is their effects on the lung cells. You can study how bacterial infections, toxicities are there. You can incorporate cells like, so here they've done epithelial cells and endothelial cells, epithelial cells, smooth muscle cells. They've also incorporated immune cells here, and they've studied things like pulmonary edema, infiltration of circulating neutrophils, so on and so forth. I'll just play this video. Just let me know if it is well. Here it is. Is my screen visible? Is the video screen visible? It's a black screen right now.
Yeah, it's a black screen, Dr. Modi.
Okay, now it's lung on a chip.
Yes, now we can see.
And you can hear it.
Yes.
No, sir, it's not audible.
Or voice.
Voice, yeah, the video is not.
It's not clear.
The video is not audible?
No.
Ouch. I thought it was before, right, when we tested it. How do I do this now?
So the screen which you are sharing, you have embedded this video in that, correct? On your presentation. From there itself, it will take it because you have put up that audio setup while sharing.
Sir, just a second. Ma'am will guide you.
So
Okay,
when
madam.
you click on share, sir, there'll be an option to include sound. Or can you navigate to Teams page?
Yes.
The last tab, sir.
Yes.
Yes, sir. So when can you stop sharing, and then when you share again, there'll be a toggle of include sound. Yes, sir.
Okay.
The default
And
is
I'll
off.
share
That's
my
why.
screen. Okay. And now am I on the lung on the video page, lung on chip
Yes,
page?
sir. Yes, sir.
Yeah, I'll keep it page. Is the audio there now?
Air sacs.
Yes, sir.
Oxygen is transferred across
From
lung.
from the beginning. Okay, there you go.
The lung on a chip is a microdevice that has the potential to replace animals for the testing of drugs and toxins. The device, which is lined by human cells, mimics the mechanical and chemical functions of a living, breathing lung. With every breath, air enters the lungs, filling and expanding microscopic air sacs. Oxygen is transferred across lung and capillary cells and into the bloodstream. This is also where aerosol drugs are absorbed and where infections and tumors form in the lung. The lung on a chip is crystal clear, flexible, and about the size of a small computer memory stick. But it contains tiny hollow channels created using microchip fabrication techniques. A porous, flexible membrane separates the two channels at the center of the device. The opposite sides of the membrane are lined by human lung and capillary blood vessel cells. This mimics the arrangement of lung and blood vessel cells in the air sac of the lung. Application of cyclic suction in side channels makes the entire flexible sheet and cells stretch and relax rhythmically, just like our lung cells do when we breathe. In the lung on a chip device, air at the top of the human lung cells and a liquid medium containing human white blood cells flows below the capillary cell layer. To test how well the lung on a chip device replicates the natural responses of living lungs, we introduced bacteria into the air channel to mimic an infection. And we introduced white blood cells to the blood channel. We then saw the white blood cells migrate across the capillary cell layer, through the pores of the central membrane, and into the airspace where they engulfed the bacteria. Here's a video that shows this response, in real time, viewed through the device. The tissue cells are not visible here, but we can see white blood cells flowing freely in the capillary channel of the device, just as they do in blood vessels of a healthy person. But when we infect the air channel by adding bacteria, the immune cells abruptly stick to the surfaces of the capillary cells on the opposite side of the membrane, located directly below the infection site. Here's a magnified view showing a migrating white blood cell making its way through the first capillary cell layer, wriggling through the pentagonal hole in the flexible membrane, and then moving out of focus to the other side. When viewed from the air channel, with all cells visible, you can see a round white blood cell popping up from below. Just like in a real lung infection, the white blood cells, which are now colored red, engulf and kill the green bacterial invaders. We've also had success in mimicking pulmonary edema, a condition in which fluid leaks from the bloodstream into the air sac. To mimic the edema, we introduced interleukin-2 into the blood channel. This is a cancer drug that is known to cause pulmonary edema as a side effect in humans. The dose we added was the same that a typical cancer patient receives. When we added the drug without breathing motions, only a small amount of fluid made its way.
Sorry, there is some network issue. I think there's some network issue from my side.
Added the drug without breathing motions, only a small amount of fluid made its way. However, when we repeated the experiment with normal breathing motions, the fluid leakage increased by more than threefold and resulted in complete filling of the air channel over the same time course as observed in human patients. We also saw blood clots form in the fluid-filled airspace, again, just as is observed in humans with this life-threatening lung condition. The lung on a chip can also mimic the toxic effects of airborne nanoparticles on the lung. In fact, experiments with the device led to the prediction that breathing increases their absorption into the bloodstream. This has been confirmed in animal studies. Bio-inspired micro-devices that can mimic whole human organ function, such as the lung on a chip, could potentially replace animal testing and bring new therapies to patients faster and at lower cost in the future.
So, I'm back. And now, am I sharing my PPT? Am I on the PPT screen?
Yes,
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 sir,
sir.
you are.
And it's moving.
Yes.
Yeah, very much.
Thank you. You saw how this is a very fantastic example of organ on chip simulating the barrier part of the organ on chip, where depending on what the question was, if they wanted to study bacterial infection, they would include the immune cells. If they wanted to study edema and inflammation, then they would add IL-2. When they wanted to study edema occurring, they would apply the forces which are required. So depending on the question that you want to ask, these organ on chips can be modulated and the designing principles may change. Another example I'll give you is for a tumor on chip. When a tumor on chip are designed where a cancer is formed in a body, the cancer is formed. The cancer is growing on a basement membrane over a cell which over the epithelial cells which are already there. The epithelial cells start proliferating, but then they want to break this epithelial barrier, get into the stroma or get into the blood and undergo metastasis. So typically you would design such a organ on chip device where you have the epithelial layer growing, you have your upper channel where the tumor is growing, and this is typically, now these are not exposed to air, but in the body they're inside the blood, so you would have the blood over there. Below it you will have the tissue organ, the stroma, and you can study stromal tumor interactions and what happens to how the stromal fibroblasts can contribute to the tumor growth, tumor dynamics, and if you give drugs to the stromal cells, can you change the epithelial cells? If you kill the tumor, what happens to stromal cells? So the entire crosstalk which is happening within a tumor growth is what you can simulate in a tumor on chip. Here they have used a tumor on chips to study drug cytotoxicity and metastasis. So here what this group has done is they have grown They have a microfluidic channel which is resembling the blood vessel. The inside layer of the microfluidic channel are layered by endothelial cells, which are the cells of the blood vessel. Outside it is the stroma, and the tumor cells are growing. And the idea is to figure out if the tumor cells growing on one side can make way through the stroma, through this epithelial layer, through this endothelial layer into the bloodstream, and even go to the other organ which is growing on the other side and metastasize. And very beautifully, the study has shown that you can simulate this entire business where cells from the tumor escape as like what we call them as circulating tumor cells. They could capture these cells out and study them separately once they have left the primary tumor and they have entered into other chamber. Either you can capture it into the blood or you can capture them after they have metastasized and study. And you can simulate this process of metastasis in a small device. And now you can study effects of drugs. And this is where it is believed that it is a game-changing scenario, because now you're actually not studying this migration of the cells in a lab dish and believing that the metastasis, your cells go to the entire actual process of tumor extravasation into the blood circulation and entering into the other organs, which is truly what happens into the body during metastasis. And when you can simulate this process and now test drugs, we believe we are way closer to humans than what you would actually simulate in a lab dish or in a mouse. The long-term idea currently, as I told you, that the organ on chips are designed for the organ or part of the organ or functionality of the organ. And as of today, multiple organ on chips are made, which now are defined as, because they are human in nature, they are called as human on chips. There are kidney on chip, there are heart on chip, there are lung on chips, there are gut on chip, liver on chip, and bone marrow on chip. Each one for different functions. For example, the lung on chip is used for studying infections and absorptions of drugs which could be given. So suppose, for example, you want to test the effect of a nasal vaccine or a drug or a chemical or a cosmetic or let's say a perfume which you want to study, whether does it irritate lung epithelium. You can use this a lung on chip. When you want to study cardiotoxic drugs or cardiometabolic drugs, or if there are drugs which may affect the cardiac heartbeat, you would use a heart on chip to study if its effects on contractility and conduction. If you want to determine how a kidney is metabolizing or excreting the drug, you can use a kidney on chip. When you want to study immune cell differentiations and effects of drugs on immune cells or develop leukemia, you can have a bone marrow on chip. Liver on chip is extensively being developed and characterized to be used in studies of drug metabolism, because all the drug that we take go to the liver and metabolize there. Now, instead of having these cells in a liver cells in two-dimensional, you can now make it exactly like a liver lobule. You can have blood flows through them and study the effects of drugs on metabolism, study the PKPD actually in the lab dish, and you can will be able to simulate the effects like what we see in humans and like what we predict from that in mouse. And you can do things using gut on a chip. Imagination one fine day is that you should actually have an entire human on chip, where you can have the lung on chip and the gut on chip to begin with, where you can study either an aerosolar drug or oral drug, where you can study the first pass effect onto the liver. And from the liver, you can connect this gut on chip to a liver on chip, where you can study the metabolism. From the liver on chip should be connected to a kidney on chip, should be connected to a bone on chip, should be connected to a heart on chip, and similarly a lung on chip. And people are now imagining to have a brain on chip, the blood-brain barrier here, and you can simultaneously study the effect of one on many organs. Here, the idea is to develop multi-organ on chips so that pharmacokinetics can be investigated. Both barriered and non-barriered organs can be added there. One needs to mimic the residence times of the drugs, the organ and organ proportions, because all cannot be of the same size and all cannot have the same numbers of cells. So one has to study these proportions. There have to be very controlled and then let it outlet. It's not just about fluid flowing through all of them, but each tissue has a very controlled rates of blood flows, and this will need to be simulated, and each tissue will have a different microenvironment. This is the imagination of the future. Right now, it sounds very sci-fi. It is indeed very sci-fi, but I do see that this will become the light of the day where one finder will be able to incorporate all of these organ on chips together on a single device. I'm going to switch gears here and I'll talk to you something about one organ on chip which we have developed, which we call it as placenta on chip. This work is done in collaboration, as I've told you, with Team IIT Bombay, Professor Abhijit Majumdar and his student Saurabh Mukherjee. And in my lab, Anshul and Pratik that are extensively involved in this project of developing a placenta on chip. Before this, does anybody have questions? We can take a short break before I put into the next heavy presentation. Does anybody have questions here? Or should I continue?
Dr. Modi, just the feedback. I must say, yes, personally, there are a couple of myths about organ on chips. Listening to your speech and the content, I'm so glad that it gives such a clear idea, and there are a lot of opportunities to work in this area. I'm sure this is such a simplified version of your PPT and presentation, and the way you elaborated and talk, it has definitely inspired a lot of students. Personally, me, I'm kind of so glad that I listened to the entire topic, and today we are so thankful that you are with us. Thank you very much.
May I add?
So
May I
yes,
add if
sure.
we have permit time? Personally, now today what Dr. Modi talked about is simulating especially the disease model, and especially when you're working on far-fetched infectious disease, where you have limitations with respect to animal primates, and you don't know how your delivery system would behave. Preliminary studies with organ on chip, especially when he gave example of lung, that inspired me to venture into that. We had worked quite a while, but we were thinking how to go about. So maybe Dr. Modi will come to your lab and we'll have a elaborate discussion how we can go about. Thank you so much for.
Thank you. Students, do you have any questions, or can I move ahead with my presentation? As of now, I don't see any questions in the chat box. But if anybody has, please do feel free. Anybody has a burning question?
I think they're pondering, and maybe at the end they will, shower the questions to you.
yes.
Sure. Should I move ahead?
Yes, sir, you may proceed.
Okay. So am I back on the presentation mode?
Yes,
Sir.
All right. Okay. So we'll talk something about placenta on chip. So when we talk about pregnancy, so currently we are in an era, and when I spoke about all these organ on chips, everything sounds very exciting and very eluding, and we are now at a place where we are thinking that probably we'll be able to kind of get these drugs way faster into the market, and we will be able to predict our toxicities better. And now even the idea is that we can develop these organ on chips for each patient separately. You can make iPSCs from these cells, and from these stem cells you can differentiate, let's say, lung and endothelial cells, and develop a lung on chip and study at the personalized level what is the effect of drug on that individual. And in case of cancer, this is actually becoming a reality where they're using this tumoroid on chip, using patient-derived cells, and studying the effect of getting to the personalized medicine, whether the tumor is responsive or non-responsive to the drug. However, when you're talking of such a fine scale of personalized medicine, a group of people who are completely not able to take drugs are pregnant women. All of you would have heard about the thalidomide tragedy several years ago, where women were taking this morning sickness pill called as thalidomide, and the baby, and this is, remember, I'll remind you that they were cleared of the preclinical safety trials. They were also in the clinical trials. They were cleared in the clinical trials because they were safe. But when the babies which were born, because they took it in pregnancy, and the babies which were born had very short limbs and legs, and they realized that that was because of this drug called as thalidomide, and that passed on from the mother to the baby, leading to something called as teratogenicity. What same was also true for corticosteroids, and since then the regulatory authorities became very stringent about it, and pregnant women were excluded from the clinical trials with the sheer fear of keratogenicity. And as of today, while we speak, and I'm not exaggerating it, for pregnant women, only 8 drugs have been deemed safe to be taken in pregnancy. And there are only 12 drugs which are very clearly contraindicated in pregnancy. However, about 60% of women who become pregnant will require to take some form of medication because of their pre-existing conditions like diabetes, like hypertension. About 70% of women who become pregnant will need to take some drugs during pregnancy due to their some conditions which they develop during pregnancy. Like, for example, if you develop a cold, cough, or start having fever, and you need to gulp in a paracetamol, or the woman is having an extreme back pain and want to gulp in a painkiller, and if she goes to the doctor will say, I don't know, you will take at your risk because this drug, whether it is safe or unsafe in pregnancy, I don't know. And trust me, 90% of the drugs which are available in the market today, some of these which are even over the counter, the safety of them in pregnancy is not known. And largely because we cannot do clinical trials in pregnant women for the fear of teratogenicity. And now that we don't know whether it is safe on them, you cannot give them. And that gets into a circular loop. And therefore, the need was felt, there's an acute need for models which could be used for drug safety testing in pregnancy. And imagine, I'm like feeling crazy when each time that I give this talk, that I get goosebumps at the whole idea that you are excluding a population for not taking drugs. I was told by my clinical collaborators that a woman who is suffering from sickle cell anemia needs to take a very simple drug called as hydroxyurea so that her sickling crisis can be taken care of. And a large numbers of deaths in our country during pregnancy is because of sickle cell anemia, and not because the sickle cell anemia has any adverse effects on pregnancy, but because a sickling condition cannot be taken care of by simply giving her a hydroxyurea because we don't know the safety of hydroxyurea. So the idea is that can we develop model systems which can predict the safety of these drugs during pregnancy? Now, in pregnancy, we have this very interesting organ called as the placenta. Now, placenta is an organ which all of us had when we were in our mother's wombs, but we have given it up when we were delivered. But this placenta has played a very crucial role in making what we are today. Primarily, placenta is essential for maintaining pregnancy and overall fetal well-being. The placenta is the first organ which does all the metabolisms for us. It also produces the right kind of hormones so that the fetal growth is regulated well. It also allows exchange of nutrients. A mother which takes the nutrients, it has to go to the baby. It is filtered through the placenta in a very regulated manner. It also removes waters and waste products and gases from the babies, which is not required. They go back to the mother's circulation and then they are excluded. Then it also protects the babies from immune rejection from the mother. The mother's immune system is semi-allogenic because it is half the baby's half mother, half father, and the baby is likely to be excluded by the immune system because it's foreign in nature, but it is thanks to the placenta who protects us from immune insults from the mother. It also protects the fetuses from maternal insults, including infections. There are a lot of studies we have done in our lab, how the maternal SARS-CoV-2, how the placenta helps the mother to protect the baby from SARS-CoV-2 infection. And if there's something that goes wrong in the placenta, SARS-CoV-2 can enter into mother-to-child transmission of SARS-CoV-2 can happen, and the baby will also have COVID-19. So the placenta acts as a very smart barrier against maternal infections, and primarily it also is a barrier to various drugs and metabolites. So all the drugs which the mother takes, either she metabolizes or the placenta metabolizes and does not allow it to get to the baby, keeping the baby safe, but some of the drugs can pass through it and lead to teratogenicity in the baby. So the hypothesis we have is that the drug could be considered as teratogenic if it crosses the placenta in high enough concentration to cause fetal abnormalities, and any insult to placental functions and impairing its functions can lead to adverse pregnancy outcomes. So we think that if we study the placenta and are able to study placenta better, we could better predict the toxicity of the drugs in pregnancy. Now, currently, what are the models to study placenta? Obviously, animal models are of choice. However, the animal placenta, the mouse placenta especially, is not like the human placenta. There are several cell types in the mouse placenta which are not there in the humans. Very many human placentas, placental cell types are not there in the mouse, and structurally and functionally, both these placentas are very different. And therefore, I'm not surprised that teratogenic safety in the animal models shown do not replicate to that in humans. Then obviously you can use placental cell types, placental cell lines. But as I told you, these cell lines are 2D in nature, they're planar, and there is no interactions, and primarily you cannot study passage of drugs in a 2D cell line. Currently, the model of choice is placental perfusion. So you take placenta from women who donate the placenta for research purposes at term. Then you do a perfusion where you simulate the maternal, where you put this placenta in a chamber and you connect these chambers with the fetal circulation using plastic capillaries and collect things in the fetal arteries and fetal veins. And now you can perfuse the placenta with the drug, and you can see how much of drug is coming into fetal artery and vein. You can measure it. However, this model has not been very useful because for each drug, for each concentration, you need a new placenta, and these functionalities will differ from woman to woman. Availability of the placenta is therefore definitely not high throughput, and whatever has happened to the mother during pregnancy, the placental profile will get altered, and therefore the results do not reproduce across time. And therefore, 3D culture, especially the organ on chip model, has been thought to be primarily a option which could be developed to study the effects of drugs on placenta. So we have begun this endeavor in the last 5 years, and it's a part of Anshul's PhD and Pratik's PhD as well. But before I go through what we have done, I'll just quickly give you a very short lesson on placenta 101, where the placenta has something called as a stem villus. So the placenta is exposed to the maternal blood, and the maternal blood communicates to the placenta through the structures called as villus, and this is a histology of the villus. So your mesenchymal villus is an avascular ending of the stem villus, which is responsible for the longitudinal growth of the placenta. Over time, they're also responsible for hormone production, especially the pregnancy hormone HCG, and they're responsible for production of nutrients. Whereas the terminal villus are vascular villus, which are floating in the maternal blood onto the stem villus. So these are the vascular terminal villus, and these have the barrier functions. I'll speak about this a little later. The other part of the villus is something called as the anchoring villus. So the tips of these villus anchor onto the maternal decidua, and these anchoring villus where the trophoblast cells start proliferating and they form the anchor onto the decidua. These are called as the column trophoblast cells, which are responsible for how much the placenta is going to invade into the mother and do the spiral artery remodeling. And both these villi are very crucial for placental functions. These anchoring villus have completely different function than the mesenchymal villus. The anchoring villus determine the extent of placental invasion and spiral artery remodeling, whereas mesenchymal villus are primarily responsible for growth hormone production and passage of nutrition and the barrier functions. So we thought, when we began this idea of placenta on chip, we thought, oh wow, let's simulate all of this in a single chip. After discussing with Abhijit and a lot of deliberations and deciding, we realized all of this is not possible into a single chip, and therefore we developed something called as modular devices. We have developed 5 different modular devices to study these different aspects and different functions of placenta. Not just functions, but depending on the question that we are asking, we use different kinds of devices. In the interest of time, I will talk only about 2 devices. One device that Abhijit Lab has developed to make placental spheroids. We have to make spheroids, and we have used this to make placental spheroids. And the other is a device that simulates a barrier function. So the question that we asked was, can we develop stem villus-like structures which are resembling the terminal villus. So this is histologically what a terminal villus is going to look like. It's going to be essentially a mass of cytotrophoblast cells with an outer layer of cytotrophoblast cells which are differentiated from the cytotrophoblast cells. You can see it here. And in the terminal and in the mesenchymal villus, these trophoblast cells have to be vascularized, and the endothelial cells start growing below the cytotrophoblast cells, and they form the barrier one. So first, we wanted to make these spheroid-like structures which resemble the human placenta. And the idea was to make it in a high throughput nature. Currently, you can make these spheroids using low attachment plates, but these are where you take a lot of time. We wanted to develop this device. So in Abhijit's lab, we have patented this now. We have developed a device which can allow us to fabricate about 100 micro wells in a 6-well plate in a single well of a 6-well plate. So typically what we do is we take agarose and we put on this device into the 6-well plate. We remove it and you can see hundreds of these micro wells. And then we do the cell seeding. Am I audible?
Yes, sir.
Okay, because I think my battery of the audio device is going low. So if I suddenly stop being audible, just remind me, okay? I think my headphones are getting some issues. Anyways, so what we do here is that once we put this, you can see on the agarose, you can see this nicely imprinted wells here. We trypsinize our trophoblast cells, which typically we use BO cells or HTR cells, which are cell lines which are resembling the human placenta, and we seed them in liquid medium, and by the principle of gravity, they go and sit inside the bottom of each of the wells, and because of the space constraints, they immediately start forming spheroids. You can see in these videos here, at the time of plating, they are dispersed, but they soon start assembling together, and they form a spheroid, and then the spheroid starts expanding. And you can now see them under a microscope. They look as very tiny dots because it's a very low magnification image. And we have stained them with calcine to show that these are viable. And the blue staining here is a nuclei. And when we do this experiment, if you use a 96-well U-bottom plate, you can typically use 8 spheroids per treatment group, and to set up this entire thing from trypsinization to adding your medias and so on and so forth will take you about 2 hours. Using our device now, we can plate about 600 spheroids per plate. Hundreds of spheroids can be there per well, and the time taken to seed this entire thing is just 10 minutes. So typically in the 6-well plate, you can have a total of more than 600 spheroids, depending on the design that you have made here, and you can have a very rapid assessment of large numbers of spheroids in a single experiment. What did we do then? What Pratik and Anshul did were they trypsinized. They took vivo cells, which are, or you can even take trophoblast stem cells. We are trying to work on this, and we seeded them onto these chambered devices. And these cells need to undergo differentiation using a drug called as forskolin. So you can have now the inner mesenchymal core, and they should have the outer cytotrophoblast core and the inside cytotrophoblast core. So on the device, all these cells are first plated and they're grown for 2 days. The drug is given to allow them to differentiate for 2 days, and in 4 days we were hoping to get these structures which resemble the terminal buds of the growing villus. And this is one of my favorite slides where you can see histologically. You can see my arrow, right? Or should I put it in the pointer mode? I don't know if there are pointer options. Yes. You can see here the actual placenta where you have the inner villus core and the outer cytotrophoblast layer. And believe me, this is the spheroid which we have done a histological section. We have taken the spheroid, put it into paraffin sections, and stained them with hematoxylin and eosin. And you can beautifully see they so resemble each other. You can have the inner cytotrophoblast core where the cytotrophoblast cells are happily growing inside, and the outside it's a syncytial layer. You don't have to just believe me because there is histology. The real life placenta, the outer layer, the syncytotrophoblast cells make the hormone HCG, and even in our device, the spheroids which we have got, make HCG. You can see this beautiful HCG production from the outside cells mostly. So it is now resembling what we call it as the terminal villus bud. The next question that we asked was, can we also now simulate the anchoring villus? Anchoring villus is where the placenta is anchored onto the maternal decidua. You can see here, and at the tip of this villus there is something called as a mesenchymal core. Now, this cannot be achieved by the BIVO cells that we have. For that, we use another cell line called as the HTRA neo cells. And these cells are trypsinized, put into the wells, and this within 2 days formed a nice spheroid. And when we histologically cut sections of the spheroid, so you can see the spheroid growing into the microwell, a nice tight structure is seen here. And on actual real life placenta, you can see that there are strong cell-cell adhesions, and you can see the histological section of this spheroid, and you can see that there were nicely, tightly formed structures which resemble that you see here in the real life is now mimicked in our device. And then when you stain for E. cadarin, the column cells of the placenta of the anchoring villus strongly express E. cadarin, and we can see strong E. cadarin expression on the histological section of these spheroids which are grown in our device. So you can see very beautifully look. And these spheroids now not just resemble structure, but also have the molecular profiles of the same. Now, I told you that the anchoring villus, from the anchoring villus, the trophoblast cells have to come out from them and migrate into decidua. So what Tatit and Anshul did was they removed these spheroids from the device and they started growing them on a matrigel, which resembles the extracellular matrix of the decidua. And in this video, I'll try to play it again. I'm not sure if I can play it. Yes, here you go. You can see that there are nice outgrowths coming from the cells. So like from the column cells which are coming out and getting into the decidua, you can grow them as outgrowth assays. And these cells are live in nature because they stain positive for calcine. And this is one of my favorite images because the cells which leave the spheroid from epithelial nature, which are E. cadarin positive, acquire a mesenchymal profile and they become vimentin positive. The real life placental the extravillus trophoblast cells which leave this villi acquire vimentin, and the cells which are left from our spheroid, which we think is resembling the mesenchymal, the stem core, the anchoring villus core, is now leaving out, and we can see this beautiful vimentin positive cells undergoing epithelial to mesenchymal transition, and this we think mimics what we call it as a stem villus. What Anshul has done is now prepared these spheroids in the using this device. We are using the device in a 6-well plate. We have seeded the HTRA neo cells. Then we have added a drug. I told you the drug hydroxyurea, which is required to be given to women who have a pre-existing sickle cell anemia, but we don't know the toxicity of this drug. So he has coupled this experiment where we have grown 600 spheroids. In each well, now you give one concentration of the drug. So you can test typically 4 concentrations of the drug. One is negative control, one is a positive control. So in a sense, you can study the effect on 400 spheroids at a go. What do? We couple it with a very high throughput, high content imaging system, and you can now in live monitor the growth kinetics of this drug. And you can see in the positive control, the black line here, it is cytotoxic and therefore it is killing the cells and the spheroid growth is severely impeded. But at most of the physiological concentrations of the drug, there is no effect except at the highest concentration, which is not physiologic, which is suprapharmacologic. There is some retardation of the growth kinetics. Now, this is just spheroid area. We have about 5 to 7 different parameters which we can test about the growth of this. At the end of the experiment, we harvest these spheroids and do live death staining. And what Anshul tells us is that in the positive control, most of the cells are dead. In the negative control, most of the cells are live. And at the highest concentration, the inner core of the cells die. But however, the proportion of live cells remain not significant, remain at the same at the pharmacological concentration of the drug we want to test. And as we speak, Anshul is technically testing the such effects on outgrowths. This is one of the very early experiments, and he tells us that at the highest concentration of the drugs, the outgrowths is impeded. So this drug at the suprapharmacological concentrations is toxic to placental cells, but within the pharmacological range, it is not very toxic, at least to the anchor villus. And now we are testing this on HCG production, the terminal villus, and so on and so forth. Now, the question here is now this design, what we have is a spheroid. But the drugs, as I told you, that one of the important functions of the placenta is the barrier, where the drugs, the outer cells, the syncytic trophoblast cells can experience the drug and they pass on into the fetal circulation through the endothelial layer. Now we wanted to simulate this barrier into our dish. And obviously the spheroids cannot do it because they are not vascularized. So we wanted to simulate this barrier function where we have a top layer of syncytic trophoblast cells, which are grown on a basement membrane, which is the placental basement membrane. Below this are the endothelial cells, which resemble the fetal capillaries, and there is fetal blood vessel here. And we wanted to simulate this, that can we simulate this barrier in our lab dish? And therefore, we made a different design. Here now, we have a two-chambered device, which is separated by a membrane, which is a porous membrane, similar to the membrane which was used for the lung-on-chip. It's not the same, but it is very similar. And on this, in the top chamber, we can grow our trophoblast cells. In the bottom chamber, we can use the fetal human HUVEC endothelium. Yes, that will be able to develop this barrier device. So we have made these devices using principles of engineering. Trust me, it sounds very simple, but it's not quite that simple. What will be the dimensions of this? How will we load the cells? How will we cover it from top? Moment we cover it, then we develop vacuums inside. So we had to make holes. These holes should not allow leakage of media. Almost a couple of years went in just figuring this out. And then the bigger challenge which we had was growing the cells in them. But before we embarked on using this device for drug testing, we wanted to determine, does it allow the differentiation of the cytotrophoblast to syncytotrophoblast? And the steroids we could, but is it doing it even in the device? Because the dimensions and the way the cells are organized is very different. Does this device produce HCG? Because these are the syncytotrophoblast cells, as I told you, produce HCG. So can we make HCG in this device? And does it have the barrier functions like that of a human placenta? So this is what we call it as benchmarking. So any organ-on-chip device has to be benchmarked before it is used for actual things. So what did we do? First thing was, can we culture the right cell types together? So what we did was, we made this device, and a lot of experiments and time went into determining what is the best extracellular matrix which is liked by the BO cells, which are the trophoblast cells, and the HUVEC cells both together. Like, for example, collagen was loved by the BO cells, but the HUVEC cells did not like it, or it was vice versa. I don't remember. Finally, after a lot of experimentation, we came down to that matrigel is something which is resembling all the extracellular matrix of the placenta. We could grow the BO cells on top and then cells on the bottom, which is typically like what you see in a real-life placental barrier. And you can see on a high-content imaging system, the yellow cells are the top layer, the endothelial cells which are in pink are in the bottom layer, and we can overlay them and we can see that and we can see here very nice monolayer of both these cell types are forming on top of each other. And now when we cut a section and we see through the side view, you can see very beautifully looking cytotrophoblast cells growing on top and the endothelial cells growing in the bottom. And what you don't see is the membrane, because membrane is transparent and it is layered with the extracellular matrix. You can see very beautifully the two cell types are growing on top of each other. Now the question was, can we synthetilize cells? So we did. So we grew the cells. Now the biggest challenge was that both these cell types require very different medias. So a lot of time went in determining which cells to grow first, which cells to grow later, which to be differentiated first, which to be added later. Finally, what Anshul came up was that you have to grow the vivo cells first, then differentiate them, and while they're differentiating, you grow the endothelial cells so that the media requirements become slightly compatible. Now, when we do that and we image on HCS, this is stained with, I think it is stained with picoderine, I don't remember. You can see this nice fused endothelial and nice fused cytotrophoblast cells. You do not see the cell-cell boundaries in between them. What happens in the real life placenta, like the syncytotrophoblast fused to become the cytotrophoblast fused to become the syncytotrophoblast. And when you add force column, you can see them. And if I cut it and show you the side view, you can see this beautiful syncytia seeing here. You can see these multiple nuclei here, not separated by a cell boundary. And below it are these beautiful endothelial cells sitting there. And when we take the media out from the maternal chamber and measure HCG, you can see that within 24 hours, there is tremendous HCG production, which remains stable over the next 24 hours. Whereas in the undifferentiated cell, there is a consistent level of HCG indicating that there is no differentiation. And very nicely, you can see this pregnancy chip we have used. And when we add the media in the chamber, post-differentiation, you can see this presence of HCG, whereas pre-differentiation, we do not see much of presence of HCG. So we were very excited with this device because now it's not just growing cells in the way we need, but it is also allowing a differentiation and producing hormones which a real life placenta produces. Now the next benchmark and the important challenge was, does it form a selectively permeable barrier? Now, when we look at the barrier functions, there are different kinds of barrier functionalities it has to achieve. Some molecules which are very small in nature have to be transported intact without getting metabolized, either through the intracellular transport or some of these have to be partially consumed, metabolized by the cells and get into a very gradient fashion. So more in the maternal chamber, less in the fetal chamber, and that example is glucose. And some of the things do not have to get transported. For example, a large molecule like IgM. IgM will not get into the placenta and it will just remain into the maternal circulation. So we challenged ourselves and asked, and for the final question we asked was urea. Urea has to pass from the fetal chamber into the maternal chamber because urea is made by the fetal kidneys and that is thrown out in the maternal blood to be thrown out by the mother. So we asked whether all of these are simulated by the device that we have made. First and foremost thing, now all of this business, which is the barrier function, is done by a protein called as ZO1. ZO1 keeps the barrier intact, and we can see that our syncytotrophoblast make abundant of ZO1 in the device, and you can see nice lining of ZO1 present here. The other yellowish ones are the whitish yellowish ones are the nuclei, and you can see the ZO1 positive cells. So that means they're forming a barrier. You don't have to believe just immunostaining. You challenge this. And FITC is a small molecule. So if we add FITC into the maternal chamber and we test it in the fetal chamber, the blue line is without cells and the red line is a device with cells. And you can see that the rate of transport without cells is very linear, but with cells, there is some amount of impedance, but it is a small molecule. It can go through in between the paracellular transport, and you can see it's nicely transported at a rate way lower than that because it was transported immediately, completely 100%. Then you should have seen a profile very similar to without cells, but there is an impedance because there is lesser rate of transport. However, if we use a very high molecular weight FITC, so if you conjugate FITC with dextran and make it high molecular weight, without cells, it easily transports from one side from the maternal chamber to the fetal chamber. But when there is cells, there is nearly no transport even at the end of 8 hours, indicating that this is a barrier which does not allow large molecules like 70 kilodalton FITC dextran to be passed. When we use IgM, which is a physiologically relevant molecule, when there are no cells. Other questions? When there are no cells, we do see transport of IgM coming into the fetal chamber. But when there are cells cultured, we never saw any IgG. We do not see any IgG coming into the fetal chamber. All of it remains in the mother's chamber. So, like the mother does not allow the IgG to cross the placental, IgM to cross the placental barrier, here also in our placental barrier, IgM is not crossed. As I told you, urea has to go from the fetal chamber into the maternal chamber. So Anshul spiked the fetal chamber with urea, and we saw within 8 hours. So this is at 0 hours, we had 60 nanogram mg per dl of urea in the fetal chamber, dropped down to about 40 mg per dl within 8 hours, and you can see exactly the same amount got accumulated. So now from the fetal chamber, urea goes into the maternal chamber. So it's similar like what we see in a real life situation from the fetal urea, which has to go into the maternal chamber urea. We do see a reverse transport also of the molecules, and here it does both through the paracellular transport and the intracellular transport, and we could simulate that in our device. So we are very excited by this because now it just not looks like actually the barrier of the placenta. It just does not make the right, it just makes the right kind of hormones, but it also behaves as a right barrier, allowing small molecules to pass, not allowing the large molecules to pass, and also do a reverse transport. The most exciting data comes now, which is a glucose transport. In the real life situation, the mother has about 3.6 millimolars of glucose, sorry, 4.4 millimolars of glucose in her circulation. The glucose does not pass through the cells directly, but it is taken up by the cells by the GLUT receptors, and these GLUT receptors uptake the glucose, and some of the glucose is utilized by the cells, and the rest of it is given into the fetus and the fetal circulation remains a steady state of 3.6 millimolars of glucose. And this difference is essential to make a steady state gradient so that the mother can, in a very steady state, constantly pass on the glucose to the fetus. So this gradient needed to be established, and we asked whether we can develop this gradient in our device. And the answer is yes. If now here, sorry, it's a little different. The red line, we measured the amount of glucose going from the mother's chamber into the fetal chamber, and without cells, it just linearly goes. But with cells, it does not go linearly. There is an impedance. And when we calculate the ratio of the glucose present in the maternal chamber to the fetal chamber, without the cells, it is nearing 1 as expected, but with cells, it is 0.86, and the fetal to maternal ratio is 0.81. So you can see we are getting very close to the ratio that we need in the real life situation. We are able to also evaluate in our chip device. And currently, Anshul is using this device for disease modeling like gestational diabetes, effect of infection. He is also testing hydroxyurea, and Pratik is testing the effects of antituberculosis drugs on these placenta-on-chip devices so that we can determine if these drugs can be given to pregnant women or not. So to summarize, we have made devices which make the placental barrier, and we have functionalized it. The functionalized device has properties similar to that we observed in the real life in vivo. From the technology readiness level, we are at a proof of concept stage. We are designing this device so that flow kinetics can be prepared. The device is fabricated and experiments are initiated, and we are patenting this device presently. Now, I'll just quickly run you through something about the flow kinetics in the device. Now, the challenge that we faced was why this is important is for you to understand that how designing and engineering people have to work very closely. Designing, engineering, and biologists have to work very closely. Now, all the on-chip devices which are available, the gut on-chip, the lung on-chip, and the placenta on-chip, which are made by others, blood is directly flown from the capillaries, fetal capillaries, and the maternal chamber in a very unidirectional manner. And there is a lot of shear stress, both in the gut on chip and the lung on chip, and even the placenta on chip. Now, the problem is in the real life placenta, the maternal chamber does not experience a flow because the placenta has an open circulation. The spiral arteries, which are now modified by the syncytotrophoblast cells, throw in a lot of blood in the feto-maternal interface, and the placenta is bathed in the maternal blood. It is like you in a swimming pool. The swimming pool water is constantly changing, but you don't experience that shear stress because of the large volume that the swimming pool you are in. So for your size, the swimming pool is pretty large, and therefore you do not feel the stress there. Now the problem is the current devices experience a lot of shear stress on the maternal side, and this is not what you see in the real life placenta because the trophoblast cells on the surface never experience shear stress. So we wanted to simulate a device where you do have the exchange of the blood in the maternal chamber for slow exchange of gases and nutrition, but at the same time prevent shear stress. So the blood flows have to be regulated without shear stress, but at the same time the exchange rate have to be maintained. What I spoke to you about is a static device. Now we challenge ourselves to see if we can make a gradient device. I spoke to Abhijit, Anshul had a series of meetings with Saurav and other people in his lab like Dave, and then we had finally came up to a very interesting design, which I'm not going to bore you too much with the principles, but we finally came up with dimensions of the device where we had these capillary chambers, and by modulating the height of the device and the volumetric flow rates within them, we are now able to achieve a device which looks something like this. If you can inject I've shown you that this is the dye. From one side you inject the blue dye, the other chamber we inject the pink dye, and you can see that they are flowing at a certain rate, but without the shear stress because of the design that we have made in there. And there is very little intermixing so that there is no transfer, there is no leakage of this device. Now, Anshul and their Saurav have been able to grow cells on both of the sides of this device. Experiments are ongoing, and we hope that we'll be able to modulate blood flows and have a device which is not just static but also with flow. But I must remind you that this is not to undermine the static device, because the static device is high throughput in nature. We can mount these devices in 12L plate and study the drugs in a high throughput manner. Those with this flow rates cannot be used in a high throughput manner because you have to engineer a lot of things. They put a lot of these pumps which are there, and as a consequence, you cannot run 10 devices at a go in a chamber, in an incubator. You will need very large numbers of devices. So static devices are useful when you want to do the high throughput testing. You would do these flow rate devices. And currently we are benchmarking both these devices to study nutrition transport, to study effects of infections, to study transplacental permeability for at least anti-TB drugs and drugs for sickle cell anemia. And we believe we will get this technology in the next 2 years up to technology readiness level 6 so that we can commercialize, and it should be used for just in the last 2 slides, the challenges for all organ on chips, not just placenta on chip, the material requirement, multiplexing, and downstream sensing. To culture relevant cell types, 2 different cells have very different media conditions. Even in our own case, the cytotrophoblast cell line and the endothelial cell lines, the medias were very different. If you individually grow these cells in both these medias, they don't like each other. So culturing them together was a big challenge, and there is a need for a universal media. So trust me, if anybody who is an entrepreneur here and can devise a universal media for all the cell types, you'll probably be the richest man in the world after Elon Musk. It's one of the biggest challenges for all organ on chip experiments. You need a universal blood substitute. You need something which is, because here we are just currently using medias which are not having the properties very similar to blood. It is still, we are mimicking flow kinetics using medias, but the viscosities of bloods and other blood cell types which are there are not simulated on this device. We need blood substitutes. And obviously the biggest challenge again is maintaining sterility of this device, because they are these micro in nature. Cleaning them up, removing all the alcohols is a big challenge. Also, we need to work on the manpower side, user psychology and training. Currently, there is still a lot of resistance from the community to use this because it involves a lot of experimentation, which is not as linear as what we are used to. We have still not been able to validate this and how we are going to regulate it. Me and Anshul and Pratik were in a meeting last week on these alternate to animal experiments, and there was a lot of debate on how are we going to regulate? What is going to be the validation benchmark? Now, this is something which I have made. Who is going to validate it? Are there other labs in the world who will take my design and validate it to show that really it works? Is it the way we are claiming it? Because in industry, reproducibility is very crucial. And the biggest challenge is scalability and production. how are we able to scale? Can we make hundreds of devices in a go? Today, in our lab, we can make 10 devices in a week and we are able to do our experiments. But if we're talking in a large scale application, we need hundreds of devices. So entrepreneurs here is also a challenge for you. You can develop companies for yourself where you can make these devices for yourself. If you have engineering friends, if you have people who are smart enough, making these devices and selling them by itself is going to be a good business. Making medias for them, making cell lines for them. And if you want to develop a CRO, you can actually start using them, developing them in your lab and offer these services to the industry. And with this, I'll close. I'm really thankful to a very smart audience that I have here. I'm grateful to Abhijit's lab, who have been a constant support. My students, especially Anshul and Pratik, sorry, I've missed this is like the oldest slide. All the money that we need comes from the Indian Council of Medical Research, my parent organization, Department of Science and Technology, through the imprint 2C. We were funded for this initial part of the Pleasant Township. And thanks to all of you, your parents, because all the taxes that you pay this is the taxpayers' money which we have used to develop this device. And therefore, I thank all of you from the bottom of my heart that your taxes have been used in this and you all are party to this innovation that we have done in our lab. We hope that it's going to change lives and we are going to make pregnancies safe. Thank you, and I'll be happy to take questions.
Thank you, sir, for your wonderful presentation. Now let's move to the question and answer session. But as we are limited to time, we'll select a few questions.
So the first question is by Ulia. She is telling that could you please let us know whether the chip be scaled up for industrial application?
As of now, no. We are all at the laboratory scale. There are some industries who are making. So it's not our our steroid device, but there are a lot of industries who have made high throughput steroid device, and they are in the market. But many organ chips are yet not very, very scalable. And there are no industries that I don't remember any industries who are right now market ready to give devices which can just take it and start using it and all that.
Thank you for the response, sir. Sujata ma'am asks, we would like to know how can we have organ-on-chip model for evaluating drug permeation for posterior eye segment, especially for age-related macular degeneration and retinopathy.
So my collaborator and a good friend of mine, Dr. Prajakta Dandekar at Institute of Chemical Technology, is developing a retina-on-chip to study these. Again, if I think it's a barrier function like device where you put the endothelial cells, you grow the retinal cells on top. So principally, this device by design remain the same. It's just that you need to decide the blood flows and how you want this. So design will need tweaking, but in principle, the idea is to grow the two cell types together and study it, and then add things to make it disease conditions. So for example, in our case, when we want to simulate gestational diabetes, we heighten the amount of blood in the maternal chamber so that stimulates a disease model. In the lung on chip, they wanted to look at bacteria, so they introduce bacteria, or we introduce bacterial products like LPS. So depending on the question, you will.
Thank you, sir.
Just give me a minute. I think my headphones have died. I will just try to increase the volume. Yes. Am I audible?
Yes, sir.
Okay, great. Yeah, tell me.
Yes, sir. So the next question is from Ranu. He is asking that how do we remove the media from the chip for testing HCG?
So in our case, we had made a design where the top and the bottoms were covered, but they were made with PDMS, and such that because PDMS has self-healing properties, we can pipette it out from the bottom chamber. If in our design where we have the flow kinetics, we add the media which is flowing from one side to other, from the other chamber it is collected, and the HCG is tested using the standard biochemical assays.
So Dr. Sayani says, excellent session. Dear sir, I have a question. How can you negate the effect of media while testing for the drug? And what about the metabolities effect on the placental cell?
Sorry, can you repeat the question?
Yes, sir. How can you negate the effect of media while testing for the drug? And what about the metabolites effect on the placental cell?
Absolutely. So at this point, it is not designed. As I told you, we need universal blood substitutes. At this point, it is only media. But again, we are looking at when we are doing preclinical testing, we are using the same sets of medias. I'm not saying that we are at the pinnacle of the organ on chips where we are able to do everything on that. As I began in my talk, you are doing like baby steps, and this is one of the early steps we are at where we are able to do it. We are able to at least make these organs. We are not foolproof. Now when it comes to effects of metabolites, for example, we know that hydroxyurea is not metabolized by the liver. It is intact, and therefore we give hydroxyurea. If you know a drug is metabolized in the organ on chip, you give metabolites and test them. So that's how you would match. Now, if I know if a drug is going to be metabolized by the placenta, I know so we will test all the drug metabolizing enzymes on the placenta and study the effects of drugs on these metabolic activities or whether it itself gets metabolized. So again, the idea of telling you this is that depending on the question, you will design your organ on chip and your design. It's not that one size fits all.
So there is one more question from Palak. She is asking that what will be the cost of organ on chip and will it be practiced in India?
The cost as of now is obviously because most of us are doing at the lab scale and we are making this at the lab scale. So we do not really do the costing part of it at big and commercial levels. I don't know how much will it come to. I think Kasturi from CCMD, Atal Innovation Center, has done this exercise, but honestly I wouldn't remember at the back of my mind what is the number. Yes, it is practiced in India. I did show our example of placenta on chip. That was the whole idea, to tell you that it is not something which is happening in the West. It is happening right here in the same city where you are residing. ICT, Institute of Chemical Technology, is also doing a lung on chip, a lung tumor on chip, or a tiny on chip. There are a couple of labs in the south, even in the north, who are trying. So all of these efforts are ongoing. We will definitely see the light of the day. Some of these will be coming in forms of publications. You can collaborate. You can speak to Abhijit and actually have your designs done. The sky's the limit. I think there was a question that says, why don't we use blood instead of looking for blood substitutes? Blood, you will need to keep it in the liquid state. You will have to add kilograms of heparin and EDTA, and this heparin and EDTA that itself kills the cells. So we need a substitute. There was a question which I found very interesting, where he said that if 9 out of 10 drugs fail in the mice, why is it still used in the process of drug discovery? That is the whole business. The whole idea is that because that's not a fantastic model, we need this model. Again, I'm not saying that it is completely going to substitute the animal use, because whatever that we do, we will never be able to make an entire human on chip, because the immune system, a lot of psychological effects. I mean, in vivo, you have 1 million of cell types. So these are very simplified versions, are very broken down versions of the organs. Now, in the placenta on chip, I did not tell you, but definitely the fetal side of the placenta has Hofbauer cells, which act as immune cells. The maternal side also has immune cells. If I'm trying to do the whole thing in the on chip, we are just not able to do it. We are not there. So animal models do have their own place. I'm not saying we're going to completely replace them, but testing's done in animal models. Parallelly, they'll be testing on organ on chip, so that we are able to predict these things way better and faster. So it's trying to shorten the timeline, not change and at least hopefully we'll be able to do better. I'm not sure if we'll reach the day where we'll be able to completely substitute, because I hate calling animals as these experiments as alternate to animal testing. I love to call them as alternates to human testing, because I can't take humans and start studying toxicity on day one. So these are nice alternate systems. Animals are their own place. On-chip devices are their own place.
So last question for the day. if you can highlight on the regulatory and ethical aspects of OOC.
The regulatory aspects definitely. So that's what I said. So all of you are interested should join the society and attend its meeting called as Society for Alternate to Animal Testing. It has its annual meeting, and for at least pharmacology students is a great society because it gives you what are the different models and the regulatory aspects of testing. So OECD currently has accepted a lot of alternate in vitro models like the zebrafish model, the hydra model for a lot of cosmetic products. Right now, it has not accepted any of the organ-on-chip device models, but at least for the cosmetic testing, now a lot of cosmetic companies advertise that these are all vegan products not tested on animals, because for cosmetics, the regulatory agencies are very clear. We are given up on this. The skin irritation tests are currently done on a skin-on-chip like devices. Obviously, they're not complete, but we are fair enough that we are able to develop these keratinized layer of skin and we can test at least for cytotoxicity. The regulatory aspect of an organ on chip would be that any regulatory experiments will require cross validations. So currently the main point of discussion is that who will cross validate, what are the companies who will independently validate these organ on chip devices. Two, the quality control. So for example, when we talk of devices in India, we have ISO certification. Then we have for all the laboratory tests, we have the certification from NABL. What will be for the organ on chip? We are still debating it, but currently they will all fall under the GLP, Good Laboratory Practice Guidelines, through which these organ on chips will be regulated. Ethical aspects, yes, all the aspects of the human, suppose if you are using human cells, especially primary cells deriving from the patients, then all human ethical committee principle, human ethics of human experiments will come into principle, where you'll have to take signed consents if you're doing personalized organ on chips or personalized organoids or steroids. As far as you're using cell lines, you don't need ethical approval, but you will have regulatory approvals from your institute biosafety committees, because these are the ones who will look at the biosafety aspects of it. So currently, as of today, from the industry perspective, it is going to be GLP and biosafety which will take care of it.
So, thank you. We are extremely grateful to you and for your eloquent and insightful address. Your remarks have truly left a lasting impression on us, and we appreciate the invaluable knowledge that you have imparted during your presentation today. Now, we would like to present the Certificate of Appreciation as a token of our gratitude. Thank you.
Thank you so much.
Thank you very much, Dr. Deepak. It was indeed an enlightening session. Thank you very much.
Thank you.
Thank you so much, Dr. Deepak. And I must say you are a passionate researcher. And we would like to interact with you more and more on many, many research platforms.
Sure. I'll be happy to come over, and let me know if there's someday if I can come over and we can.
Sure, definitely, sir.
Great.
Thank you. Thank you very much.
Bye, everyone. Have a nice day and a nice scientific interaction.
Thank you.
Thank you very much.
Thank you, sir.
Thank you, sir.
Wish you a great weekend.
Thank you. Wish you the same, and all the best to the presenters for the day.
Yeah, thank you.
Thank you.
Bye.
It is with great pleasure and honor that I extend a warm welcome to our second keynote speaker, Dr. Srinivas Savle. Dr. Savle is a distinguished leader in the pharmaceutical and healthcare sector with over 23 years of experience spanning drug research, development, regulatory compliance, and the innovation ecosystem. As the CEO of AIC LMCP Foundation and Atal Incubation Center, hosted by LM College of Pharmacy in Ahmedabad and supported by Niti Aayog, he spearheads initiative in pharmaceutical and healthcare innovation.
Dr. Savle holds a PhD in pharmaceutical sciences from Gujarat University and has a prolific career including leadership roles at Torrent Pharmaceuticals, Syngene International, and the Torrent Research Center. He specializes in regulated bioanalysis, biopharmaceutics, early clinical development, GXP compliance, electronic data workflows, and vendor qualification, among others, supporting CROs and pharmaceutical organizations in achieving excellence in these domains.
Actively involved in academia and the innovation ecosystem, he has contributed as an adjunct professor, mentor, and committee member at LMCP. His professional affiliations are extensive, including leadership roles in the Indian Pharmaceutical Association in Gujarat branch, the Society for Pharmaceutical Dissolution Science, and international organizations such as the AAPS and the Global Analysis Bioconsortium.
Dr. Savle's contributions have been recognized with prestigious awards, including the BioContribute Award and the Best Research Paper Award by the IPA. As a prolific scholar, he has authored several publications and book chapters and has been an invited speaker at numerous national and international conferences. His steadfast dedication to advancing pharmaceutical sciences and fostering innovation has made him a prominent figure in the global healthcare ecosystem.
We now look forward to the insightful perspective that Dr. Srinivas Savle will share with us. We are confident that his words will both enlighten and inspire us. Please join us in offering a warm and sincere welcome to Dr. Srinivas Savle.
I would like to add few words.
Thank you.
So, Dr. Savle, good morning, and thank you very much for accepting our invitation and being with us today. And what a wonderful, fully placed your talk after talking about research and talking about the applied research, applied science, and now we are at how to convert those ideas into reality. So here we have the CEO of Atal Incubation, who has handled multiple startups and guided them very successfully. And I'm sure on his behalf, I can tell my students and all the participants, if whenever they happen to visit Ahmedabad, you must visit Ahmedabad Incubation Center at LM College of Pharmacy. I'm sure he would be more than happy to take you on a round, and wonderful to visit that center. So thank you, Dr. Savle, for being with us. Really appreciate your time today.
Yeah, so nice of you, Dr. Munira. It's always a pleasure kind of participating in such forum. I hope I am audible, and I'll try and share my slide. Just let me know if you can share, see the slides.
So you're audible, but the slides are not visible.
Yeah,
I'm just sharing. Just
okay.
Yes. Yes, sir. It is visible.
So we're all set. At the outset, let me thank Dr. Munira for cordial invitation, and of course, Dr. Sujata and the entire team of SRC, the 5th edition. I had a chance to participate earlier edition of this event, and it's really such an encouraging environment that this event creates for the students to think differently. So this year again, the theme is innovations for better health. So I think I'm trying to touch upon the current trends, innovation trends in pharma and healthcare sector. So that, and I'll also give you some case studies how the problems students like you have identified and translated into workable solutions that I've seen markets. So I always start my talk on innovation with this very favorite slide of mine. So when you look, I'll go back to the first box. So you see that nowadays we have a lot of information available. And these dots, like it's a wealth of information, and there are many resources through which you can get the information at the fingertips. But important is how to derive the meaning out of this information. So what is the meaningful information? So experience teaches us how to derive the right meaning from the given information or the resources that we have. But the most interesting thing is how you connect these dots of information translates into your creativity. So it's the same information, but you think differently and use it differently. So that's your creativity. And believe me, the most important skill in the future will be the ability to connect these dots in your own way. That's how it makes you stand different from the mass. But it is not that straightforward. So there are certain barriers when we want to translate our creative instincts into new products which will see market or solve certain problems, existing problems. So as we know that creativity actually is the expression of skills, curiosity, imagination, and motivation together. So it's bundled together to come up with a creative idea. But this creativity needs to be honed. So you have to have right mentoring support, duration of these creative thoughts and ideas so that it can be translated into innovation. And if the innovation further needs to be translated into the products or the workable solutions, you definitely need something called as the economic environment. Now, when I say economic environment, as Dr. Deepak was just mentioning about how it will be a long and arduous journey to take this organ shape for the placental model, what he was talking about, to a workable laboratory solution for in vitro simulation of the placenta per se. So you need a lot of funds to invest into, to bring all the resources to study. So as he said, his team has reached to a stage where the idea they could develop a proof of concept. And now from there on, taking it to a usable solution, you really need a lot of economic support. Now, when I say not only funding for investing into the work that you are doing, but also it should have market potential so that you can reap benefits of all the efforts that you have put into this developing this particular solution. So it is very important to undermine this little difference when we talk about invention and innovation. So invention is anything which is done for the first time. So be it a creation of a product or introducing a new process for the first time. But innovation is something, it is related to the improvement on the existing knowledge or improvement on or a significant value addition to the existing product, process, or service. So when we talk about innovations for health as a theme, so probably we'll not touch upon the discoveries per se, but we'll touch upon how some of the discoveries are getting translated to solve the problems of healthcare globally. So whenever we think of innovation, the innovative idea should be helpful to the people. It should save time. It should add to the convenience. It has to be cost effective, and it should ultimately improve the quality of life. So when we try to put this criteria to innovation, so there are broadly two types of innovations. One is demand pull innovation, and second is technology push innovation. So demand pull innovation is nothing but it originates from the intention to satisfy needs of the market. So the market is already there, but you need to innovate or add value to the existing solutions. Some examples you can see are like Uber, Solars, Zomatos, you have Tata 1mg or Pharmacies. It's not that there were no travelers or no patients. Only thing is now you are adding to their convenience through these innovations. And when it comes to technology push innovation, these innovations actually originate for R&D activities. As we were talking about organ on chip just few minutes back, we're also talking about it brings a competitive advantages provided by the new technologies. So something which is coming for the first time, like you have uh, Facebook, you're talking about 3D printing, a lot of technology these days we are kind of boggled with is artificial intelligence and machine learning. We have variables, cell and gene therapy. So these concepts were not there. They have they got newly developed, and they have been contributing to address some of the healthcare needs. So when it comes to innovation, So if I had to just give you a little theoretical aspect. So innovation process starts with problem identification. So it's very important that you identify what is the problem. And believe me, you are halfway done if you have done the problem identification correctly. So most of the times, the problem is not the real problem, but it's a perceived problem. And then we try to work on solution for a perceived problem, which actually is not asked for, or which is not required by the consumers or by the patients for that matter when we talk about healthcare. So once you identify the problem, you may have variety of solutions for the same problem. So you can have hundreds of different ways to tackle that problem. So it is very important that out of those 100 options, which is the one which probably will work. So you need to actually evaluate which idea may be feasible as converting into a prospective solution. So that's where now you start working on developing that innovation or actually building the solution in a very primitive way, which is like it's called as POC model or proof of concept model, and then you challenge that model and kind of validate that it works, and then you look at its first use and try to validate by doing it again and again and taking it to the relevant environment, and then you're confirmed that yes, this particular solution works for this problem, and then you actually test it into a real life situation, and then actually either whatever technology you have developed during the process you would probably like to commercialize, or you may want to kind of, it's called as technology transfer. So you can sell up the technology, make your own money out of that, and but whatever knowledge is that is getting generated through the process, from the business perspective, you protect your intellectual property as patents, trademarks, right, registrations. But then if you talk from the holistic point of view, so it's always the dissipation of the knowledge generated. It can be through the open source or through the intellectual property, the patents that we study. So ultimately, in the process of innovation, you are generating wealth of knowledge that can be shared with the relevant stakeholders. So it is actually empowerment. You can see that this is basically you are empowering people through innovation. And especially when we talk about developing technologies in the pharma and healthcare sector, so there is something called as technology readiness level or the maturity of your solution in the process. So broadly, there are 9 technology readiness levels bucketed into 3 different phases. So your early research happens in TRL 1, 2, and 3. So TRL 1 is where the basic principle is observed. Then you'll formulate the solution or the concept, and then you generate the experimental proof of concept. So whatever Dr. Deepak was showing us, that is actually at that stage. So it is at TRL level 3, where you are saying that the concept has been proven by the initial experimentations, and then you actually take it on the larger scale and validate that particular solution in the lab. And again, when we talk, there was a question on the preclinical models. So that's the relevant environment to humans. So that's why people started working on the animal models as the starting point. But you need to prove your solution. For example, if you want to see the drug works or not, you are definitely going to go through the effectiveness and safety aspects of it in the preclinical setting, and then move on to the humans. And in humans also, you are doing a variety of clinical trials, phase 1, 2, 3, 4. And ultimately, after phase 3, you are convinced or you say that it works in the right target population. And that's when the drug gets the marketing authorization. So when you see these TRLs, it is very important for us to understand that today's idea need to walk a long way before it sees the market. So one has to be prepared for walking this entire phase of maturity of the technology or the product that we are developing. So this is just I'm giving you some understanding. If somebody, one of you or some of you really want to take your idea to the next level. Otherwise, the research, I mean, you call this as Student Research Congress, right? So nowadays, the most of the people are thinking that the research, how can we make it as an applicable research? How can I see the translation of research into reality or the workable solutions? So that's the reason I'm giving some of these basic concepts or aspects. And believe me, globally, all the economies are thriving on innovation. There is something called as a Global Innovation Index, and I'll tell you where India stands. So this is the latest GII published in 2024, September 2024. So you'll be surprised that there is something called as the innovation inputs and innovation outputs. So these are the parameters on which 133 economies of the world have been rated. And you will be surprised to know that it is the Switzerland which tops these economies, not US. And being such a small country, they have a very good innovation output to input ratio. That's where they excel. And if you look at India, we stand at 39 positions in 2024. So from 2015 to 2024, we have jumped almost 41 places, which is an amazing achievement for a vast country like India. No doubt we are first as far as Global Innovation Index is concerned in the Central and Southern Asian region. But then there is a lot of scope for us to further improve upon. There is something similar called as India Innovation Index, which was published in 2021, where you can see that the states and union territories were separately bucketed, and you can see that the Karnataka remains the leader within India as far as the innovation index is concerned. But heartening graph, if you see on this right side, I'll try to pull the pointer. So you'll see that there is a steady increase in the number of patents that are being filed in India. And that's very encouraging. The last year's number were still much, much higher. So what it indicates is now the environment within India also is very conducive for innovation, or the environment is facilitating converting innovative ideas into realities. And especially for our sector, everything changed once we went through the phase of COVID, right? So since we have seen that March 2020 to March 2023, there has been a significant impact of COVID. And on May 5th, 2023, WHO declared this as no more emergency. And it's like it's no longer constitutes a public health emergency of international concern. So that's where now the pandemic turned into one of the routine infections, because we are now prepared to handle these inspections. So how it changed the entire way of looking at the solutions or the drug options, or you talk about delivery of healthcare to the patients. So in 2022, you will see that when COVID was at peak, you will see that COVID-19 vaccines, out of top 10 by sales, you will see that 3 were COVID vaccines. So 30% of the sale of medicines globally was related to COVID vaccines. But then as it would remain no more a global emergency, in 2023, we can see that it is the Keytruda which was leading the market, which is an anti-cancer monoclonal antibody. And you will see then there is an anticoagulant, apixaban, and again some of this adalimumab, which is for rheumatoid arthritis, became the third top-selling drug in the globe. And the new entrant was Ozempic, that is the semaglutide injection, which works for type 2 diabetes. But you'll be surprised that it is more taken for weight reduction or obesity solution as well. So the problems, their severities keep on changing with the time. And if you look at 2024, it is again Keytruda continues to be the top leader with respect to the global sales, and Ozempic has jumped to second position. So what it says is the disease burden, the patient burden, the demography of patients has been changing, and so the demand for certain solutions or certain drugs is changing over the time. Now, not only that, if you look at the emerging drugs in 2024, so you will see a mix of small molecules to very complex molecules to even AAV kind of therapy. So it's very important for us as a curious research students. We need to look at the trends. So depending on the health burden, we can decide on which way to work on. So those who are into those who are into discovering new drugs, they have different targets to work on. Those who are trying to work on better deliveries of drugs, so they have different strategies to work on the existing drugs, so on and so forth. So you'll see that this is a mix of monoclonal antibodies. I give you example of antibody drug conjugates, ADCs, like this one, and you also have the associated adenovirus based drug deliveries, which are like here you can see this is for treating the pediatric low grade glioma. So there are certain niche conditions where the different therapies are being developed. So as most of you must be aware of rare diseases, so there are certain work is happening for bringing solutions for such rare diseases. Like in this particular chart, you will see that there are many drugs which are being targeted for schizophrenia or cancer, but also there are drugs for hemophilia like hereditary conditions. So what it tells is where the innovation is going. So what are those disease burdens as the demography of diseases is changing, the thrust is changing for the pharma companies as well as drug discovery companies. So to bring such solutions to market, as you would have seen in COVID, we could get the vaccine in less than 1.5 years from discovery to market. But that is an unprecedented situation, so this is not the daily scenario. Now we know that any drug traditionally used to take 1 year to 50, I mean, up to 15 years to see the market, right, from the discovery. So the things, it is not only that process, but people have started innovating the way the discovery works, the way the clinical development works. So they try to reduce the time frame for certain activities within the drug development cycle. And it was very important to bring the cost also down. So there were a lot of technology interventions that were thought of within the research and development space itself. So you'll see in this slide, so we are already having augmented and virtual reality being deployed in clinical trials, where the clinical trials participants' experience is improved. So you have genetics and genomics that is being used for certain oncology therapies, where you need to identify the right responders. So it's not that one chemotherapy agent will work for all cancer patients. So it's very important you bring this understanding of genetics, genomics, and big data for actually stratifying the patients for whom a particular therapy would work. Then you have blockchain, cloud edge computing, variables, and sensors, which actually help you to capture the real world data for better trial design. And AI, but natural, this has become a common terminology, but there's a lot of data that is required to actually validate a particular AI model. So not only R&D, there have been a lot of innovations happening by deploying these technologies in patient-centric, agile, and self-learning pharma manufacturing as well as supply chain. So you have blockchains where you track and trace to understand what happens to the drug during the transit, right, leaving the manufacturing site and reaching to the patient. what is the fate of the drug. So 3D printing, but natural. Yesterday, Dr. Parijal was also mentioning about 3D printing of levetiracetam, right? So not only the drugs, even the precision medicine is being 3D printed. So this is coming in a big way, but this is with respect to this slide describes it more with respect to manufacturing and supply chain. So you'll see that there is a robotic process automation which is coming in fast to avoid failures of the batches. You have online platforms, you have cloud-based solutions, wearable sensors, and again, AI is also coming into improving efficiency of your processes. So how can I reduce the failures of the process? How can I also build reaching to the market, reaching to the patient, the time to reach to the patient, right? So you have a lot of inventory management tools, you have quality control and management tools, which are deploying this AI technology. Further, clinical trials, there have been a lot of advantages by deploying the technology. So you'll be surprised to see that AI, if you look at drug development process, AI has the biggest influence on the clinical trial phase so far. So it is helping to improve like the medication adherence of the patients. Not only that, it is also helping to match the right patient pool on which you need to design the trial. So you actually reduce the subject enrollment failure rates. So that is a big saving in terms of time, cost, and resources for any clinical trial sponsor. So you also have tools like you have video conferencing, wearables, mobiles, so you can really do global trials very effectively. So it is actually the new rather deployment of technology in the development path is something bringing the overall time drastically, reducing it drastically. You also have digital pills where you can have sensors, ingestible sensors and cameras, which will get into your body and keep monitoring the physiology, and you will have real-time understanding of what is happening uh, after a particular therapy. And also nanotechnology helping is very much helping in tracking the short and long-term treatment outcomes in real time. So clinical trial advancements have been going to be a really big boon to reduce the timeline from safety establishment to bringing the therapy to the patients. Not only that, when you talk about healthcare, patient is at the center, and there are a lot of patient-centric and personalized therapies are coming in. As we know, we see cell and gene therapies. We are also seeing individualized therapies. We also talk about life-saving therapies, where actually, no doubt you to go for personalization for very specific and effective therapies, but they are very costly, so they cannot be probably afforded by masses. So there are certain models of even reimbursing the value and patient outcome. So all these costly therapy, it's not that you just buy over the counter or like pay and get it, but there are reimbursement models that are being established globally where depending on the effectiveness of therapy, the patient will pay to the drug manufacturer. So that way, so this is just an operational tweak or innovation to make people use this personalized medicine. Now you talk about, as I was mentioning, performance-based contracts. So you are talking about CAR T cell therapy, but what if it doesn't work? All your millions of dollars you would have spent to get a treatment will get, it will go into waste. So if it works, then you pay. So that's the concept is coming in Europe. And when it comes to future trends, there are a lot of information tools that are also useful in our sector, pharma and healthcare sector. So yesterday also Dr. Pereza touched upon big data analytics. So this big data analytics is helping in a variety of phases. It's not only in the research and development, but also in business analytics. So understanding, getting information from non-traditional sources like social media and patient forums and converting it into the insights for physicians, enterprises, and also for the insurance company. So it's like the entire value chain, it can be benefited by using the big data analytics. No doubt, we already touched upon AI or digitized medicine, but also there are many mobile apps now, we know, when it comes to healthcare or health related apps. So a lot of technological interventions have been helping in one or the other way, and you have, it's not only the wearable devices, your smart watches and other things. Your smart rings are there available in the market now. So it is very important that what is the data that is being fetched by these devices and how it helps in making the decision on the healthcare. So nowadays, I mean, the days are not far when you have these wearable and IoT solutions, which will monitor you real time every time. So it's like everywhere care is something the consumer is looking for. So you have quantified cell body sensors. We also talk about virtual medicines. So there's something called as digital therapeutics or DTX that is also coming in big way. So the digital content also is working as medicine. So there is new guideline that has come up from US FDA, which talks about SAMD, that is software as a medical device. So there are several advancements that are happening, which is actually coming to improve the health outcomes of the patients. And of course, augmented reality, virtual reality are being used in certain conditions where the patient outcome is much effective. And broadly, the three buckets which need to be watched, they are already in place. So I'm giving three kind of problems. So, for example, there is a critical staff shortage in US, and it is like projected that by 2030, almost 140,000 physicians and 63,000 nurses uh, will be short in the healthcare system with the growing aging population of the country. So people have immediately started working about advanced robotic solutions for patient care. So now you will see that you will have these robotic solutions which will cater to supporting the patients, hospital operations, logistics, lab automation, as well as remote care delivery. There's another thing where we are trying to have early diagnosis. So AI is coming big way in precisely evaluating the patient's symptoms even before they appear actually. So it's going to be a very effective way of, probably we can save many lives if we have early detection in most of the cases. So AI enabled testing and screening solutions are in a place these days. On the therapeutic segment, there are traditionally undruggable proteins which are found in genetic disorders, cancers, and cardiovascular diseases. So all the genetic disorders are hereditary conditions. So one class of therapeutics, that is RNA therapeutics, that is coming in big way. We already saw mRNA-based vaccines, but like you have RNA interference, you have antisense oligonucleotides that are coming in big way, and also mRNA, but natural. So these are some of the trends one need to watch for, and there are like hundreds and hundreds of technologies being evaluated for actually improving the healthcare outcomes. So some of them I just list here. So you have, of course, this particular graph you will see here. This talks about the technology readiness level at different colors and the specific technology. So the brown ones are TRL1, that is at the idea stage or initial research, and the dark greens are actually the technology readiness level 9 that is there in the market. So you'll see the long list here: 3D bioprinting, antimicrobial packaging, your bioelectronic medicine. You're talking about biomimetic devices, biomanufacturing. Also, CRISPR has been another very good technology tool that is available. We talk about digital twins and synthetic biological engineering. You have Not only that, nowadays we are talking about embedded health monitoring sensors. You have nano manufacturing, you have portable medical devices, self-healing materials, so on and so forth, virus-proof fabrics. So people are working on variety of technologies which will be helpful in improving the health outcomes for the patients. So you may have variety of such ideas, and probably it is you want to take it from TRL1 to TRL9. So there's an excellent ecosystem that is available in our country. They are called as incubation centers. So just like in our microbiology practicals, you incubate the microorganisms to grow. So you provide conducive environment, be it the nutrients, the temperature, right? Everything we provide so that the microbes grow. Similarly, the idea, if that needs to be matured to a sellable product, so the incubation centers take care of all that support for such brilliant ideas. So AICLMCB Foundation, which I represent, is one such incubation center, and there are many in our country. And this is more, I'm covering this just to create awareness that if you have certain interesting challenges, interesting innovative ideas, which you think that can be useful for improving health outcomes, you should take help of such incubation centers. And it's not just the modern medicine, you have alternate medicine, you have now drug delivery systems, the technologies. There are plethora of opportunities. Like I remember Dr. Munira also talking about the there are like sky's the limit. Many opportunities are there where we can actually you look at those small little problems and try to solve them. They may convert into a big solution. So this is just I'm giving you those glimpses of some of those upcoming areas where people are working with novel solutions. And these incubation centers like ours, we actually facilitate this through domain expertise, networking, mentoring. So we have collaborations with industries, research institutes, and even institutional collaboration go a long way. So we also have collaboration with uh, BNCP. So likewise, there are many such institutes who can bring in their expertise to support some of these innovators. And of course, not only this, but also support for understanding the legalities, the compliances, the protection of intellectual property. We talk about building the business strategies or marketing strategies and so on and so forth. So we have right resources who can guide you for developing the business model in a holistic way. And of course, funding. So when it comes to funding, you get government grants, you get angel investors putting in your money, you have venture funds which come and pump in money when you are at a advanced stage of your TRL. And so this is a holistic support that is being created. So far we have been supporting 55 startups at variety different stages of their development, and they really could create a lot of value or intellectual property. And we also are promoting women entrepreneurs. So we have almost 50% of, 40 to 50% startups are with women co-founders. And pharma and healthcare sector is, as you know, it is regulatory intensive, it is time intensive as well as cost intensive or the funds intensive. So you will not see that a lot of employment is generated in a short time. So still we have about 300 jobs being created by these startups over the last 5 odd years. So when I said collaborations, it is very important to have the ecosystem developed. So you have corporate partnership, academic partnership, and ecosystem partnerships. They bring a lot of insights for variety of requirements of the startup for taking the idea to the next level. So you need some business support, you need some uh, geographic expansion support, you want to export to other countries. So we have some such collaborators who can help you in geographic expansion. So I'll quickly give you a few case studies so that you can relate better. So how innovation can be translated into an enterprise. So the first case is, so this is again a team of I'm not, I can say team, but two engineering students walked in our incubation center 4.5 years back. So they wanted to solve a problem of medication adherence. So what they developed till now is a smart pill box which has reminders. So you have an app and a device for storing the medications in this device. The device also has alerts, physical, audio, as well as readable alerts. And mobile also gets alerts once there is a time to take a particular medication. So you have a particular compartment, which medicine to be taken at what time. And the beauty of this combination is it's a Wi-Fi enabled and you can monitor the treatment adherence sitting in any part of the world. The best part of this application, this was thought more for healthcare settings, but even pharma companies have found this as a best solution in even clinical trials. So they can replace the patient diaries and other stuff, and you get online real-time adherence report from this device. So this is like already they have raised funding about a crore and a half, and they are already in the market. So this is here we could add value to their concept and connecting and giving right ecosystem support to grow to this level. Another startup is into artificial intelligence based treatment response and assessment, especially in respiratory diseases as well as cancers. So this is a relatively mature startup, but they were looking for right connects to validate, clinically validate their idea. So we could connect them to the hospitals, the pulmonologists, the oncologists, where they could actually validate their AI solution. And they have got like they started in COVID. In COVID, they could develop this solution which has 13 different respiratory elements identified, and they had a predictability of about 96%. So it was a wonderful solution. Now they have been selected as one of the startup contenders in Government of India's top TV program. So this solution is scalable. It can be deployed in the uh, government, I mean public healthcare system. So that's where the evaluation is being done. And recently they were tied up with, as we were talking about AI-enabled diagnostics. So they tied up with an X-ray manufacturer and they have come up with AI-enabled X-ray machines. So which actually can immediately give you the diagnostic insights by using these AI-enabled X-rays. This third particular example I would like to give is this is a small matchbox like device which actually monitors your close environment, the micro environment. So this has 9 different sensors. Like you can see here, you have motion, noise, temperature, you have ultraviolet light, relative humidity and particulate matters, PM1, PM2.5, and PM10. So what this device helps is it collects the environmental parameters, and based on these environmental parameters, when the user gives the health experience to the device, the machine learns and it alerts you next time for what precautions to be taken if you are getting exposed to a similar environment. So this is like a wonderful solution. Like if we today talk about pollution and the kind of diseases because of pollution, this is going to be a very it's a preventive health solution for many of us. Then this startup has found the problem of the inefficiency in retail pharmacies. So what they did is they created a SaaS-based solution. So it's a cloud-based platform where the eVital gives subscription to the retail pharmacies, and they can automate the entire workflow of retail pharmacy, right from identifying the distributors till delivering the medicine to the patient. So this was one of the fastest growing startups that we have seen. They faced 18 crores of funding, and they are now more than 100 cities in India in the span of 2.5 years. So identifying the right problem is very important, and then you build the solution which is scalable. Now, this is another startup which has worked on advanced therapeutic modality like cell therapy. So they have worked on autologous cell therapy for diabetic foot ulcer and chronic wounds. So the initial clinical trial that they did, so it's just a single topical application, which is of course the cells, the autologous cells. It has they have seen that on one application within 6 weeks there is almost 50% wound closure, and in next 6 weeks follow up, the wound has closed in more than 88% patients. So it's a very promising cell therapy. Now they have set up their own manufacturing unit for GMP production for the larger clinical trial that they want to perform for marketing authorization. There's another startup we were talking about novel deliveries yesterday. Dr. Pirzad mentioned about Cipla's achievement of bringing inhaled insulin, right? So this startup has the oral delivery of peptides. They have a platform technology to give peptides by oral route. And what we are talking about, Ozempic injection. So they have oral Ozempic. So they have semaglutide already proven in the clinical trials. So we could help them by giving the laboratory support for their product development, their R&D work, and in like last just a month back, they also established their own bigger R&D facility, which they can now have variety of products that are being produced with using the same technology. There is one startup which works on artificial sensory evaluation. It's electronic tongue mainly for taste, odor, and flavor analysis. So whatever test masking attempts we do for oral delivery of variety of formulations, so you can, instead of going for the certified testers, you can avoid this clinical exposure and you can do it in the lab. There is another startup which has identified that import dependency is a big problem as far as APIs are concerned. But it's not that just making in India the same drug. They have identified niche APIs and intermediates to make cost-effective greener. So you remove all the hazardous solvents from the processor, and a sustainable supply is what they are targeting. And the first two prototypes they have, processes they have, they could reduce the route of synthesis by 7 steps, from 9 steps to 2 steps synthesis. They have, so you can imagine the savings on the raw material that is required for synthesizing that particular drug. And recently, they also built a library of novel chemical entities for one of the universities in Spain. And so it's like the process innovation is something they have brought in. Now, this is another interesting startup. We see cosmetics, but if you talk about care, so it's more into skin and hair care. So they have all the products which are based on the Nigella sativa, so which is rich in thymoquinones. So now we could provide them ecosystem. Now they have actually kind of developed other geographies. So they have, in last 4 months, they have gone into Middle East and they have also expanded into Australia. On the menstrual health. So they make menstrual cups. Now there are many menstrual cups available in the market, but they have got the design patent for these cups in terms of removing the pores in the cups and replacing by the rich. So which helps actually for better cleaning, and there won't be chances of infection because of the pores. There's one startup which works on functional foods. So they have this freeze-dried vegetable and fruit mixes, that are juices which are freeze-dried. And without losing any of the functional properties of the contents, you can use this. So this is a simple alternative of the bottled juices. And of course, the combination, if you look, the combination of the products that are, the raw material selected has certain, you have, most of these are antioxidant blends and improve your immunological benefits. So this startup is helping only for the pets. So they are working on pet care solutions. But you will be surprised that they launched India's first pet sanitizer without alcohol. during the COVID time. So looking at the skin structure of the fur of the pets, they have about 30 odd different products suiting to variety of breeds of the pets with their skin structure, be it a single coat shampoo, double coat shampoo, they have paw balm and so on and so forth. So very innovative thought products for the pets. So these are some examples which I thought you can relate that how a problem can be identified and taken to the next level. And quickly I will go, this is my last slide. So what I wanted to show you here is there's a gentleman called Richard Watson, and he has developed this mind map of what are the mega trends and technologies in variety of sectors and how it will look like in 2050. So I'll try to zoom it. I am not sure if you can see it. To do some zooming. So you'll see there are a variety of sectors on this side. So out of that, I'm picking only health. And what his mind map suggests is you have what advancements are happening. So the triangles is something which is already there, right? So you have AI already in general practice and surgeries. 1 in 2 people are suffering from mental health problems. So this was seen in 2017, this mind map was made. Then he talks about gamification of healthcare. He talks about internet-based therapies, which digital therapies are already, we are seeing popping up. Doubling of dementia by 2050. So this mind map actually gives some problem statements if somebody wants to work in or somebody wants to get in. Then we talk about microbiome-based healthcare, so which we know that is already we are talking about microbiome, microbiota. We're talking about robotic surgeries, 3D printing of human organs. Just some time back, Dr. Deepak also was mentioning upon organ on chips. So you also have now 3D printed human organs. Growth of multi-resistant organisms. This is the, I think the very apt time where we are seeing there's a lot of need of awareness on the antimicrobial resistance, and every other organism we are seeing there is a development of resistance and we are short of the solutions. And there are so many other, like you may not imagine of this, talking about personal digital diagnostic devices, 3D, 3-parent pregnancies, growth of internet addiction, sperm-free reproduction. It talks about medical records being implanted in the human body, and 3D printing of drugs, we are already started seeing it. So it's very interesting piece of information which I think one can have a look at and study to understand what is expected in near future. So with this, I'll stop. And I don't know, you can see this. So if you have any innovative idea, in pharma and healthcare, you can reach out to us. Don't hesitate. These are the contact details. And thank you. I know we were supposed to, the entire session is delayed, so I am one of the contributors to that. Sorry for that.
Thank you, Dr. Srinivas. You have laid down the entire landscape for students to explore in the various areas of innovation and the idea bringing to reality. Thank you. Over to you, students.
Yes, thank you, sir. We are truly appreciative of the wisdom and clarity that you have brought to today's session. Your profound insights have deeply resonated with us, and we are grateful for the knowledge you have generously shared. As a gesture of our sincere regards, we are pleased to present you with this certificate of appreciation. Thank you.
Thank you.
Thank you, sir.
Thank you so much. I'm sure there won't be any questions because.
No, the figures that you mentioned are quite encouraging. Dr. Savle, the figures that you mentioned are quite encouraging, and it has motivated all of us to how to commercialize and translate our technology and our products. Thank you, Doctor.
Thank you so much.
Now that brings us to the end of today's session in the first half. We are deeply grateful to our distinguished speaker, Dr. Deepak Modi and Dr. Srinivas Savle, for sharing their expertise and inspiring us all.
Now, as we take a brief 15 minutes break, we will continue at 12:30 and reconvene for the online oral presentations. We sincerely thank our audience for the active participation so far.
I now kindly request that participants join their designated rooms using the allotted codes as their presentation names. The PhD presentation will be continued in the same room, and the link for Innovative Idea Challenge presentation is given in the chat box.
Participants, please note that any display of personal names or affiliations will lead to disqualification. For other attendees, please proceed to your respective rooms as well.
Respected judges, I kindly ask you to join the rooms as outlined in the mail. Should any issue arise, please inform us immediately. Once again, thank you for your cooperation and valuable participation.
Thank you.
Thank you.
ठीक है ठीक ठीक
afternoon to one and all present here. I, Mr. Parth Bharkar,
And I, Ms. Himani Sangvi,
am delighted to welcome you all to the PhD session of the 5th Student Research Congress, organized by SVKM's Dr. Bhanuben Nanavati College of Pharmacy, co-hosted by the University of Mumbai, along with our esteemed industry partners, ACG Worldwide and SciTech Center.
never stop fighting until you arrive at your destined place. That is, the unique you. Have an aim in life, continuously acquire knowledge, work hard, and have perseverance to realize the great life. This powerful quote by Dr. APJ Abdul Kalam perfectly encapsulates the spirit of today's event.
The oral presentation competition for the PhD category serves as a platform for researchers at the pinnacle of academic pursuit to showcase their research and groundbreaking discoveries. A PhD journey is not just about achieving milestones, but about relentless inquiry, countless hours of effort, and an unwavering commitment to expanding the horizons of knowledge.
Today, we come together to honor the imagination, creativity, and sheer determination that define the path of every dedicated PhD scholar. Their work represents not just innovation, but also the resilience to overcome challenges and the passion to contribute meaningfully to their fields of study.
I am privileged to welcome our distinguished judges whose expertise and valuable insights will not only evaluate but also inspire our scholars, encouraging them to reach greater heights in their remarkable research journeys.
The judges for the PhD category are Dr. Anil Gaikwad, Dr. Dhanraj Thopchom, Dr. Atul Shirje.
I would now like to introduce them. Dr. Anil Gaikwad is a professor at the Department of Pharmacy, Birla Institute of Technology and Science, Pilani. He did his master's and PhD from the Department of Pharmacology and Toxicology, Naipur, SAS Nagar. He was awarded a doctoral sandwich fellowship from DAAD during his doctoral studies. He visited reputed overseas institutes as a visiting scientist in the Department of Medicine, Nephrology, Albert Einstein College of Medicine, New York, USA, and Nephrological Center, Medicinage Polyclinic, Ludwig Maximilians University, Munich, Germany, in 2010 and 2008, respectively. His research grants are from SERB, UGC, DBT, ICMR, and CSIR. To date, he has provided essential and novel evidence on histone post-translational modifications and the conventional and non-conventional axis of the renin-angiotensin system in developing diabetic kidney diseases.
He has contributed to several book chapters published by Elsevier and has 100 peer-reviewed research and review publications published in reputed international journals such as Cardiovascular Research, Pharmacological Research, Drug Discovery Today, British Journal of Pharmacology, BBA, Molecular Basis of Disease, Food and Function. He has supervised 1 PDF and 11 PhD students and at present is guiding 1 RA and 9 PhD students. He is also an associate editor for Frontiers in Endocrinology Journal, Frontiers in Pharmacology, Renal Pharmacology, and BMC Pharmacology and Toxicology Journal. He also served as head of the Pharmacy Department and Associate Dean of the Practice School Division at BITS Pilani. Thank you for your gracious presence and for dedicating your time to this gathering.
Today, we also have with us Dr. Thopchom. Dr. Thopchom is a distinguished scientist currently serving as the Deputy Director of the Regional Facilitation Center, Northeastern Region at CSIR NEIST, Jorhat, Assam. With expertise in free radical research, radiation biology, conservation, and bioprospection, he has made significant contribution to these fields. Previously, Dr. Thopchom held the position of Dean at the College of Biotechnology, FGI College of Agricultural Sciences, and served as a senior scientist at the Ethno-Medicinal Research Center, where he facilitated the filing of 2 patents, one of which has been successfully commercialized.
His notable achievements include discovering and naming a new orchid species, Cymbidium hengbungates, and pioneering India's first microhydel solar hybrid power plant capable of generating 52 kilowatt-hour of energy year-round. Additionally, Dr. Thokchom introduced mass-scale lemongrass cultivation in Manipur for essential oil extraction, collaborating with CSIR CIMAP under the National Aroma Mission.
Dr. Thokchom has established collaborations with leading national laboratories such as CSIR CIMAP, CSIR CDRI, and CSIR NBRI. And his research has been featured in prestigious journals like Radiation and Environmental Biophysics, European Journal of Medicinal Chemistry, and Free Radical Biology and Medicine. His innovative work and commitment to science has significantly advanced the fields of conservation and sustainable development. Thank you for honoring us with your valuable time and being a part of this occasion.
I would also like to introduce Dr. Atul Sherje. Dr. Atul is a seasoned academic and researcher with 19 years of teaching experience in pharmaceutical sciences. He is currently the head of the Quality Assurance Department at Dr. Bhanuben Nanavati College of Pharmacy, Mumbai. His expertise includes nanocarrier-based drug delivery systems, analytical method development, and validation, with a focus on enhancing solubility and bioavailability of poorly soluble drugs.
Dr. Sherje completed his PhD in pharmaceutical sciences in 2016 and has guided over 25 postgraduate students in their research projects. He has authored numerous publications with an H index of 21, I-10 index of 31, and over 2,000 citations, including articles in high-impact journals such as Carbohydrate Polymers. His research is supported by multiple grants and is recognized among the top 2% scientists in the world by Stanford University. Dr. Sherje has also received several accolades, including the BNCP Shining Star Award 2023 and international travel grants for presenting his research globally. In addition to his scientific contributions, he has actively participated in syllabus design, quality improvement programs, and conferences in pharmaceutical education and research. Thank you for your generously dedicating your time and gracing us with your presence at this event.
Before we begin, I request all the participants to make note of the instructions carefully.
I'll just take a quick 1 minute to thank Dr. Dhanraj and Dr. Anil for graciously accepting our invitation. On behalf of my entire SRC team and the Institute, BNCP, and the management, we sincerely thank your time today and your efforts. I'm sure your inputs and your presence will definitely inspire the participants as well as the audience in taking up science as their profession and do much better in their career. So thank you once again, sir, Dr. Anil and Dr. Dhanraj, for being with us today. Thank you very much, sir. And I must share here that this is our 5th SRC. And we are very happy to share that we have the presentation or the audience from 25 states of India. Over 100 institutes have taken part, and we had 249 abstracts, out of which we have shortlisted only 49 abstracts in different category of UG, PG, PhD, and IIC. So definitely this conference, this Congress has shown the path ahead to all those students and researchers, those who have taken part, and where the judges are playing very important role. So thank you very much. In spite of your busy schedule today, Saturday, you are spending your valuable time with us, and we sincerely appreciate that, sir. Thank you very much.
Thank you.
Over to you, students.
Yes, thank you, ma'am. Always remember, the foundation of every successful presentation lies in four key pillars: collaboration, innovation, dedication, and preparation. Before we begin, I request all the participants to make note of instructions carefully. The presenters have to rename themselves as per the allotted codes. The presenters are requested to stay online, switch on the camera when the code is called out. Pre-recorded video will be presented on the screen. Participants cannot reveal their names or any affiliations. Each participant is requested to stay alert for their roll call. All the participants should be present for the session. All members from the group are allowed to answer during the Q&A session. The duration for the Q&A session is limited to 5 minutes.
With this note and the permission of our respected judges, let's begin with our oral presentation competition. As all our participants and audience are eagerly waiting to present and learn about the new, unique, and interesting ideas that will be present here. Also, I kindly request the judges to confirm that they have received the scorecard.
Yes, we have received.
So now I would like to call upon code number PhD01 to be ready, and I would request PhD01 to kindly switch on the camera and confirm their presence. Code number PhD01.
Yes, I am there, ma'am.
Yeah, kindly switch on your camera. Yes, yes.
Thank you.
Yes, thank you. In your video now.
The video is inaudible.
Just a minute, I think there's some technical issue. We'll look into it.
There is no audio yet. Please look into.
Sir, please give us a minute. We are checking it, sir.
Sure, sure.
Sorry, we are facing some problems with PhD01. Can we start with PhD02?
Yes, you can start the next.
PhD02, please open your camera and confirm your presence.
PhD code 02. Kindly switch on your camera. Yes.
Hello, am I audible to all of you?
Yes.
Yes.
Okay.
We'll play your video now.
Okay.
It's the same issue with this also. Not able to hear.
Good evening, all of you. Today here I am discussing about my research, design and synthesis of epidermal growth factor receptor tyrosine kinase inhibitor to overcome the resistance problem in the non-small cell lung cancer. What is non-small cell lung cancer? Lung cancer is the second most common cancer for the both men and women according to the statistics reported by the American Lung Association. Lung cancer comprises two types of the cancer, small cell lung cancer, which having the 15% overall the lung cancer, and second one is the non-small cell lung cancer, which having the 85% of the total counting of the lung cancer. The differentiation between the small cell lung cancer and non-small cell lung cancer is based upon their cancerous type. Small cell lung cancer having the less type of adaptation, while in the case of the non-small cell lung cancer is having the more type of the adaptation or is more aggressively. If it happen in any person, it can grow very drastically. Non-small cell lung cancer also comprising the four subtypes, including large cell carcinoma, squamous cell carcinoma, adenocarcinoma, and other types of the specified of cancer. When this lung cancer is detected, they have only 5 year survival rate after the detection. When we discussing about the victims of the lung cancer, we observe that one of our Bollywood actor Sanjay Dutt having this type of the cancer. Also, one of our batsmen, Indian cricket team batsman Yuvraj Singh, also this type of the cancer. These are the two well-known personality is the victim of the lung cancer. Now we will discuss about how this lung cancer happen in our body. Before discussing about the cancer, we will discuss about how our normal body cell function is performing. When we discuss about the cell growth, cell proliferation is the mainly responsible for the EGFR, epidermal growth factor receptor tyrosine kinase. EGFR containing extracellular domain, transmembrane domain, and intracellular domain. Whenever any type of the epidermal growth factor bound to the extracellular domain, it initiates triggering to the intracellular domain. When trigger initiated here in the intracellular domain, ATP is bind to the tyrosine kinase, or when this bind, there is a phosphorylation, or due to this phosphorylation, different secondary messenger of is initiated, which including RAS pathway, RAF pathway, ERK pathway, MEK pathway. Due to the initiation of this pathway, it generate it generate protein synthesis. This protein synthesis is responsible for cell proliferation, cell differentiation, and apoptosis. This is the normal procedure where EGFR tyrosine kinase is working. But in the case of the cancer, if any of the mutation happening in this domain, it initiate irregular or irresponsive triggering to the intracellular domain. Due to this irregular triggering initiated the non irregular secondary messenger. Due to this irregular secondary message, uncontrolled growth of protein synthesis is happening over here. Due to this uncontrolled protein synthesis is responsible for the uncontrolled growth of the cell, which may we can say that cancerous cell. This is the overall overview of the lung cancer, how it is happening because of the EGFR. This overexpression of EGFR is mainly responsible for in the lung cancer. So due to the treatment of this lung cancer, in 2004, the first generation EGFR tyrosine kinase inhibitor known as erlotinib and gefitinib has been come into the market. This drug containing quinazoline myotis in their scaffold. Okay, we can see that here the quinazoline scaffold in their structure is the erlotinib structure, is the gefitinib structure. Both this first generation EGFR inhibitor bind to the receptor for protein and and block the ATP binding. Due to this ATP binding, the phosphorylation is inhibited. Due to this inhibition of the phosphorylation, it block the EGFR pathway. Due to this, we can control the growth of the cell. But after the some time of the after the some time of this use of this drug, the drug is they make mutated by the Balkera amino group is replaced by threonine. Due to this, this drug is irresponsive for the treatment. That's why second generation EGFR tyrosine kinase drug has been come into the market, which is known as apatinib, canatinib. Is also drug containing the quinazoline in their structure, but also simultaneously also contain the Michael acceptor in their structure. This Michael acceptor bind to the solve the problem of the first generation EGFR drug, but the problem in the second generation is that they're having the same affinity to the wild type EGFR. Due to this side effect, number of the skin related and diarrhea related side effect has been observed due to the use of his drug, second generation drug. That's why third generation drug has been coming to the market, which containing the Michael acceptor, also containing the pyrimidine ring instead of the quinazoline ring. This third generation drug known as osimertinib first get the approval from US FDA in 2016 for the treatment of non-small cell lung cancer. But after the some times of the use of this drug, it also create a mutation in the cysteine 797 residue where say cysteine is replaced by serine. Due to this replacement, this drug is irresponsive for the treatment. That's why there is a need of new generation of EGFR inhibitor to treat this type of the cancer. That's why in my research, these are the roadmap where we synthesize the molecules for the treatment of this non-small cell lung cancer. In the roadmap, first we have designed the compound, then synthesized in the lab. They're characterized by IR, NMR, mass spectroscopy, then evaluated for their anti-cancer activity. Then we go for the molecular dogging and dynamic study, and then next for the patenting and publication. The rationale for the designing of my compound is based upon U-shape strategy. When we observe all the molecules which are reported in the market having the U-shape strategy, U-shape structure, 3D structure, that's why we synthesize our molecule like a U-shape. We can here you can see that the molecule having contained that 4-amino 2-oxidisubstituted pyrimidine have the U-shape strategy. Based on the U-shape strategy, we synthesize number of the molecule by using this reaction. These are the reaction used for the synthesis of the molecules. Where 2,4-dichloropyrimidine and different types of amino containing molecule has been used. By this reaction, we synthesize number of the molecules. These are the molecules 58 to 98 the molecules. These are the structures of the molecules. After the synthesis, we did check the biological activity of the compound on the NCI H1975 compound and A549 cell line. And also we observe the selectivity index of the compound, whether this compound is mutant selective or whether this compound in the wild type is selective. We found that our compound number 60 having the 3 times more mutant selective than the wild type cell line. These are the activity of my compound. We can see that. Then we check the compound activity on the enzyme. These are the enzyme assay. We found that compound number 60 and compound number 90 having the more potent activity as compared to the standard drug. Then we observe the structural activity relationship. These are the structural activity relationship we observed from our biological activity. We observed that the meta substitution is more responsible for the activity as compared to the para substitution. Then last we did the blocking study of my compound to check whether this compound is reversible or irreversible. Then we found that this compound is causing covalent bonding with this cysteine 797 residue, and says proved that this compound is irreversible in nature. This is my overall of the research. Thank you.
Invite the judges. I now invite the judges to ask their questions to the respective presenter.
Yeah, I have one question. Like, you have shown this enzyme activity, right? Can you hear me?
Yes, sir. Yes.
Yeah, which enzyme you have used? You have not clearly mentioned over on the slide. Can you get my question?
Yes, sir. Yes, sir.
Yeah,
There is
which
two type
enzyme?
of two type of enzyme is there, sir. Double mutant EGFR enzyme is there, and the wild type EGFR is there, sir.
Okay. Is it there on the slide?
Yes, sir. Yes, it's
Okay.
given in the slides.
Yeah, yeah.
How do you get selectivity? What is that
Selectivity.
selectivity means what? What do you want to check?
Selectivity is mean in the cancer treatment, sir. There is a major problem is the selectivity. The compound which are acting on the mutant cell line, they are also acting on the wild type cell. That's why there is a problem in the selectivity. For that one, we observe that we check the compound activity on the wild type cell and the mutant cell. By comparing the IC50 value of the wild type and the mutant, we got the selectivity. Means is the ratio
What is
of the
the
IC50
difference?
value
What is the
divided by the
difference?
mutant IC50.
Difference between wild type and the mutant type.
Yes, sir. Wild type
And
cell
how
and the
did
mutant.
you bring the selectivity in the compounds? Why not it is towards more other tyrosine kinases?
I didn't get what exactly what.
Yeah, I'm saying why it is selective towards only this tyrosine kinase. There may be multiple tyrosine kinases also. The compounds which we have synthesized. How do you
Okay.
bring that? So why? Did you check any other tyrosine kinases also?
No, no, sir. No. Our more focus is that EGFR tyrosine kinase. That's why, sir, we check the activity on the EGFR kinase. But rest of the kinase we didn't check in our lab, sir, because of the lack of the facilities to available.
What are the current side effects which are associated, challenges with the tyrosine kinase?
The major challenging part in the tyrosine kinase is that the selectivity, sir. The compound having the more activity on
Adverse effect. Clinically, what are the problems associated with these?
in my research, we observed that there is a major problem is that side effects of the compound. There are many of the compound which are acting on the wild type cell line. That's why due to the acting on the wild type, they have a significant side effect like skin rashes, diarrhea, and gastrointestinal tract effects, sir.
How do your molecules will avoid those side effects?
Because due to the structural features of the binding cavity of the EGFR, sir.
Have you predicted the toxicity? Have you predicted?
Yes, sir. We predicted the toxicity. A different type of for my research work, we also check the compound on the HEPG2 cell line, sir. So, lung cancer HEPG2 cell line for the cytotoxicity of the compound, sir.
Okay.
Thank you, sir.
Okay, I have just one question. Like in the introduction, I mentioned that the first, second, and third generation, one of the common problem associated with those drugs is drug resistance, right?
Yes, sir.
So, what is the probability of overcoming those limitations with your synthesized molecule? Because that is one of the challenges in the current cancer therapy.
Yes, sir. There may be the challenges upcoming for the mutation.
the major problem in the cancer is that the mutation. When the drug is continuously taking for the long time, our body, our cells are adapted to mutation, which can convert the one amino acid to the another amino acid. Due to this mutation, our drugs can be irresponsive in this case.
Okay, all right. Okay, so that is all from my side.
Thank you, sir.
Are there any other questions?
Thank you so much, judges, for your valuable questions.
We will now go ahead with the presentation of PH01. PH01, please turn on your camera to confirm your presence here. Yeah. We will now present your video.
A very warm regards to one and all present here. My presentation ID is PhD01 and my topic is Morphological Dental Features. It's a biological identity card, a study of genial population of North India. These will be my contents for today's presentation. So let's start with the introduction. So as we all know, that the teeth are the hardest part of the human body. Being the hardest part of the human body, teeth resist various types of disasters, mass murder, fire, explosions, even the train accidents or even the airplane crashes. Even the teeth can resist the decomposed or unidentified bodies. So, moreover, the unavailability of other bodily identifying features make the teeth as the best suitable type of evidence for human identifications. Furthermore, the reliability of the dental evidence has been established by the Interpol by stating that the forensic odontology or the odontology is the primary method of human identification. Further, considering the dental morphology for biological identity of a population is a novel method that can scientifically identify a population. Therefore, however, literature lacks to recognize a dental morphology for identifying endogamous populations with high accuracy, which can help in forensic identifications, human identification, and anthropological studies. So, considering this literature gap, the present study was conducted with the aim to identify the unique dental morphological features in the genial population of North India. Further, for the data collection, the data was collected from the 207 participants, including 93 males and 114 females from the endogamous populations, that is the genial population, the North India, the state of Haryana. The age of the participants was between 21 years to 45 years. Further, the ethical approval for this study has been obtained from the Punjab University Institutional Ethical Committee, and moreover, the informed consent was also obtained from all the participants after explaining them the nature of the study, the purpose of the study, and all. For the data collection, dental casts were prepared from all the participants using the alginate impression material and then pouring with the dental stone. So this is an example of the dental cast which has been prepared by me, showing that various dental features. The symbol A or the point A is showing the shoveling of the incisors, point B is showing the supernumerary teeth, and point C is showing the H type of groove pattern in the molars. For the dental features, these are the dental features, morphological dental features, which were recorded on the dental cast, and then further these morphological features were mentioned in the pre-designed performer. For the statistical analysis, the data was first entered in the MS Excel, and then the statistical analysis was carried out in IBM SPSS version 20. For the data analysis or the statistical data analysis, frequency distribution of each morphological feature was accessed so that the association can be drawn between the morphological feature and the genial population, both for the male and the female of this population. And when we talk about the results, so these are the morphological dental features which show a frequency distribution of more than 50%. Uh, if I say more specifically, it is more than 60% or more than 70%. You can see that the hypocone and shovel-shaped incisors are 98% and 95% of frequency distributions are showing in the G-Nagar population, both in the male and the female. So I can say that these are features, including the hypoconionute, hypocone, class 2 malocclusion, overbite, shoveling in the incisors, and the crowding can be associated with the G-Nagar populations being their dental identifying features. Similarly, at the second end, these morphological features which are showing in this graph cannot be associated with the G-Nagar population being very low in the frequency distribution in the population. So you can say that 1.1%, 1.8%, these all are below 10%. And I can say below 5%. Most of them are below 5%. So they cannot be associated with the G-Nagar population as they are identifying a dental feature. So for the present study, we want to conclude that the G-Nagar population, being a genetically homogeneous population, can be distinguished from the other genetically homogeneous groups of the state Haryana on the basis of dental features such as the hypocone, hypoconolute, overbite, shoveling, crowding, and the class 2 malocclusion. Therefore, the present study can help in scientifically identifying the G-Nagar population of the Haryana state, which can further help in anthropological studies, forensic studies, and in human identification in various disasters where only and only the teeth are available. We can locate or we can associate the jaw we are having as an evidence. We can associate that individual, that jaw, to a particular population. If he or she from is having the hypocone, is having the hypoconolute, we can associate that particular jaw or individual to the G-Nagar population, irrespective of the disaster. And so these are the some of the selected references. And thank you.
The
I now
application of
invite
this study. It's the application of this study to the population. Clinical
Sir,
application.
the application of this study. Yes, sir. So the application of this study is first and the foremost application is in forensic studies. Sir, we find various types of evidences of the dead bodies where the facial features, the primary purpose of identification, the primary evidence of identification is facial features which get destroyed due to decomposition, due to putrefaction, or sometimes the perpetrator deliberately destroy or mutilate the facial features or the bodily features, or the body burns in explosions or other cases where we cannot identify the person or the dead body or the deceased from these features. In these situations, we find only teeth to recognize the person. So based on the present study, the results of the present study, if we found these morphological feature in that particular teeth, we can locate or we can identify the person that he or she is belonging to the G-Nagar population of the Haryana state. That can provide a lead in the investigation and identification of the deceased, sir.
Why you are selected only this population? Because I see a very limited application of this. It's not to a general population, and that you have started with a narrow population, and then how do you want to translate or scale them to a larger population?
Sir, in anthropology or forensic studies, we do work or research work on an endogamous population, because in endogamous the G-Nagar population which I have selected is an endogamous population. The benefit of endogamous population selection is that the characteristics, the genetic characteristics are limited in the population. They do not bifurcate, being the endogamous population marriage within the population. They do not marriage outside the population. So the characters, the traits of the population, the genetic traits being the secured. So sir, the results being the more reliable. So that's why, sir, I have chosen the endogamous population, that is G-Nagar. And moreover, sir, Based on the present study, the Jinegar population will get the legal identification which is not being given to the population in the state of the Haryana. So this is the application, sir.
Okay.
Thank
Okay, what will be the limitation of your studies? Any one limitation?
you. Yes, sir. Actually, when we visit to the population houses, I have done the door-to-door survey for the collection of the data. Sir, I think convincing the participants to participate in the study is I think is a big challenge, and that is a limitation of the study also, sir. I think
Limitation with
because
respect to
environment,
results. Sorry, limitation with respect to the results or findings.
sir, with respect to the results is that most of the results are being is our sample size due to the limited time is restricted. The limited sample size is a major, I think, limitation in itself, sir, because the generalization of the results to a larger population of the Jinegar in the Haryana state, because we have conducted the research only in the district Karnal of the Jinegar population, but the generalization of the results to the whole of the Jinegar population of the Haryana state will be a limitation for the study, sir.
And can it be used as a primary tool of identification, in your opinion, in addition to, like, there are multiple, if when it comes to forensic application. So do you recommend it as a primary tool for identification?
Yes, sir. I surely recommend it as a primary source of identification, being the 95% of accuracy in the results, and moreover, sir, the standardized or the establishing forensic odontological morphological feature as identification mark by the Interpol USA is also as sir, a supportive material, a standardized material that shows that these features can be used as a primary method, sir.
Okay, all right. Over to Dhandar, sir, please.
Yeah, I have one question. Like when you perform this, what you call studies, have you taken any other population as a reference?
Yes, sir. Yes, sir. Definitely. Sir, this is a part of a PhD study. In a whole PhD study, we are considering another population from the same environmental condition, from the same state, from the same locality, to compare that whether this population is unique from the other population based on the dental features.
No, I
Sir,
mean
we are
the
taking.
same population from the same community means most of the features will be similar, right?
Sir, we have taken the another endogamous population for the comparison. This is the second endogamous population which I have shown in the present slide presentation.
Okay,
Sir.
okay.
We have taken two
Apart from the
populations.
Zinger population.
Yes, yes,
Yes,
sir. Yes, sir.
yes. Mm. So, like, have you thought of trying some, molecular biology techniques also, like to correlate with your findings?
Yes, sir. Surely, sir. In the second phase of the analysis, we will definitely go for the molecular studies, such as we conduct in DNA fingerprinting in forensic studies. We will definitely go for that, sir, to confirm the results, sir, to enhance the accuracy and reliability of the results, sir.
Because I feel that only the dental features, studying only the dental features will not be significant enough to properly identify. So I think you have to go for some more studies, right?
Sure,
Okay,
sir.
thank
Sure,
you.
sir.
Thank you.
Sir, more. Thank you, sir.
Yeah,
Thank you,
please,
sir.
please.
Sir, moreover, in forensic identification, sir, we have superimposition technique where if some bite in sexual assault cases, more specifically, we have biter's marks on a victim's body. So, sir, based on the dental cast and the dental features, we prepare a bite mark on the software, sir, and then the bite mark of the biter on the victim's body. Then we superimpose both of these
Yeah, yeah.
based on these morphological features,
Yeah,
and then, sir,
that
then
comes
we
to
compare, sir.
biometrics analysis. Yeah, biometrics.
Yes, sir. Yes, sir. Yes, sir.
Okay, thank you.
Thank you, sir. Thank you, sir.
Thank you, Douglas, for your valuable questions.
Now I would like to call upon code number PhD03
Kindly switch on your camera to confirm your presence.
We will now be presenting your video.
Hello everybody. My work is on profiling of circular RNA mRNA interactome identifies dysregulated endometrial pathways in recurrent miscarriage and recurrent implantation failure. Either embryo or endometrium can lead to pregnancy complications like recurrent implantation failure and recurrent miscarriage. There can be several reasons which can lead to recurrent implantation failure and recurrent miscarriages. In case of recurrent miscarriage, despite the quality of embryo, endometrium allows the embryo to implant due to the over-receptive or under-selective nature of the endometrium, which leads to...
It's not audible. There is some problem.
In case of recurrent implantation failure,
It's audible to me.
transferring good quality embryos in several IVF cycles due to the over-selective or under-receptive
I think the
nature of
volume can be
endometrium,
slightly increased.
there is a
Yes, sir. Yes, sir. We're looking
failure
at.
of implantation that causes recurrent implantation failure. So in our study, we are focused on understanding how the endometrial factors are distinct with embryos in several IVF cycles due to the over-selective or under-receptive nature of endometrium. Hello everybody. My work is on profiling of circular RNA mRNA interactome identifies dysregulated endometrial.
Sir, is the audible? Is it audible now?
Audible to me.
In recurrent miscarriage and recurrent implantation failure. So, the interaction between embryo and endometrium is critical to achieve successful pregnancy. Disruption in either embryo or endometrium can lead to pregnancy complications like recurrent implantation failure and recurrent miscarriage. There can be several reasons which can lead to recurrent implantation failure and recurrent miscarriages. In case of recurrent miscarriage, despite the quality of embryo, endometrium allows the embryo to implant due to the over-receptive or under-selective nature of the endometrium, which leads to consecutive miscarriages. In case of recurrent implantation failure, despite transferring good quality embryos in several IVF cycles, due to the over-selective or under-receptive nature of endometrium, there is a consecutive failure of implantation that causes recurrent implantation failure. So in our study, we are focused on understanding how the endometrial factors are distinct between RM and RIF and how that leads to pathogenesis of RIF and RM. So the current studies about understanding the factors, endometrial factors that causes RM and RIF are focused on mRNA profiling, and also the test and the recent study to identify the receptive nature of endometrium and the endometrial receptivity array is also detecting 248 genes, that is mRNA profiles, for qualifying the endometrium as receptive or not. Other than mRNA, there are several other non-coding RNAs like circular RNA, miRNA, long non-coding RNA, which regulates the expression of mRNA at various levels. And also, several studies have shown that non-coding RNA's involvement in the pathogenesis or onset of various pathological conditions. So, in our study, we have focused on circular RNA, which is one of the non-coding RNAs which is originating from the same gene from which mRNA gets transcribed. Generally, circular RNAs are formed by backsplicing of the downstream exon to the upstream exon, forms a circular structure that are called as circular RNAs. Several studies have found that circular RNA regulates the expression of mRNA at several levels like transcription, ribosome binding protein sponging, and microRNA sponging, protein scaffolding, and translation. Thus, circular RNA regulate mRNA only directly like through RBP sponging or microRNA sponging? No. Recent studies have found that circular RNA can directly interact with mRNA and regulate their expression. The rationale of our study is to understand how the direct circRNA mRNA interactome is distinct between control, recurrent miscarriage, and recurrent implantation failure. By understanding this distinct nature of circRNA mRNA interactome between these conditions will help us understand the pathophysiology and disrupted mechanisms in the endometrium of these conditions. So, for our study, we have used publicly available rRNA depleted SRA datasets of which are RNA-seq data of endometrial biopsies which are collected during the receptive stage of menstrual cycle from the normal control women and recurrent miscarriage and recurrent implantation failure women. We have used RNA-seq, a Python-based platform which is developed in our lab for both coding and non-coding RNA analysis. Using that, we have done our circular RNA analysis and mRNA analysis. To identify the circRNA mRNA interactome, we have done command line blast using RNA-seq, and we have found unique circular RNA mRNA pairs in recurrent miscarriage and recurrent implantation failure. As moving to result, we have performed principal component analysis to identify the correlation between the samples. We have found that control and disease groups are showing distinct profiles, and further analysis of the disease condition shows that RM and RIF endometrial transcriptome are distinct, which suggests that RIF and RM, although they share similar clinical characteristics, they are still distinct entities. So, after circular RNA analysis, we have identified around 26,000 circular RNAs among control RM and RIF, and Circos plot shows that the distribution or the expression of these circular RNAs are from all over the human genome. And among these circular RNAs, exonic circular RNAs are expressed more in all the groups. And we have identified the unique circular RNAs of recurrent miscarriage and recurrent implantation failure, and which are around 4,000, which is huge number to work with. So we have used a filtering criteria to select circular RNAs. They are splicing length of the circular RNA is between 200 and 1200 base pairs, and whose exon size is between 1 to 4 exons. After applying these filtering criteria, we have identified 192 and 182 circular RNAs, unique circular RNAs, are in recurrent miscarriage and recurrent implantation failure condition. So, to identify the interacting partners of those unique circRNAs, we have done command line blast, and we have identified circRNA mRNA interactome, and further we have analyzed the unique circular RNA interacting mRNAs. And among the total mRNAs, only 7 to 8% of mRNAs are interacting with the circular RNAs, and these circular RNAs are interacting with the 5' UTR end of the mRNA, which suggests that these circular RNAs are involved in regulating the translation of these mRNAs. So, gene ontology analysis of circRNA mRNA interactome of recurrent miscarriage shows that cortisol synthesis secretion pathway and cell-cell junction pathways are altered. So, recent studies on chronic stress demonstrated that cell-cell junction morphology or the cellular permeability got disrupted due to the dysregulated expression of tight junction proteins. So, this suggests that dysregulation of cortisol pathway may disrupt the cell-cell junction morphology, which affects the cellular permeability, which can contribute to recurrent miscarriage. Similarly, in case of recurrent implantation failure, gene ontology analysis have shown that response to type 1 interferon and its upstream pathways are altered. Ongoing study in our lab shows that altered immune response, specifically interferon response, leads to impaired implantation. So this suggests that dysregulation of interferon response here may impair the improper implantation, which contributes to recurrent implantation failure. To conclude, endometrium in women with recurrent miscarriage and recurrent implantation failure have shown distinct gene expression profiles, and circular RNA analysis have shown that they have unique circular RNAs in case of RM and RAF, which shows that they are distinct entities. And circular RNA, mRNA interactome have revealed distinct endometrial signatures of RM and RIF. In case of RM, dysregulated cortisol pathway can lead to recurrent miscarriage, and dysregulated interferon response pathway can be the one of the causes of recurrent implantation failure. So we are planning to further study about understanding how the disruptions in cortisol pathways and interferon response pathway contribute to recurrent miscarriage and recurrent implantation failure. Identifying their roles will aid in identifying transcriptomic biomarkers to predict RM and RIF, and also finding novel therapeutic targets for these conditions. And these are my references. Thank you.
Have you validated any circular RNA?
No, sir. That is going to be the next plan of our work.
And how do you want to validate? You have mentioned like these are potential to be a therapeutic target, and then again, second is a biomarker. So unless if there's no process to validate and explore, like how do you want to validate basically any circular RNA from a therapeutic perspective?
Sir, can you please repeat the question?
I'm saying, how do you want to validate this circular RNA from a therapeutic perspective?
Sir, actually, we are going to do check the presence of these circular RNAs that are present in the samples, and is there interaction between to find the interaction between circular RNA and mRNA is actually happening.
Can you take any example? Like, how do you want to prove this? I'm asking because see, there are microRNAs, circular RNAs, then there are also non-coding RNAs, LNC RNAs are there. So it's not the only circular RNAs who may regulate any particular gene. There may be several factors. So out of those many RNAs, how do you select a specific RNA and which will have more significant impact?
Sir,
second is how do you target?
So to look at the circular RNA, which are like, as I said, it is a circular RNA that they are forming a closed loop structure. We are using an RNase R, which doesn't cleave circular RNA. So we are just identifying those circular RNAs are involved in this process.
Oh, that is your identification. I'm saying from a disease perspective or from a clinical perspective, you should choose which RNA which is more relevant, right?
Yes, sir. Yeah.
So what will define that relevance, I'm asking?
Sir, as studies have shown that circular RNA regulate the functionality of mRNAs. I mean, especially the translation of those mRNAs, which get translated into protein and affect the various pathways. So by looking at how these circular RNAs are expressed and which has the capacity to regulate mRNA.
Yeah, between LNC RNAs and circular RNA, which one you use?
Sorry, sir?
Between LNC RNA,
yes, sir,
and circular RNA, which one you'll use from a therapeutic perspective? Which one is easy to work?
Sir, circular RNAs.
Not LNC RNAs.
No, sir. And actually, the circular RNAs are more stable than long non-coding RNAs because they don't have they are not linear like long non-coding RNAs, which gives an added advantage to using circular RNAs. Hello.
I'm done. I'm done.
Yes, okay. Thank you.
Yeah. One question.
Yes, sir.
Yeah. What is the percentage of positive interaction between your mRNA and circular RNA?
Sorry, sir.
What is the what positive interaction percentage of positive interaction you get between the messenger RNA and the circular RNA? When
Sorry, I
forming a
couldn't.
complex,
Positive interaction. What do you mean by positive interaction, sir?
means
Sorry.
they form a complex, right?
Yes, sir.
Which
Yeah.
which we are using. So, like you are performing in silico studies only, or you are doing wet lab also.
Sir, we haven't started yet, but we are planning to do in vitro analysis,
Okay.
looking at how they are interacting and yeah.
So in silico, it is interacting properly.
Yes,
Right.
sir. Yeah. Yeah. We are
So
performing the
in
labs to identify the
uh,
interaction between circuit.
okay, but you have not figured out or in wet lab how much percentage you get. I mean, in interaction wise, right?
Yes, sir. The next part of our plan is to look at the in wet lab only.
Okay. All right. Thank you.
Thank you.
Okay, in the future direction you have mentioned that is application of this interaction that we have studied, identification of therapeutic agents and biomarkers. So is it going to be a part of your study, or you are going to limit the study only till the mechanistic interaction?
Sir, we are planning to identify how these circular RNAs can be used as biomarkers to predict this RM and RAF in earlier, because they are differentially expressed in RM and RAF. So just how can we use this as biomarkers to identify these conditions in earliest? So we're planning to do that.
Okay, all right.
Thank you.
Done from my side also.
Thank you so much, judges, for your valuable questions.
I would now like to call upon PhD 04 for the presentation. Please turn on your camera. PH04, please turn on your camera to confirm your presence.
I've already turned on my camera.
Yes, we will now present your video.
ये किसका ऑन है? Hello everyone, I'm here to present my PhD. Hello everyone, I'm here to present my PhD work titled as IRGM1 is required to regulate inflammation during embryo implantation. Let's start by understanding what is autoimmunity. It's a condition wherein your immune system attacks its own cells and leads to the hyperinflammatory state in the body. Now, women with autoimmunity have a high infertility rate as compared to normal healthy women. Immunity-related GTPase-M, that is IRGM, plays an important role in innate immunity and inflammatory responses by modulating interferon pathway, inflammasomes, and autophagy pathways. This IRGM1 molecule is a genetic risk factor for several autoimmune diseases, and polymorphisms have been seen in this gene when in women with autoimmunity, and they've been found that they are also infertile. So all of this put together tells us that IRGM1 defect leads to autoimmunity. This autoimmunity increases the infertility in women. But what is the role of IRGM1 in infertility is yet to be explored. Which leads us to the question that does loss of IRGM1 lead to infertility? And to answer this, we have a name wherein we want to find out the role of IRGM1 in embryo implantation and early pregnancy. To achieve the same, we have used IRGM1 knockout mice model, and we have first looked at their fertility profile, for which we have observed the litter size and the days taken to conceive. We could observe that IRGM1 knockout had produces half the litter size as wild type, and they take twice as longer to conceive as compared to wild type, telling us that they are not completely infertile but rather subfertile. Now, what leads to this subfertility? Is it because of an ovulation defect? To check that, we checked their estrous cyclicity for 2 consecutive cycles, and we could observe that both wild type and IRGM had similar expression, similar estrous profile, telling us that it is not because of an ovulation defect. Next, we question that then is it because of an implantation defect? Because implantation is an indispensable process when it comes to successful pregnancy. So we looked at the histological analysis. We did the histological analysis of day 5 wild type and IRGM knockout uterine tissues, wherein we looked at the implantation site. We could see that the proper implantation cup was formed in wild type, along with the proper orientation of embryo and decidual zone. However, in IRGM knockout, we could see the deformed implantation cup formation along with epithelium slugging off at one side, and the zone of decidua was also reduced, telling us that there is indeed implantation defect. But what is leading to this implantation defect? For that, we went ahead with RNA-seq of day 5 IRGM knockout uteri along with wild type, and upon analysis, we observed that there were totally 277 genes differentially expressed, of which 195 were upregulated and 82 were downregulated. Now, we went ahead with the pathway analysis of these differentially expressed genes, and in the biological process, we could see that amongst the top hits, we had response to interferon alpha, interferon beta, chemokine production, and regulation of innate immune response, telling us that there is some alteration in interferon response. Coming to molecular function, we could observe T cell receptor, natural killer cells, and cytokine activity in the top hits, telling us that there is again immune alteration happening. Similar thing was observed in KEG pathway. Now, next we wanted to know whether this is upregulated or downregulated. What is happening to the gene? So, because the interferon is one of the most constant hits that we were getting, we looked at the interferon stimulated genes. MX, and what is the profile? We could see that MX1, OS15, OS1, ISG15, most of the immunity related genes were highly upregulated in knockouts, telling us that indeed there is some alteration and some defect happening because of that. So we went ahead with the validation of few of the genes, MX1 and ISG15 by RT-PCR. And we could see the several fold higher expression of MX1 and ISG15 in knockouts. To conclude that, which tells us that at the time of implantation, there is upregulation of interferon and inflammatory responses in IRGM knockout. Next, what is happening when this interferon response and inflammation is hyperactivated? So when we talk about implantation, receptivity is one of the main event and happening, which is required. Receptivity is basically a window of period wherein your maternal endometrium is conducive enough to accept the semilogenic blastocyst. Previous studies from our lab have shown that increase in inflammation leads to disruption of this receptivity. So we wanted to check the receptivity profile by looking at the hallmark markers. Estrogen is one of them. So we looked at the ER alpha, ER beta expression at implantation on day 5. We could see that in wild type, there was baseline expression in glands and stroma of both. However, in IRGM knockout, there was significantly increased expression in stroma as well as in the epithelium, telling us that there is something happening. Now, this estrogen leads to increased proliferation of cells in the epithelium, and under the influence of progesterone, now they need to switch their fate to differentiation. So next we wanted to check what is the effect on the proliferation, for which we have used Ki67 as a proliferative marker, and we could see that in wild type there was baseline expression in the stroma. However, in IgM knockout there was very strong expression in the epithelium and stroma both, telling us that these cells at the epithelium fail to exit the cell cycle and thus fail to enter the differentiation, leading to implantation defect. Next comes the hoxatin and beta-catenin, which are again a few of the main markers. We observed a similar expression profile as estrogen and Ki67. So we could see that increased expression of hoxatin and beta-catenin was there in IRGM knockout. Now, to summarize, IRGM knockout are subfertile, and which is because of interferon response and inflammation that leads to compromised receptivity and subfertility. So what if we block this interferon response? That's what we wanted to ask next. So can this subfertility be rescued by blocking interferon response? So to answer that, we have used double knockout of IRGM1 and interferon alpha, and we looked at their fertility profile. And very surprisingly and interestingly, we saw that the litter size comes back to normal in DKOs, that is double knockout, and the time they took to conceive was also reduced and was comparable to wild type, telling us that indeed if we block this interferon response, we get the rescued phenotype in IRGM knockout mice models. Also, to relate it back to the autoimmunity, this tells us that high infertility rate in women with autoimmunity could be because of defective IRGM1. Now, to conclude my study, that is, what is the role of IRGM1 in embryo implantation and initiation of pregnancy? We can say that semi-anogenic blastocyst is not accepted by the maternal system if there is interferon response and inflammation in the absence of IRGM1. However, if IRGM1 is there, so this interferon response and inflammation is regulated, and this leads to the successful implantation of blastocyst into the maternal endometrium, telling us that indeed IRGM1 is responsible and is required for implantation and initiation of pregnancy. Thank you so much. These are my references.
What is the clinical value of this?
Hello, sir. Sir, if we get to know that if we control inflammation and that can lead to increased fertility profile. So in future, we can look into the problems, for example, recurrent implantation failure or recurrent pregnancy losses wherein inflammation is very high. So we can say that it can be applied, this knowledge can be applied there and looked at if we can do something with IRGM and regulate that inflammation, we'll be able to get higher fertility rate in ART, people who go for assisted reproductive technology.
And how specific is inflammation? Inflammation may be a very non-specific in nature if you are looking only for implantation.
That is true, sir. So here, yeah, sorry.
Go ahead.
So, sir, when we talk about embryo implantation, then the inflammation that we are talking about with respect to embryo implantation is very specific because embryo implantation is a process wherein the inflammation is needed in a controlled manner. So, for pregnancy to sustain, after this implantation has happened, the inflammation needs to go down. If it doesn't go down, then that leads to pregnancy losses. So if you're talking with respect to embryo implantation, inflammation is specific in terms of its regulation.
How do you control it? You said it needs in a control room.
Are you talking physiologically how we can control it?
No, with this, like suppose you are making a knockout, you are saying. How do you make a knockout?
Yeah.
Single knockout and double knockout.
So, sir, basically these are CRISPR-Cas9 originated. So we have got this from the Jackson Laboratories with the help of our institute, and we have used gene editing technology in this. And for getting the single knockout, we can basically we have used this vector backbone wherein we have crossed it with the homozygotes and we have gotten the knockouts for IRGM1. And for double knockout, again the same approach was used.
Did you confirm knockout specific with CRISPR that it is only specifically it is knocking it out?
Sir, in terms of, so we have done genotyping. We have done the genotyping for it. So, and we have got that, so we have seen in the age and electrophoresis by using electrophoresis that the particular animals that we have used are knocked out for IRGM1 and even Western was used. So, yeah.
Okay. Thank you.
Thank you, sir.
I have one question.
Yes.
What is the mode of function of your this one IRGM? Like whether it is directly interacting with the what you call interferon, or how does it reduce the inflammation?
So basically, IRGM is a molecule which regulates via controlling multiple pathways. So if I talk about its mode of action, it has multiple modes of action. One is wherein it stops the inflammasome complex formation. So inflammasomes are the complex formed by NLRP3 ASC, which leads to, when they oligomerize, it leads to activation of caspase, which in turn leads to release of IL-1 beta maturation, which is an inflammatory molecule. So IRGM, what it does is it goes and binds to NLRP3 and blocks this complex formation, and thus regulating inflammation. Another aspect is wherein it activates autophagy process. Autophagy is basically degrading the inflammatory molecules. And third pathway is wherein it activates CGAS-STING signaling by SQMT, and which leads to proteasomal degradation. And then all of these pathways help in regulation of inflammation by IRGM. So in this, so if IRGM is less, for example, it is not active, it will lead to inflammation and hyperinterferon response, because then interferon stimulated genes are activated and further signaling is happening.
Okay. So, like, if you think of what you call developing a product with this work for use in the society, then how do you see it in for future purpose?
Okay. So, sir, as we know that IRGM regulates inflammation. So, if we specifically talk about embryo implantation or autoimmune women who are suffering from autoimmunity, we need to regulate the inflammation per se. So, as we can see that in the absence of IRGM1, inflammation is hyperactivated, interferon response is hyperactivated. So, to control that, we can work on certain small molecules or antibody therapeutics for that matter, which can control this inflammation. So if we talk about specificity, or like, for example, immune cells, which are more responsible for towards this adaptive immunity, we can synthesize some therapeutics which will control the cells and thus the signaling of inflammation.
Okay, basically some sort of a molecular injections sort of thing. Yeah, thank you.
Yes, sir. Yes, sir. Yes. Yes, sir.
No questions from my side. Please go ahead.
Thank you so much.
Thank you, Jaljas, for your valuable questions.
I would now call upon code number PhD05. Please turn on your camera to confirm your presence.
Am I visible?
Not yet. Yes, you are visible now. Yes. We'll be presenting your video now.
Hello everyone. My oral presentation identifier is PhD05. I am presenting my work on topic in vitro in silico investigation reveals potential cytotoxic activity of fermentation metabolites from actinomycet isolated from lunar soda lake against HeLa cancer cell lines. Background and rationale. According to the recent data published in 2024 on topic global cancer incidences and death statistic report published by International Agency for Research on Cancer of World Health Organization and American Cancer Society, there is an increase in the prevalence of the cancer, and therefore there is a need of alternative anti-cancer agent. Therefore, the researchers are investigating the molecules from the plant's microorganism, and the molecules were chemically synthesized for probable anti-cancer activity. As far as the microorganism is concerned, microorganism from the marine source are rich source of the secondary metabolite that may have a probable anti-cancer activity. The similar kind of environment is possessed by the Lonar Lake situated in Buldana district of Maharashtra. It is created by the meteor impact 52,000 years ago, and it is having the high salinity, high pH, and it possesses the unusual sort of ecology, and therefore it contains diverse array of microorganisms including rare actinomycetes. Thus, we are tried here to screen certain actinomycetes and prepare the fermentation bioactives, and these bioactives were screened for probable anti-cancer activity against HeLa cancer cell line. Method isolated by using a selective medium, and further it is purified, then it is subjected for the fermentation by using shake flask culture by using starch casein broth as a medium at 150 rpm for 10 days at 37 degrees Celsius. Later on, whatever the fermentation metabolites were purified by using a downstream processing, and after purification of the compounds, they were characterized by using hyphenated analytical techniques such as LC-MS and NMR. The characterized compound were screened for the in silico screening, and we found that these compounds are having activity against HeLa cell, and therefore we have chosen HeLa cell to carry out the cytotoxic activity. We have also performed the molecular mechanism study by studying the effect of metabolites on oxidative stress markers such as GSH, MDA, then pro-inflammatory markers such as TNF-alpha and apoptotic cell marker capsis-3 activity. The detailed method is given in slide. Due to the lack of time, I'm just switching myself to the result. So as far as the result is concerned, according to the results of the present investigation, that is effect of fermentation isolate on cell viability, the HeLa cell viability against the standard drug cisplatin treated group at the concentration above 1.56 microgram per ml was significantly lower than that of the normal control group, with IC50 value noted at 4.58 micromole. On the other hand, the one-way ANOVA followed by the post-hoc test showed, when compared to the normal control treated group, the testing probe LLSD5 treated group at concentration about 10 microgram per ml was significantly lower the cell viability of HeLa cell with IC50 value at 34.17 micromole, whereas LLSD7 treated group at the concentration about 20 microgram per ml was significantly lower the cell viability of the HeLa cell with the IC50 value of 53.85 micromole. The testing probe LLSD9 treated group above 40 microgram per ml was significantly lower the cell viability of the HeLa cell with IC50 value of 69.54 micromole. Then effect of fermentation metabolites were studied on the oxidative stress markers such as GSH and MD activity. As per the result of the GSH activity, the HeLa cell normal control group was significantly lower, whereas the standard drug cisplatin treated group at the concentration of 1.56 microgram per ml was moderately significant, and above the concentration 3.12 microgram per ml was significantly higher than that of the HeLa cell normal control group. On the other hand, the one-way ANOVA followed by post-hoc test showed that when compared to the normal control group, testing probe LLSD5 treated group at concentration of 10 microgram per ml showed moderately significant, and at concentration about 20 microgram per ml shown significantly higher GSH activity. Testing probe LLSD7 treated group at 20 microgram per ml showed moderately significant activity, whereas above the concentration 40 microgram per ml showed significantly higher GSH activity. The testing probe LLSD9 at concentration 80 microgram per ml showed significant GSH activity, and concentration above 160 microgram per ml shows significantly higher GSH activity in the HeLa cell compared to the normal control group. Similar effect is shown in the MD activity. LLSD5 group at concentration of 20 microgram per ml showed moderately significant activity, and concentration more than 40 microgram per ml shows significantly high level of MDA. At concentration above 80 microgram per ml showed highly significant increase in the MDA level. The LLSD probe LLSD7 treated group at concentration of 40 microgram per ml showed a less significant activity, whereas above the concentration 80 microgram per ml showed highly significant activity by LLSD7. Whereas testing probe LLSD9 treated group at concentration 80 microgram per ml showed moderately significant activity, and the concentration above 160 microgram per ml showed highly significant level of MDA in the HeLa cell. The effect of fermentation isolate on the pro-inflammatory and apoptotic marker. According to the result of present investigation, the TNF alpha level in the HeLa cell normal control group were found to be significantly higher, whereas standard cisplatin treated group at the concentration of 1.56 microgram per ml shown moderately significant, and above the concentration 3.12 microgram per ml, highly significant in lowering the TNF alpha level as that of the normal control group. One way ANOVA followed by post hoc test showed when compared to the HeLa cell normal control group, testing probe LLSD5 treated group at concentration 20 microgram per ml was moderately significant, and above the concentration 40 microgram per ml was significantly lowering the TNF alpha level, whereas LLSD7 treated group at the concentration of 20 microgram per ml shows less significant activity. At concentration 40 microgram per ml shows moderately significant activity, and above the concentration 80 microgram per ml was shown significantly lower the TNF alpha levels. Testing probe LLSD9 treated group at the concentration of 40 microgram per ml shows less significant activity, whereas above 80 microgram per ml shows highly significant activity in lowering the TNF alpha level. Caspase-3 activity according to the result of the present investigation. The Caspase-3 activity was performed, and it was found that the standard treated group at a concentration of 1.56 microgram per ml shows significant activity, and above concentration of 3.12 microgram per ml, considerably restoring the Caspase-3 activity than that of the HeLa cell normal control group. On the other hand, LLSD5 treated group at the concentration of 10 microgram per ml shows less significant. At concentration of 20 microgram per ml shows moderate significant, and above 80 microgram per ml it was found to be highly significant Caspase-3 activity. The testing probe 7 treated group at the concentration 20 microgram, it has shown less significant activity, and significantly higher activity has been seen above 80 microgram per ml concentration by LLSD7. LLSD9 treated group at concentration of 40 microgram per ml shows significant activity, and above the 80 microgram per ml significantly in restoring the Caspase-3 activity of the HeLa cell. Then molecular docking investigation reveals substantial interaction among the metabolite and essential protein that regulate the cell proliferation and death, including BCL2 family members such as BCL2 and BCLW, MCL1, AKT, and BRAF protein. These interactions show that metabolite may induce apoptosis by altering this important pathway, which validate the in vitro outcomes. LLSD5 CID complex exhibited the most stability among the system as indicated by its lowest RMSD and RMSF value. Additional information on the compactness and solvent interaction of the complex was given by SASA and radius of gyration values. Although fluorouracil CID and LLSD5 CID exhibited more compact and consistent behavior, LLSD9 CID had a largest solvent exposure as far as the result of molecular dynamic is concerned. Data analysis, we use Prism 5 for Windows version 8.42 to analyze the data in our study. The mean plus standard error of mean was used to summarize the result, and we use one-way ANOVA and two-way ANOVA to establish the significance and highlight the changes across the viable studies within each group, followed by multiple comparison of parameter within the group. The statistical significance as p-value more than 0.05 is less than 0.05 is considered as a significant. Conclusion: the current research effectively isolated actinomycet from the Lonar soda lake observed that fermentation metabolites have substantial cytotoxic action against HeLa cancer cell line. The combined efforts of in vitro cytotoxic testing and in silico molecular docking and molecular dynamic simulation studies gives in-depth understanding of probable mechanism of action. The finding highlights the under-realized potential of severe settings as source of new bioactive chemicals with important therapeutic application. These findings add a growing body of data supporting the use of natural ingredient for development, particularly in cancer. Actinomycetes from lunar soda lake create unique metabolite that might be used to build the novel anti-cancer medicine. The research contribute to expanding our comprehension of natural product based therapy for cancer, and emphasize the need to investigate varied context of the drug development. These are the references for the studies conducted. Thank you very much.
What would be the mechanism for anti-cancer activity?
Am I audible, sir?
You are audible. Are you getting my question?
Yes, sir.
I'm asking what is the mechanism for this anti-cancer activity?
As we have performed the studies on the oxidative states marker, as well as we have performed on anti-inflammatory markers, pro-inflammatory markers, and apoptotic marker. So probable mechanism of action may be it cause apoptosis. It is having a capsus
mechanism I'm asking. That is the outcome. Apoptosis is happening. I'm saying from whatever the isolates you have. Yeah. What is the mechanism which causes this activity? Second question is how oxidative stress is so specific in nature. It could be oxidative stress is very non-specific in nature.
Yes, of course, sir. But this may be the probable way by which this one may happen. So we have taken the probable sort of markers like pro-inflammatory markers, apoptotic marker, and this one oxidative state marker like MDA and this one GSH.
But you have only done at one level. You have not validated at the downstream also. Just measuring the caspase activity will not tell the apoptosis is only because of caspase. There may be so many other mechanisms as well.
Yes, sir. Yes.
Similarly in oxidative stress also. So I'm asking what is so specific about this isolates that whatever the activity metabolites you have isolated. Have you characterized those metabolites?
Yes, sir. I have characterized these metabolites by CMSMS as well as by 1HNMR.
What is the stability of those isolates?
They are quite stable, sir.
Okay. Thank you.
Somewhere you mentioned that the isolates were purified by using some methods. So did you check the purity of isolates as well?
It is 98%, more than 98% pure.
What is the method of?
First of all, we did whatever the extract we have got that we have filtered through the membrane filter, 0.45, then followed by that 0.22 micron. And then it was crystallized by using organic solvent. And then after crystallization also, we have performed the column chromatography. And after column chromatography, the segregated fractions were characterized. And from the segregated fractions, we got to know that it is containing three different sort of a metabolite. That is neophytidine. The LLSD5 was containing the neophytidine. LLSD7 that was containing the taucodinol. Tetrapentagontan, and third one is containing taucodinol.
So what was the method of analysis? You said like 99%.
Actually, not exactly. The percentage purity means some traces of the impurities were noted when we have performed, because we got the tailing in the TLC plate. So it is indicating certain sort of impurities may be there, and the spot was not that much prominent. So it is the tailing indicated the impurity may be there. So on the basis of that, I'm saying that it may be near about 98% pure.
Okay. Any previous study on same actinomycetes on hair cancer?
No, actually, actinomycetes are the good source of a secondary metabolite. But no, this was the first time we have done the fermentation product, and we have produced the fermentation bioactives, and that fermentation bioactives we have screened. Whatever the studies which has been taken place on the Lonar Lake, that is up to the characterization of microorganism only. So majority of the research has been done by the microbiologist and not by the pharmaceutical people. So my approach was to take that research, not only restrict ourselves to the identification of the microorganism, but why not to check the application of those microorganisms for the potential industrial and pharmaceutical bioactives.
Okay. Thank you. Over to you, Dhanraj sir, please.
So, what was the substrate used for fermentation?
Actually, we have used amino acids. 3-4 amino acids we have tried, and we have used the media, that is starch casein broth. So with the hypothesis that the microorganism may convert this amino acid into the bioactives, and these bioactives may contain the parent structure of this amino acid. So that was the hypothesis behind that, and we found that several bioactives has been produced, and out of which we could able to purify very few, and not many potential is there. Yes, sir.
Okay. And yeah, and as you see, like apoptosis versus necrosis, what is the range between the apoptotic cells and necrotic cells?
Frankly speaking, sir, actually we need to do the work at the molecular level for these bioactives, whatever the bioactives we have got, so as to know the exact mechanism of action. So we have merely done the in silico and in vitro activity. That doesn't give the clear picture of the mechanism of action. We need to go for the further investigation, and that will be the part of my next research, sir.
And then why have you specifically chosen HeLa cells? Because
it has shown a very good activity against the BCL2.
I mean, HeLa cell is basically a cervical cancer cell line, right?
Yeah. Yeah.
So you have other cervical cancer cell lines also apart from HeLa cells, like SPV16 mediated. HeLa cell is SPV18 mediated, right? So when you try on those cells also.
Yeah. We need to screen it on the different kind of HeLa cell. That is a future prospect I have given.
Because if at all you are targeting the HeLa cell, it means that you are targeting the cervical cancer, right?
Yes, sir. Yes.
So you should look for some other cell lines also, because while working with HeLa cell lines, you can also incorporate some other cervical cancer cell lines also. So they will give a clear picture.
Yes, sir. Definitely.
Thank you.
We will include your suggestion. Thank you, sir. Thank you so much. Good day.
Thank you so much, judges, for your valuable questions.
Now I would like to call upon PH06 to turn on their camera, please.
Hello, I'm there in the meeting.
We will now be presenting your video.
A very good afternoon to all present over here. Today I'm here to present my research self-assembled hyaluronic acid nanoparticles. A very good afternoon to all present over here. Today I'm here to present my research entitled Self-assembled hyaluronic acid nanoparticle for co-delivery of beta-glutamic acid and paclitaxel for improved cancer therapy. As we all know, the prevalence of cancer is increasing day by day, particularly in breast cancer. Every 1 in 8 women tend to develop breast cancer, and this number is going to get increased in the upcoming years, which highlights the need of more novel therapies which can effectively manage this disease. The conventional treatment mainly includes administration of the chemotherapeutics like paclitaxel, which mainly act as a mitotic inhibitor and induces the apoptosis. Although the efficacy of this drug is well proven in the area of cancer, still there are some challenges associated with this immunotherapy. First is its rapid metabolism, because of which a limited concentration reaches to the tumor site, and thus the efficacy gets compromised. Second is the increased resistance, mainly because of the tumor cellular reprogramming that occurs. And the third one is dose-dependent toxicity. So to overcome these challenges, we have come up with the combination of paclitaxel with the glycolytic inhibitor, that is betulinic acid. The combination together gives us the advantages of synergistic action by targeting different cellular pathways. Then the dose will be reduced, and thus the dose-dependent side effects can be overcome. Betulinic acid is a known glycolytic inhibitor which can effectively manage the metabolic reprogramming, can overcome the resistance, and ultimately increasing the efficacy. So to deliver this drug combination, we have proposed a self-assembled hyaluronic acid betulinic acid based nanoparticle. This nanoparticle itself has a biological action, and because of which the synergistic activity will be there. Since we are including a hyaluronic acid, which is a known targeting ligand, which effectively targets CD44 receptor overexpressed on the tumor cell. Also, the hyaluronic acid is important for dual action. First, it acts as a vehicle, and second is the targeting. Further, this is also responsible for the enzyme-mediated release in the tumor cell. The hyaluronidase enzyme, which is specifically present in the tumor cell, can cleave the hyaluronic acid-based nanoparticle, and it will give a site-specific release as well. So, based first, the synthesis and characterization of the amphiphilic polymer was done, wherein betulinic acid was converted into its aminate form using ethylene diamine as a linker. The aminated betulinic acid was reacted with hyaluronic acid using carbonamide chemistry, leading to formation of an amphiphilic polymer. The synthesis polymer was characterized using FTIR, wherein the amide 1 and 2 band peak was present, suggesting the amide bond formation. Further, the proton NMR showed peak between the 6.8 to 8 ppm, which is corresponding to CNH group of amide bond, and overall suggesting that the successful synthesis of conjugate was done. Synthesized polymer was evaluated for degree of substitution, wherein it can be seen that as the molar ratio of HA to BTA increased, the degree of substitution was also increased. However, at 1:2 ratio, there is an insignificant increase in the degree of substitution was observed, and therefore the 1:1.5 ratio was considered optimized. Further, the critical aggregation concentration was determined, which was found to be 44 microgram per ml, which is low as compared to the surfactants, and which suggests that the nanocarrier or the nanostructure will be formed. It will be able to maintain the structural integrity upon dilution. Then the in vitro hydrolysis study was performed at pH 7.4 and in the tumor lysis. The high hydrolysis was observed in case of tumor lysis, suggesting a site-specific hydrolysis and release of betulinic acid. Further, the synergism between the paclitaxel and betulinic acid was determined, wherein it can be seen that the combination index value of 1:1 ratio was found to be less than 1. Further, the dose reduction index value was determined, which at all the ratios was found to be more than 1, which suggests a favorable dose reduction, and also the dose-related side effects can also be overcome in this case. Next step is the preparation of nanoparticle, for which self-assembly method was used. The amphiphilic polymer was dissolved in water, and ethanolic solution of paclitaxel was added into it at constant injection rate. Further, prosonication was done, and dialysis was done in order to remove the organic solvent. Morphological evaluation showed a spherical size of nanoparticle. Further, the particle size determination showed nanoparticle had size less than 200 nanometer, which ensures that it can also accumulate inside the cell via passive targeting through an APR effect. Zeta potential of -18 could be attributed to the carboxyl group of hyaluronic acid, and it suggests a high colloidal stability of the nanoparticles. Further, the developed nanoparticle had high entrapment efficiency as well as the drug loading, which is highly desirable. Dilution study was done in order to prove the stability upon dilution. Herein, it can be seen that up to 20-fold, only 11% of the drug loss was observed, which signifies that the nanoparticles were effective enough in order to maintain their structural integrity on dilution. Further, the in vitro release study was performed at pH 7.4, which mimics the physiological condition, and pH 5.5, which mimics the tumor cell environment and in presence of the hyaluronidase enzyme. As expected, the release was accelerated at pH 5.5 in presence of hyaluronidase, which could be attributed to the hydrolytic action of hyaluronidase enzyme on hyaluronic acid-based nanocarrier, and which is desirable so that the release will specifically occur in the tumor cell and not in the physiological condition. In vitro biological characterization was performed in triple-negative breast cancer cell line. First is a qualitative cellular uptake studies using CLSM imaging, wherein coumarin 6 was used in order to mimic the hydrophobic nature of paclitaxel. Here it can be seen that upon the treatment with dye-loaded nanoparticles, there is a green intense fluorescence was observed, suggesting higher cellular internalization. Further, in order to substantiate the exact mechanism of this internalization, the cells were pre-treated with hyaluronic acid, and again the internalization was evaluated. Here it can be seen that upon the pretreatment with hyaluronic acid, there is a substantial reduction in the intensity was observed, suggesting the predominant mechanism of internalization could be CD44 mediated endocytosis. So to substantiate this, quantitative cellular uptake studies were performed, and here it can be seen that upon the pretreatment, there is a significant reduction in the uptake was seen. again to suggest that the CD44 mediated endocytosis is the predominant mechanism for internalization of hyaluronic acid based nanoparticle. Further, the determination of ATP level was carried out since we are delivering a glycolytic inhibitor. So the effect of nanoparticle based delivery system on this ATP level was also determined. Here it can be seen that the treatment with nanoparticle resulted into 18 to 20% reduction in the ATP level, which would be attributed to the synergistic action as well as the increased accumulation of the tumors within the tumor cells. The next is cytotoxic potential determination and apoptosis assay. So in case of cytotoxic potential, all the test groups showed dose-dependent decrease in the person's survivability. However, the highest reduction in person survivability was observed in the nanoparticle treated group. Further, the reduction in IC50 value was evaluated, wherein nanoparticles showed nearly 1 to 2-fold reduction in IC50 value as compared to synergistic drug combination. Apoptosis assay was performed using Annex UV, wherein it can be seen that the nanoparticle treated group showed a strong yellow fluorescence. That means that the majority of cells are in early apoptosis stage. To substantiate these results, further the quantitatively apoptosis index value was determined, wherein it can be seen that a high apoptosis index was observed in nanoparticle treated group. So the possible reason is more amount of drug is available in the tumor cell, and because of which there is increased efficacy of the synergistic combination can be. So next is estimation of mitochondrial membrane potential. So both of these drugs are known to cause mitochondrial membrane depolarization, ultimately leading to cytochrome C release and apoptosis induction. So the effect of nanocarrier system on MMP was determined, wherein it can be seen that the nanoparticle treated group showed highest green fluorescence, which means that the mitochondrial depolarization is highest in nanoparticle treated cells. Further, to support this, quantitative determination of R by G fluorescence ratio was determined, wherein it can be seen that the lowest value of fluorescence ratio was obtained in nanoparticle treated group, suggesting the high mitochondrial membrane depolarization. Further, the lactate level were determined, wherein it can be seen that the nanoparticle treated group showed nearly 30 to 35% reduction in the lactate level, suggesting the glycolytic inhibition potential of the developed nanoparticles. Next is a in vivo animal study wherein the pharmacokinetic profile of PTX was determined. It can be observed when the PTX is delivered through a nanocarrier system, there is a significant improvement in pharmacokinetic parameter was observed, which could be attributed to the hydrophilic nature of the hyaluronic acid as well as the nanometric size which avoid the RES uptake. Later, the anti-cancer efficacy study was performed in 41 based TNBC model, wherein the nanoparticle treated group showed significant reduction in the tumor volume, percent tumor weight, tumor size, and percent tumor burden. However, moreover, the percent tumor inhibition rate was highest in the nanoparticle treated group, which was nearly 85%, and the probability of survival was also highest in nanoparticle treated group, suggesting that the nanoparticle was effective enough in order to show the anti-cancer efficacy. Once the efficacy is established, later on the safety profile of nanoparticle was determined. So herein the nanoparticles show no signs of hemolytic toxicity. Further, there is insignificant changes in the biomarker level were observed, and histopathological evaluation showed no signs of organ toxicity, overall suggesting the superior safety profile of nanoparticle as relative to the free drugs. So to conclude, the combination of paclitaxel and glycolytic inhibitors such as betulinic acid is found to be effective to exhibit a synergistic anti-cancer action. Further, the synthesized hyaluronic acid betulinic acid based nanoplatform offers a powerful avenue by not just dual targeting, but it also a biocompatible carrier, and hence it can be explored to deliver other combinations of anti-cancer drug as well.
I now invite the judges to please ask their questions to the presenter.
Why did you study mitochondrial potential in this?
The mitochondrial membrane potential is done by Jessamine assay. So it is basically mitochondria is a powerhouse, and any changes in the cell status, as in healthy cells, the mitochondrial membrane is polarized, nearly 150 to 220 millivolt. And when the cells are going under apoptosis, the depolarization of that membrane occurs. So basic mechanism of this mitochondrial-based apoptosis is mitochondrial membrane depolarization will occur. Then cytochrome C will release, which ultimately leads to activation of caspase and the apoptosis induction. So by checking the depolarization, whether the combination or the formulation is actually working and activating apoptosis or not. So this is one of the indicator to determine the apoptosis.
No, I asked why mitochondria.
Acha. So the combination of the drugs, that is the betulinic acid, one of the action of its apoptosis is mitochondrial membrane depolarization as well. So to evaluate whether the two drugs in combination are able to uh, more depolarization is occurring or not by the combination therapy. That's why we have evaluated the mitochondrial membrane depolarization. Also, it also indicates the efficacy of our formulation as well. Since the formulation uptake is more, the more concentration of the drug will be available in the cell, and because of which it will also prove the efficacy of the formulation as well as the synergistic drug combination. For that reason, we have evaluated the mitochondrial membrane potential.
But will it not affect the mitochondrial function? For example, have you heard mitophagy?
Yes, sir.
What is mitophagy?
So the cellular function within the mitochondria will get affected in this.
No, what is mitophagy? I'm asking.
So the disruption of mitochondria, I think. I'm not sure exact definition of it. But since we are carrying out the study in the apoptotic cells, the main goal is the mitochondrial membrane depolarization to occur.
That's what I'm asking. Since you are looking into mitochondrial depolarization and all these things, so mitophagy is a selective process which plays a role in removal of the mitochondria which are not in healthy in nature.
Okay.
Right?
Okay.
So, what is if mitochondria is damaged? What will happen to the cell?
So cell death will happen, obviously.
Only cell death?
Majorly, the impact will be the cell death.
Cell death is because of?
So if the powerhouse has been disrupted, obviously the energy will not be there. So how the cell will survive, and that is the ultimate goal in case of the cancer cell, that we need to deprive it from ATP utilization ultimately, so the cell shrinkage and the apoptosis will happen. So that is the
Any further questions?
Yeah. I have one question. Like in your in vivo studies, like what is the mode of this one? Mode of
induction.
Drug induction. Yeah.
So for in vivo study, first I will just give a protocol. We have induced a 41 base TNBC model wherein the cells are injected to a subcutaneous way, and it is a nearly 1 month procedure, and right from the induction till the treatment period. And then I think you are asking about the administration route. So it is through the tailwind, since it is IV administration route. So we have given the solution, the formulation to Kelvin. And different tumor parameters were evaluated. That is the tumor volume, which is evaluated when the animals is animals is alive. And then after the sacrificing humanly the animals, we evaluate the tumor weight, then percent tumor burden, then the percent tumor inhibition, as well as the morphology and the tumor size.
That is okay. That is okay. Like you said that your nanoparticle is around 200 nanometers, right?
Yeah.
And you have, 200 nanometers.
Yeah.
And you have given the administered the drug intravenous. So, like, have you thought about the clearance of the nanoparticles after the drug has been used up?
Clearance of nanoparticle after the drug has been used. So, okay, I'll try.
So when the nanoparticle, basically in case of tumor, because of the EPR effect, that is enhanced permeation and retention effect, the ideally the nanoparticle should have a size less than 200 nanometers. So it will be specifically by passive targeting, it will accumulate in the tumor cell. Apart from that, when we evaluate the PK parameters of this, the main goal when we develop a nanoformulation is to improve it. So the clearance will be, as compared to the free drug, the clearance will be prolonged. And apart from that, the since it is present in the circulation for prolonged period of time, I think I have shown the MRT, that is a residence time of this nanoparticle. So when the nanoparticle are present in the circulation for prolonged period of time, ultimately the PD parameter will get improved because it will have more time to reach out to the tumor site. So ultimate goal is to improve the pharmacokinetic parameter when we develop
No,
the nanoformulation.
the tumor size will decrease. There is okay, but it is going into the bloodstream, right? Now
Yeah,
it has to go outside the body through renal pathway. So
yeah,
renal for passing out through the renal pathway, it has to be around 6 to 8 nanometers.
Okay.
I think you need to work more on the size of the nanometer. I mean, nanoparticle.
Okay, sir.
Okay, thank you.
Thank you.
Thank you, judges, for your valuable questions. Sir, did you want to ask anything?
No questions. Go ahead, please.
So now I would like to call code number PhD07 to switch on the camera and confirm their presence, please. Code number PhD07. Please turn on your camera and confirm your presence.
Hello, is my audio and video available?
Yes, we are able to see you. Yes. We'll present your video now.
Greetings to all. The topic for my presentation is investigating the promising potential of ionic liquid mediated localized transdermal delivery of olutasidenib in breast cancer treatment. To start with a background on the hypothesis, breast cancer, we all know, is the most common type of cancer among women, and the data here highlights the need of promising therapy required for its management. Among the existing treatment options, chemotherapy is the most widely used and the current mainstay option required for the breast cancer management. However, it has several systemic side effects. Erlotinib is a chemotherapeutic drug which is a tyrosine kinase inhibitor. However, it has several side effects and it suffers from low solubility. Hence, to overcome this low solubility, here we are proposing a system known as cage ionic liquid system, which is composed of two compounds known as choline bicarbonate and geranic acid. Upon incorporation of the drug into the system, it will lead to solubilization of the drug as well as enhanced transdermal permeation. Further, the system will be loaded in a gel system and it will lead to the localized transdermal drug delivery effect. Here, it is important to know the breast anatomy a bit, that it is in such a way that it has thick memory fat layer, as well as it has internal lymphatic circulation, due to which the drug which passes through the breast skin, it is allowed to accumulate into the breast region. However, it does not pass into the systemic circulation. Hence, the cage ionic liquid system will lead to the enhanced permeation and accumulation of the drug in the breast region, which will lead to the reduced systemic side effects. Further, these are our objectives. First one is the preparation and characterization of the erlotinib loaded cage ionic liquid system. Second is loading of the system into the gel. Third is in vitro release characterization and cell culture studies. Fourth is ex vivo permeation studies, and lastly in vivo studies. Coming to our first objective, which is preparation of erlotinib loaded cage ionic liquid system. As we said earlier, it involves two compounds known as choline bicarbonate and geranic acid. A reaction will take place and it will lead to a product known as a cage ionic liquid system, which will be formed by ionic interaction of these two. The process is very simple and it involves simple mixing of these two compounds, and upon continuous stirring, there will be evolution of carbon dioxide gas, and through rotavapor, the water will be removed. It will lead to blank cage ionic liquid system in which drug will be incorporated and will lead to the formulation of the final ATB loaded cage ionic liquid system. Further characterization of the system. Firstly, we have here characterized the cage ionic liquid system, which is a blank system, and this is the compound which has been formed. Hence, to confirm these interactions, first one is the H1 NMR spectra. Here we can see the absence of the two prominent peaks, one from choline bicarbonate and one from the gyranic acid in the final compound. Firstly, here we can see the absence of the ammonium ion at 7.1 ppm, and secondly, here we see the absence of the carbonate ion peak, which is at 11.8. Hence, we can see from this interaction as well that it has the cage system has been formed. Further, to confirm, we have C13 NMR, where there is the absence of the bicarbonate peak at 160 ppm. And then there is FTR spectra, where there is no broad band between the 2500 and 3300 centimeter, which signify the loss of hydrogen ion from the gyranic acid. Further, to characterize the allotinib-loaded cage ionic liquid system, firstly we have done the apparent solubility of the drug, and here we have found that solubility in the cage system was increased tremendously, about 476-fold. And further, there is XRD spectra in which a halo pattern was observed after solubilizing the drug into the system, which signify that the drug has been completely dissolved. Second objective, which is preparation and characterization of allotinib-loaded cage inogel, and for that we have here used HPMC, which was dispersed in water, and after continuous stirring and continuous mixing, our liquid system, which is allotinib-loaded cage ionic liquid system, was added to it, and this led to the final formulation known as allotinib cage inogel. It was further characterized, and the characterization included visual observation, the pH and rheological behavior, which was found to be pseudoplastic. It exhibited good spreadability, and viscosity testing was also done. Further, coming to our third objective, which is in vitro cell culture study. Firstly, the cell cytotoxicity assay, which was performed using the MTT assay and was performed in 3 cell lines, MDMB231, 41, and MCF7. And a concentration-dependent decrease in cell viability was seen in both the groups of allotinib-free drug and allotinib ionic liquid system. And a significantly reduced IC50 value was found for allotinib ionic liquid system in all 3 cell lines. Further, the quantitative cell uptake study, and similar results were found here, that higher drug uptake was found from the allotinib ionic liquid system as compared to that of the allotinib. Then a qualitative cell uptake study was performed using rhodamine dye, which is a fluorescent dye, which was incorporated in the cage ionic liquid system to check the extent of the cell uptake. And here we also performed this in the 3 cell lines, and it was found that the rhodamine high fluorescence was found in case of the rhodamine loaded ionic liquid system as compared to that of the free dye, and in case of the blank ionic liquid system. Further, the apoptosis assay was performed in also in 3 cell line, and higher fluorescent intensity was found for allotinib ionic liquid system, which denoted the enhanced apoptosis, also which was accompanied by the higher apoptosis indexes in all of the 3 cell lines. Then the in vitro release study was performed, which was carried out in a bath shaker, and a dialysis membrane was used, and it was found that the allotinib loaded inogel system showed a sustained release of approximately 60% in 24 hours. Coming to our fourth objective, which is ex vivo permeation study, which was carried out in the France diffusion cell using the pig ear skin, and it was found that the ionic liquid showed the maximum permeation through the skin, and then which was followed by the allotinib loaded inogel, and then which was followed by the least was found in the allotinib gel. Then the drug disposition study was carried out to know the amount of the drug in the different layers of the skin, that is stratum corneum and in the viable dermis layer, and it was found that a significantly higher amount of the drug was found in the dermis layer for ionic liquid system and for the inogel as compared to that of the free ETB gel in both the layers. Further, the qualitative skin permeation study was carried out. It was also carried out using the rhodamine 6 dye, which is a fluorescent dye, and it was found that in case of ionic liquid system and in case of inogel, enhanced permeation was seen in different layers, and however, which was not seen in the plain gel. Our last objective was to perform the in vivo studies. Firstly, we performed the pharmacokinetic study in female split dolly rats, and we calculated the parameters such as AUC, Cmax, T half, and MRT, and as compared to the free ETB gel, ETB cage inogel showed the enhancement of the PK parameters. The pharmacodynamic study was carried out to check the anti-cancer efficacy of the formulation, and it was carried out in the female biopsy mice. The mice were divided in the different groups, and a different treatment was given post the tumor induction. And after the treatment, the mice were sacrificed and the tumor were excised, and it was found that the lowest volume and the size of the tumor was found in that case of the ETB inogel. Also, significant tumor burden reduction was also seen in that case of the ETB inogel. Further, tumor penetration study was carried out. A diluted gel and a diluted inogel was prepared, and it was applied to the mice induced with the tumor, and it was done to check the penetration of the inogel inside the tumor, and it was found that the higher intensity was there in case of the diluted inogel, and which showed a better skin permeation, and it reached the tumor tissue as well. Lastly, the toxicity and skin compatibility study was performed. In the toxicity study, the kidney and liver biomarkers were checked, namely ALT, AST1 and creatinine, and for the ETB inogel, they were found to be within the reference range and did not increase significantly. Further, skin compatibility study was performed on the mice, and the negative control was taken as capsaicin, and PBS was taken as the positive control, and the TWL value was also checked, and it was not found to be increased significantly. And further, the images here we can see that for ETB inogel, it was comparable to that of the positive control. Hence, we can say that there was no skin toxicity. So finally, coming to the conclusion, firstly, the erlotinib loaded cage ionic liquid system showed increased solubility of erlotinib, which was 476 times relative to water. And then cell viability assay concluded that formulation gave better uptake, lower IC50 value in breast cancer cell lines. And the inogel system provided a sustained release and provided enhanced intradermal permeation. And firstly, in vivo studies reflected increased in the PK parameters and a good efficacy in the formulation, final formulation with a non-toxic nature. And thank you, everyone. That's all from my side.
I will now invite the judges to ask their questions.
Did you do the kinetic studies in this case with gel?
Yes, sir. Could you please repeat?
And how did you perform the kinetics studies in this case? The gel.
So the pharmacokinetic studies you are saying?
Mm.
Yes, sir. We applied it on the breast region, the tumor, on the breast tumor, and like after the application, we took time points on different interval. Like we took blood on different intervals, and we checked the amount of blood. We took we checked the amount of drug which has been passed to the systemic circulation.
You applied a gel.
Yes, sir. Yes.
Not the free drug you have checked.
Sir, we have applied the gel in two forms, in which one in which the drug was in free state, and the other one in which the formulation we have prepared, the ionic liquid formulation in which the drug was loaded, and it was in the gel form. It was also applied. So in the free drug in gel, and secondly the ionic liquid system which has drug that was incorporated in gel. Both were applied.
Okay.
And both were compared.
Same you did for dynamic studies.
Yes, sir. Yes.
There are no local effects of the drug.
Sir, local effects, if we say related to toxicity, they were not found, like no skin irritation was found here. But if we say like per se drug only, the erlotinib, it has found some like local effects like eczema. It has been reported in some patients.
This will not affect your delivery, the drug, choice of drug delivery system.
Sir, here the drug is solubilized, in the solubilized form, and we have loaded it into the ionic liquid system, which is a biocompatible carrier.
Most preferred route of administration is first is oral.
Yes, sir. Yes. Sir, actually, for this drug, oral formulation is already there in the market in the tablet form, but it has several other side effects as well. For example, like it causes liver damage, hepatitis also it causes, and other like renal damage also it causes because of the systemic side effects. So therefore, like here,
this way also the drug is coming to the systemic circulation only, no?
Sir, it is coming to systemic circulation, but it is far less as compared to what was in oral, and most of it is accumulated into the breast region, breast tumor region, because of which a localized anti-tumor effect is seen.
Okay.
Yes, sir. So which toxicity is also less.
So did you check the mucoadhesion property of your gel? Mucoadhesion.
Sir, could you please repeat?
Did you check mucoadhesion property of gel?
Sir, mucoadhesion property we have not checked since like we are applying it topically, but we have checked other properties such as like rheological properties and viscosity. All these we have checked.
Okay. Now, in case of tumor reduction studies, you have shown that when you compared with your gel containing drug only and ionogel, so there is significant decrease in tumor reduction in case of ionogel. So what can be the mechanism or what can be the reason behind it?
Sir, in case of drug-loaded gel as such, the drug did not pass into the did not pass the breast tissue and did not enter the tumor as effectively. We have shown also in case of where we have, but our formulation, which is ionic liquid system, it has a solubilizing property as well as it has a permeation property as well, due to which it has been permeated deeper into the skin layer as well as inside the tumor cell as well, which we have showed in that in one of the study where we have loaded the dye-loaded system we have entered and high fluorescence was there. So this is one of the reason that the tumor burden was reduced.
So permeation is because of your permeation is because of formulation or because of drug. So what is the BCS class of your drug?
Sir, this is BCS class 2 drug.
Mm-hmm. This is highly permeable.
So not highly permeable, like it has log P around 2.7. So we cannot say it is highly permeable, like since it is classified into BCS class 2, but not high permeable. It has low solubility of around 0.4 mg per ml, but the solubility was also increased in case of our ionic liquid system to 190 mg per ml, and the permeation was also increased. It was significantly increased.
Okay, all right. That's all from my side. Go to the next.
Okay, sir.
You have done the toxicity studies using this cell lines, right? So,
yes,
apart from those cancer cell lines, have you tried on some normal cell lines also?
Sir, on normal cell lines, we have not performed. We have performed in cancer cell lines on only 3 cancer cell lines, MCF7, MDMB, and 41.
Yeah, I'm asking it because you have told that some skin rashes and eczema occurs in some patients, right? So apart from that, there may be some other toxicities also which needs to be investigated.
Sir, with drug that was after oral the like skin irritation I told, that was after oral administration. But here we are applying it transdermally or topically, so like the chances are less. But however, we will incorporate that.
Sure. Okay.
Yes, sir.
That's all from my side.
Okay, thank you, sir.
Thank you so much, judges, for the valuable questions.
Now I would like to call upon board number PhD08 to turn on their camera and confirm their presence.
मैंने सुना लेकिन अरे नॉर्मल में हम करते ही नहीं ये
PH07, please mute yourself. PH08, please turn on your camera and confirm your presence here.
Yes, I'm here.
We'll present your video now.
A very good morning, respected session chairman and my dear colleague. Today I'm talking on cell fibrin film and TiO2 nanoparticle cell fibrin film nanocomposite for wound healing application. Wound healing is a complex multi-step biological process which involves tissue growth and regeneration. For the treatment of such wound, the wound should be immediately treated with a proper dressing material for rapid healing. Therefore, there is a need to explore novel biomaterial for such dressing with enhanced biocompatibility, biodegradability, and antimicrobial property. Coming to the property of cell fibrin and TiO2 nanoparticle, cell fibrin, they are known for their excellent biocompatibility, biodegradability, and high mechanical strength. The cell fibrin protein can be fabricated into different formats such as film,
camphor, or hydrogen. These are some of the biomedical application of cell fibroid, which include wound healing, corneal regeneration, and drug delivery. Cell fibroid is known to promote wound healing by increasing the cell growth, proliferation, and migration due to the presence of a very specific RGD tripeptide in the cell fibrin protein, which are reported to bind to the integrin receptor. The TiO2 nanoparticle, when they are radiated with the UV light, generate free radical that produce antimicrobial effect. Therefore, our present study focus on the potential utilization of this cell fibrin protein with enhanced biocompatibility and biodegradability property, along with the TiO2 nanoparticle with their antimicrobial property for wound healing application as a biomaterial. There is an overview of the extraction of the cell fibrin protein from the Anthraeophyte cocoon to the fabrication of the cell fibrin film. A thorough characterization study of the cell fibrin film were performed. Then there is a synthesis of TiO2 nanoparticle by thermolysis method. To evaluate the potential application of the cell fibrin film as an ideal wound dressing biomaterial, we have performed in vitro wound healing assay using L9 2-line cell lines. Firstly, the cell fibrin film were coated onto a 12-well plate to which the cell line was grown. Once a confluent monolayer of L9 2-line is obtained, a wound gap is created using a microtube and observation at different time points at 6 hours, 24 hours, and 48 hours, and the images were taken. And to access the in vivo wound healing study, we have used female Wistar rats, which were divided into 4 different groups: group 1, group 2, group 3, and group 4. Firstly, the animal were anesthetized with ketamine, and the dorsal side of the animal were safe and cleansed with 70% ethanol, followed by a circular excision wound created with sub 10 mm thickness. Then the different materials were placed over each wound, and the wound closure was observed for different days: 7 days, 14 days, and 21 days, with photograph taken for each respective days. And coming to the resonant discussion, these are the biomaterials which we have successfully fabricated. And the overall characterization study revealed that the cell fibrin film indeed has excellent structural and thermal stability property, and also the presence of trace element and the cell fibrin study was analyzed using trace element analysis, and there was no presence of trace element in the cell fibrin film. And coming to the result and discussion, from the in vitro wound healing assay, we were able to confirm that the cell fibrin film has shown better wound closure compared to the control, even to the collagen film. And this was supported again by the in vivo wound healing study. Here also, we observed that the cell fibrin film has shown better wound closure compared to the group 1 and the group 2, and followed by the TiO2 nanoparticle cell fibrin nanocomposite has also shown good wound closure. Then there is an ongoing experiment. We are currently performing in vitro wound healing assay of the TiO2 nanoparticle cell fibrin nanocomposite. Also, we are studying the biodegradability of the body material, and then we are presently doing the mechanism study of the actual signaling pathway involved in the wound healing, and also histopath and the biochemical study will be carried on. And then from our present study, we report for the first time the successful fabrication of cell fibrin film from the cocoon, and then preparation of TiO2 nanoparticle cell fibrin film. And then from the result of in vivo and in vitro wound healing study, the cell fibrin has shown a promising potential as an biomaterial, and also the cell fibrin film, in comparison to the commercial cream used for the wound healing, will provide better alternative wound dressing material, because these are naturally extracted from a natural source than the chemically synthesized compound which will have difficult in their biodegradation. Therefore, the cell fibrin film will provide better wound dressing as the biomaterial. Thank you for your kind listening.
I would now like to ask the judges to ask their questions to the presenter.
Why in this study only female rats were used? Any specific reason?
There is no specific reason due to the ability of the animal during the experiment. We have taken this one female rats.
Do you expect any changes with gender in terms of wound healing?
Do you mean, sir, between the female and the male rats? Actually, sir, I have confirmed, I have read paper. I don't think so. That there are some hypotheses that if we use the female rats, there will be a chances of the changes in during the hormonals and all. But as we this one discussed further, we were able to find out that there will be no much difference between the females and the male rats. As this study will be also carried out when the human trial, it has to be given to both male and female. So like, to be honest, the study should be performed between a group of the group should be consist of half male and half female. So like speaking, there should not be much different apart from that hormonal changes that happen in the female rats. So but there should not be any changes.
So these were normal animals.
Yes, sir. Yes, sir. Yes, sir.
And you created a wound in those animals.
Yes, sir.
So, what you would it not be good to take animals where there is impairment in the wound healing, rather than taking a normal animals? In your study, you say that it has a potential to heal the wound. Instead of taking normal animals, you should have used animals where there is impairment in the wound healing.
Yes, sir. This is our preliminary study. We started with the normal animals, then we will target to use the diabetic rats, where diabetes will be induced in the rats, and we will like to study with the wound healing in the diabetes rats. And other corneal regeneration is one of the application we are targeting for our study using the biomaterial. Apart from wound healing, the corneal regeneration is also one of the most potent application for this biomaterial.
Okay, I'm done. Others can ask.
Any further questions?
Yes, I have one question.
Yes, sir.
Yeah, what was did you study interaction between TiO2 and sil fibron? Like, what is the mechanism of interaction between them?
Mechanism is that, sir, what we are trying to explore from when we prepare a nanocomposite of these two mechanism is that when we have already the cell fibrin film, onto this we have done the drop casting of the TiO2 nano suspension of the TiO2 nanoparticle, and we allow them to dry and then put in the UV before the application of this material for any application. So what we are expecting from this nanocomposite is that when this nanocomposite is prepared between the cell protein and the TiO2 nanoparticle, we believe we were expecting that the TiO2 nanoparticle will provide the antimicrobial property along with.
Just sorry to interrupt. Yeah.
Yes,
so you prepare TiO2 nanoparticles and then incorporate that into film.
The cell.
So how did you prepare a film? How did you prepare a film?
Preparation, we started from the raw cocoon of the silkworm. We have
just tell me the key ingredients of that.
You mean the main ingredient of the cell fibrin?
Yes.
Okay. There are three major proteins that are present in the cell fibrin protein. That is the alanine, glycine, and serine. These are abundantly present compared to any other amino acid residue. These are the three main amino acids that is present in the cell fibrin protein, which are mainly responsible for the secondary structure of the cell protein, which provide a crystalline structure. When these have a proper crystalline structure, more the stability of the protein is. So there is a one positive point of using the cell fibrin protein because they are more stable. When we do the TG analysis also compared to any other cell fibrin film prepared from other sources, the film we have
Okay, did you
prepared.
carry out characterization of the film?
Yes, sir. We have carried out a very thorough characterization starting from
Okay,
the.
all right. Fine, fine.
Yes.
I'll hand it over to next. Done from my side. Yeah.
I don't have any questions.
Thank you, judges, for your valuable questions.
So we come to an end now. And thank you so much to all the judges. So, dear judges, I would like to request you to share a few words on your experience for today's event.
Anil
Share
sir,
a
please.
few words. Okay, sir. Dr. Anil, would you like to share a few words?
Okay, I'll start from my side. Yeah, so I really enjoyed all the presentations. Like I come across the variety of research areas, and really all presentations were very good. And yeah, it's very like very versatile research across. I come across actually today. So it will be like we are evaluating the presentations, but yeah, ultimately that all presentations are good, and I congratulate all of you for presenting at this platform. Thank you.
Thank you so much, Dr. Atul. Dr. Dhanraj, would you like to share a few words?
Yeah, very good afternoon.
Good afternoon, sir.
It's been a pleasure to be a part of the fifth Congress, Science Congress, organized by Dr. Bhanuben, Nanavati College of Pharmacy, co-hosted by University of Mumbai. And as we go through the presentations, the most of the presentations are all superb, right? A very versatile work has been carried out by different workers, and like although we are judging this particular event, we also got to learn new things from these presentations, and I would like to appreciate all the speakers of this particular session, and I wish them all the very best in whatever endeavors they carry out in near future. Thank you so much.
Thank you so much, sir. Dr. Anil, would you like to share your experience?
Yeah, thank you. Most of the points already covered by Professor Atul and Dr. Dhanraj. Both of them highlighted. Like the students who have presented have done excellent work and diverse work. For us also sometimes it's a challenge also, and we also take it as an opportunity to learn from them. So my best wishes to all of them, and congratulations to BNCP for arranging this Congress. Thank you.
Thank you very much, sir. Thank you, Dhanraj sir, and thank you, Gaikwad sir. Thank you very much.
Thank you.
And thank you, Atul sir.
Thank you so much.
Thank you so much, judges. On behalf of the organizing team, it is my privilege to deliver the vote of thanks on this momentous occasion. First and foremost, I express my heartfelt gratitude and utmost respect to our distinguished judges. Your valuable time, insightful feedback, and immense expertise have not only elevated the quality of this event, but also enriched our collective understanding. Thank you for your generous contribution and for inspiring us all.
We would like to hand over a certificate of appreciation to Dr. Atul, Dr. Anil, and Dr. Dhanraj for their presence here today.
Thank you very much.
Thank you so much.
Please join us for the valedictory meeting at 3:30 p.m. The valedictory session will be held in the same meeting. So please stay in the meeting if you want to attend it. A sincere thanks to our incredible participants. Your enthusiasm, creativity, and hard work have truly made this event unforgettable. You are the driving force behind this competition, and we are immensely proud of each one of you for your dedication and innovation. Lastly, I extend my sincere appreciation to everyone present here today. Your support and encouragement have been crucial in making this event a resounding success. Thank you for taking the time to be with us and for contributing to this enriching experience. Once again, thank you to each and every one of you. Wishing you all continued success and growth. Thank
Thank
you.
you so much, everyone.
Judges, we are really very sorry for a small technical glitch due to which we just missed out on presentation. PhD 10, can you please turn on your camera to confirm your presence? PhD 10.
Yeah, I'm there.
Yes, please turn on your camera to confirm your presence.
My camera is on, yeah.
Yes, I'm able to see. We'll present your video now.
Good morning to one and all. I'll be presenting my work entitled GPER1, is it a promising target for chemo prevention of prostate cancer? So prostate cancer is the second most common cancer observed in men. And it is reported that by 2040, the incidence of prostate cancer will increase from 1.9 million to 3.9 million. This poses a great burden on the healthcare system, especially for a developing nation like India. Thus, there's a need to identify chemo preventive strategies. So prostate cancer development is a slow multi-stage process wherein the normal prostate epithelium undergoes certain genetic insults leading to the formation of prostatic intraepithelial neoplasia, which is a precursor for prostate cancer, which then progresses to invasive adenocarcinoma and then castration resistance and metastasis. So around 20 to 30% cases of PIN have been known over an average of 5 to 10 years to transform into invasive adenocarcinoma, thus providing us a suitable window to design chemopreventive strategies. So there are many molecules that are involved in the pathophysiology of cancer. One such molecule is GPER1, that is G protein coupled estrogen receptor. This receptor is localized onto the cell membrane, and in the presence of its agonist, G1 or estradiol, it leads to the activation of secondary messengers, leading to gene transcription, and further leading to pro-tumorigenic or anti-tumorigenic effects depending on cell type. Thus, GPER1 has been postulated as a therapeutic candidate in different cancers such as uveal melanoma, colon cancer, and gastric cancer. Coming to prostate cancer, it is seen that the treatment of prostate cancer cells in vitro or in the xenograft model with G1 leads to the inhibition of tumor growth. However, there are no studies that have explored GPER1 as a chemopreventive target for prostate cancer. Thus, we have formulated the following key questions. First, whether GPER1 expression is modulated during prostate cancer progression, whether loss of GPER1 or activation of GPER1 affects the characteristic of prostate cells, and whether GPER1 activation prevents cancer progression in TRAMP mice. To address this, we have 3 study models. One is the human benign prostatic human tissues, prostate cancer tissues grade 5. Then our second model is the TRAMP mice that demonstrates disease progression, which is similar to that observed in humans. And third are different cell lines, LNCAP, PC3, and RWPE1. So to address our first question, we have undertaken GPER1 expression studies. First, we have done in silico analysis. And in the second, we have done flow cytometry in real time to determine GPER1 expression in the human and mice prostate tissues. To address the second question, we have developed GPER1 knockdown cell lines, and we have treated PC3 cells with G1, and we have assessed its proliferation ability, migration and invasion ability. We have also performed RT-square profiler array for human tumor metastasis genes, zymography, and real-time PCR. To address a third key question, we have treated TRAMP mice at the age of 2 months, that is high-grade prostatic intraepithelial neoplasia stage, with 3 doses of G1 and vehicle for a period of 1 month, and the mice were sacrificed at 5 months, and further we did a histological analysis. Coming to our results, so on reanalyzing geo data sets, we find that there is a decrease in the expression of GPER1 in the epithelial compartment and in the whole tumor tissues in most of the data sets when compared to normal, paired normal, or benign prostatic hyperplasia tissues. When we look at our flow cytometry data, we find that the percent GPER1 positive cells is significantly low in the epithelial and non-epithelial compartment of prostate cancer patients compared to benign prostatic hyperplasia patients. We also assess the expression of GPER1 in TRAMP mice using RT-PCR when our data is expressed as delta CT. So a higher delta CT means lower expression. We find that the expression of GPER1 was found to be low in well-differentiated carcinoma stage, while it was found to be high at the high-grade prostatic intraepithelial neoplasia stage when compared to age-matched control. Not only the expression, but also the person-positive epithelial GPR1 epithelial cells were found to be significantly low in the well-differentiated carcinoma and poorly-differentiated carcinoma stage, while it was found to be high in high-grade prostatic intraepithelial neoplasia stage when compared to its respective age-matched control. However, this trend was not seen in the non-epithelial compartment, thus stating that loss of GPR1 in the epithelial cells was associated with prostate cancer progression. Now, how does this loss of GPR1 affect the characteristics of the cells? So, we developed GPR1 knockdown cell lines, LNCAP, PC3, and RWP1, and we assess its proliferation in vivo through xenograft assay and in vitro through Alamar Blue assay, and we find that there is no change in the proliferation of GPR1 knockdown clones compared to its empty vector cells. However, we do see a significant increase in the migration and invasion of GPR1 knockdown clones compared to its empty vector transfected cells, thus stating that GPR1 negatively regulates epithelial to mesenchymal transition. Now, what could be the possible factors that are involved in this GPR1 regulated EMT? To understand that, we performed the RT-square profiler array and we find 8 genes that were differentially upregulated, from which we validated the expression of MMP9 using zymography assay. And we find a significant increase in the pro-MMP9 levels in all the 3 GPR1 knockdown clones compared to its empty vector. Further, we looked at the expression of known EMT molecules, wherein it is well known that MID208 inhibits ZEP2, and increased level of ZEP2 in turn will decrease encadherin and increase Vimentin. We assess the expression of these molecules using RT-PCR, and we find that there's a decrease in the MID200A levels, increase in ZEP2 and Vimentin, and a decrease in the encadherin levels in all the 3 GPR1 knockdown clones compared to its empty vector transfected cells, thus stating that GPR1 regulates EMT through MID200A ZEP2, encadherin loop, and MMP9. So till now, we have seen what could be the effects of a loss of GPR1. However, what happens when GPR1 is activated through using G1, its agonist? So we treated our GPR1 knockdown clones and empty vector with different doses of G1, and we assess its proliferation, and we see that there's a decrease in the proliferation in the empty vector cells. Further, when we treated PC3 clones with G1, 1 micromolar and vehicle, we see only the empty vector treated cells to show a decreased invasion compared to its vehicle, while no effect was seen in GPR1 knockdown clones, thus stating that activation of GPR1 inhibits proliferation and EMT. Further, we now wanted to look whether these effects seen in vitro on activation of GPR1 would be also observed in vivo. So using the tramp mice, we developed a study wherein we treated tramp mice with 3 different doses of G1 at the HG-pin stage, that is high-grade prostatic intraepithelial neoplasia stage, and we find that 2 mg and 4 mg treated animals show inhibition in the tumor growth, while there was no change in the vehicle and G1 treated animals. Further, in histological analysis, we find 2 mg and 4 mg treated animals to display high-grade prostatic intraepithelial neoplasia morphology, while vehicle and 0.5 mg show poorly differentiated carcinoma morphology. There were no effects of G1 observed in the prostate of transgene negative mice, thus stating that activation of GPR1 prevents progression of high-grade PIN to poorly differentiated carcinoma. Thus, coming to our conclusions, we can say from our in vitro data that GPR1 inhibits ZEP2, and ZEP2 in turn positively regulates Vimentin and MMP9 expression and negatively regulates encadherin and MID200A, thus leading to a negative regulation of EMT. Further, in vivo data in tramp mice demonstrates that on G1 treatment at an early time point, it leads to the prevention of prostate cancer, thus demonstrating that GPR1 could be a good chemopreventive target for prostate cancer. Thank you.
I now invite the judges to ask their questions to the presenter.
You take this one.
Can you please explain your findings done through real-time PCR?
Yes, so when we knocked down GPR1 and we developed stable clones, we found that there is a decrease in the levels of E-cadherin and MIR200A, while it is leading, while there's an increase in vimentin levels and increase in ZEP2. So these molecules are very well known to be in EMT. So there is a whole cascade that is taking place wherein MIR200A is downregulation leading to increased ZEP2 and vimentin and decrease in E-cadherin expression.
So for knockdown, what do you use? You use
For
CRISPR
knockdown,
Cas9.
we use the shRNA technology.
Okay, not CRISPR.
No, not CRISPR.
Okay. So for application in pharmaceuticals, how do you imagine what will be the outcome in future for which you can use this particular technology for treatment?
So our in vivo study wherein we are demonstrating that the early activation of GPR1 could be a chemopreventive therapeutic chemopreventive for prostate cancer. I think if this is taken into clinical trials further, then we can actually especially in patients which have high PSA levels and a familial cancer history, they can be given this chemopreventive drug, and I think prostate cancer progression at least can be prevented.
What will be the best mode for administration?
Best mode for administration, in my model, we have given subcutaneous, but I think oral dosage would be a better because it becomes easier for the patients also to intake.
Okay. Thank you. That's all from my side.
Thank you, sir.
Okay, in one of the studies, like you are taken 2 mg and 4 mg, right? Dose
Yes.
in the animal studies mice. So did you observe same effect in both the cases, or like I
Yes,
must
so
have missed that point?
with 2 and 4, we did observe tumor inhibition, and the prostate size or the weight does not differ much. But we did see with 4 mg, out of 6 animals, at least 3, half of the animals showed us additions to the spleen. So we think that may be a bit of toxic, although on different organs, when we check toxicity levels, it never showed a drastic effect. But I think 2 mg would be a better suitable dose to go forward with.
Okay, so what would be the prospective application of your work, like whatever mechanism you have established?
So I think with the current treatment that is going on, so we have not looked at the therapeutic effect of how GPR is, but from a chemopreventive point of view, if we initiate a study wherein we initially give patients, as mentioned previously, I think it would be beneficial since it would prevent progression. Also, it could be a combinatorial therapy, like along with maybe later on, if we can do studies wherein we can do a combinatorial therapy of androgen deprivation and G1, and maybe it will lead to a few resistant cases or better outcomes for patients.
Okay, all right. All from my side. Thank you.
Thank you, sir.
That is all for today's session. If you want to attend the valedictory session, it will be held at 3:30. So please be with us. Thank you.
Thank you.
Thank you so
Thanks.
much.
Good afternoon, distinguished guests, respected judges, esteemed faculty members, and my dear friends. On behalf of SVKM's Dr. Bhanuben Nanavati College of Pharmacy, it is both my privilege and honor to deliver the valedictory address for the 5th Student Research Congress themed Innovations for Better Health. Over the past 2 days, we have witnessed an inspiring convergence of ideas, innovations, and intellect that will undoubtedly propel the future of healthcare research and development.
Let us begin by expressing our heartfelt gratitude to our co-host, the University of Mumbai, whose collaboration has significantly elevated the stature of this Congress. A special mention to our industry partners, ACG Worldwide and SciTech Center, for their unwavering support. Your sponsorship of prizes and steadfast commitment to promoting innovations have been pivotal in fostering a competitive yet nurturing environment for our participants. It is our distinct privilege to extend heartfelt thanks to all those who have made this event a grand success.
Firstly, we would like to express our sincere gratitude to Dr. Ravindra, Honorable Vice Chancellor of the University of Mumbai, and Dr. Shivram, Dean of the Faculty of Science and Technology at the University of Mumbai, for their unwavering support in making this event possible. A special thank you to our esteemed industry partners, Mr. Ajit, Chairman of ACG Group of Companies, for graciously accepting our proposal to collaborate on this event. I would also like to express my gratitude to Dr. Ketan, General Manager at ACG Worldwide, and Dr. Parizad, Principal Technical Consultant for Pharma at ACG World, for their continuous support of the Student Research Congress. We are also grateful to Dr. Prema, Assistant Manager of Marketing at ACG World, for her impeccable coordination throughout the event.
I would like to express our deep appreciation to our distinguished keynote speaker, Dr. Deepak Modi, a scientist in molecular and cellular biology at ACMR NIRRCH, for enlightening us on in vitro testing methods, organ on a chip models, and micro devices designed to mimic organs in the context of reproduction studies. His valuable insights have greatly enhanced our understanding of these innovative technologies and the potential in advancing research in this field. Dr. Srinivas Savle, CEO of AIC LMCP Foundation, delivered an enlightening talk on innovation, offering valuable insights into the transformative power of creating, thinking, and technological advancement. Drawing from his extensive experience in fostering entrepreneurship and innovation, Dr. Savle emphasized the importance of cultivating a mindset that embraces productive change.
I am deeply honored to thank our judges for the undergraduate category, Dr. Kavita, Dr. Ketan, and Dr. Gaurav. For postgraduate category, Dr. Rajkumar, Dr. Radhika. For PhD category, Dr. Anil, Dr. Dhanraj, and Dr. Atul. For the IIC category, Vijay Shivpuje, Sir Dr. Srinivas Savle, and Dr. Meenal. Your dedication to evaluating each research endeavor with diligence and impartiality is commendable. Your insights, expertise, and encouragement have left a lasting impact on all the participants. Thank you for sparing your valuable time and enriching this event with your wisdom.
We would like to extend our sincere thanks to the BNCP Internal Core Research Committee for their meticulous work in screening the blinded abstracts and selecting the best of the best presentations. Their dedication and effort have played a crucial role in ensuring the quality and success of this event. A special acknowledgement to our beloved principal, ma'am, Dr. Munira Momin, for her visionary leadership and constant support. Ma'am, your guidance and encouragement have been a constant source of inspiration for all of us. Your commitment to fostering excellence in research and academia motivates us to push boundaries and strive for greater achievements every day. Thank you for always believing in us and guiding us towards success.
Now, we would like to present the highlights of the 5th Student Research Congress. The Congress has been a resounding success with an overwhelming response of over 800 registrations from across the nation. We received a total of 249 abstract submissions across both undergraduate and postgraduate categories, showcasing the depth and diversity of research. Over a rigorous evaluation process, 49 abstracts were shortlisted, representing the top 20% of all submissions. The breadth of participation highlights the growing interest and enthusiasm for research and innovation in our community. This year, we witnessed participation from 25 states, 105 universities, and 200 institutes and colleges, including 176 from the pharmaceutical field and 24 from non-pharma disciplines. This impressive turnout reflects the vibrant research ecosystem and the collective pursuit of excellence in academia.
The Congress also saw exceptional contributions from undergraduate and postgraduate students, PhD scholars, and professionals, highlighting the strong spirit of collaboration between academia and industries. This synergy has not only enriched the discussions but also laid a solid foundation for future breakthroughs in healthcare. The diverse perspectives and innovative ideas shared during this event serve a reminder of the collective potential to drive progress and make meaningful contributions to the advancement of science and technology in the field.
We would like to take a moment to deeply appreciate the tireless efforts of the Student Research Congress Organizing Committee, faculty members, and student volunteers. Your hard work behind the scenes has been instrumental in ensuring the seamless execution of this grand event. A special thanks to our dedicated teachers, Dr. Lokesh Kumar Bhatt and Dr. Sujata Savarkar, for her unwavering support and guidance. Your meticulous planning and attention to detail have not only contributed to the success of this event, but have also set a high benchmark for future endeavors. We would also like to extend our heartfelt thanks to Dr. Afreen Khan and Ms. Siddhi for her exceptional coordination of the event.
Thank you all for your invaluable contributions and commitment to excellence. To all the participants, your enthusiasm and dedication have truly been the heart and soul of this Congress. Each one of you have brought something unique and valuable to the table, and we hope this platform has inspired you to continue your journey and research and innovation. To the winners who will be announced shortly, congratulations. Your exceptional contributions have set a new standards of excellence, and we are thrilled to celebrate your achievements today. Your hard work and dedications are an inspiration to all of us, and we look forward to seeing the incredible impact you will continue to make in this field.
Now, we would like to announce the winners for all the categories. We kindly request Dr. Munira Momin and Dr. Sujata Savarkar to please do the honors.
Thank you, Team SRC. And let me congratulate all participants, all presenters for the fantastic presentation and study which you have done, the fantastic research work which you have shared. And believe me, it was tough for all the judges. This is what the feedback I received from all the judges, that it was very, very tough for all the judges to decide 1, 2, and 3 rank. Everyone is a winner, but at the end of the day, we have to announce few of you as a winner, but for me, everyone is winner. So let me start with undergrad. And I'm especially very much fond of undergrads research. It makes me very happy as a teacher, as a researcher, as a head of the institute, when undergrads present their research work. It shows their inclination towards research. So let me start with the undergrad. We'll start from number 3. So we have number 3, BPH08 as the third number, Miss Salome Firi, Sacred Heart College Autonomous. Congratulations, Miss Salome. Now we go for and the Sacred Heart College of Tamil Nadu. We go for this second position, and here we have a second position of BPH 11, Miss Neha Sheikh stood second from Dr. D.Y. Patil Institute of Pharmaceutical Science and Research, Pimpri, Pune. Congratulations, wonderful presentation. Now, coming to number 1. Number 1 was tough whom to select. So it's a tie. 2 students are sharing the number 1 position. Congratulations to BPH 2 and BPH 3. Both are sharing the first position. Miss Nidhi Mishra, MET Institute of Pharmacy, and Miss Ketki Joshi, SK Somaya College, Maharashtra, Mumbai. Congratulations to all UG participants. Keep up your research work. There is a long way to go for you, and I'm sure this competition has given you the way ahead. The presentation made by all your peers in a UG category, I'm sure that has paved a new path for all of you. Keep up your research, and congratulations to one and all. And very soon your prizes will reach you and my team will connect with you. So congratulations once again to MET Institute of Pharmacy, Miss Nidhi, SK Somaya College, Miss Ketki, Miss Neha from D.Y. Patil Pimpri, and Miss Salome Firi from Sacred Heart, Tamil Nadu. Congratulations to all of you. And now over to Dr. Sujata Savarkar for PG category.
So, first of all, congratulations to all the participants. Your presence was extremely overwhelming because we had participation all over India. So it was really fascinating, and I resonate what Principal Madam is saying. So every participant is winner to us. In fact, when judges approached us, they were highly impressed with the scientific deliberations that happened over the 2 days. Having said that, I would also like to congratulate the SRC team, the first year students and the faculty who guided them. Meticulous arrangement. So over to the winners of PG category, and we have some exciting announcements for the winners. So beginning with position 3, it is PG09. Miss Ritu Patel from SVKM's Dr. Bhanuben, Nanavati College of Pharmacy. So congratulations, Ritu, and to your mentor. For position 2, we have PG02, Namish Manchanda from Naipur Sas Nagar, Mohali. Congratulations, Namish. And now comes position 1. Oh my God, it's a tie. Absolutely tie. So for position 1, we have PG01, Chirag Ketan Gala, again from Naipur, Mohali, and Sandeep Vittal Shinde, Institute of Bioinformatics and Biotechnology, Savitribai Phule, Pune University. Congratulations, Chirag, Sandeep, Namish, and Ritu. Congratulations, happy researching, and do good research in your near future. Be good research scientist. Best wishes to all of you all. Now coming to PhD category. So for PhD category, at position 3, we have again tie. So PhD 01 and PhD 02, they are the winners. So Nandini Chitara from Department of Anthropology, Punjab University, Chandigarh, and Mr. Ikrar Ahmed from R.C. Patel Institute of Pharmaceutical Education and Research, Shirpur, Maharashtra. Congratulations, Nandini and Ikrar. For position 2, we have PhD 10, Junita Anna D'Souza from ICMR, National Institute for Research in Reproductive Research and Child Health, NIRCH. So congratulations, Junita. And again, we have PhD 08, tied, Sansanam Yaibhai from Manipur University. And for position 1, it is Babita Govind Singh Negi, PhD 04 from ICMR NIRCH. So congratulations and happy researching. Congratulations and best wishes to all of you all. And with this, thank you, I request Principal Madam to announce the winners for Innovation Challenge. All the best.
So congratulations to all PG, PhD category. Wonderful research. You can see your research was of so good quality. We had a real hard time there. I mean to say the judges had a real hard time to say which one is good. So everyone did wonderful. And at some point of time, we had a tie. But I'm very happy that the judges really worked hard and they came up to some out the solution, and they came out with the final numbering as a tie. Everyone was fascinating. Everybody's work was so good. Wonderful. Congratulations. So let me go ahead now. The last category. Once again, we had the judges had hard time, and I can see that on again there is a tie. So even IIC category has a tie. So let me start with number 3. And believe me, all IIC were so good. And I wish all of you, if you need any help in mentoring, the Atal Incubation at Ahmedabad has agreed to groom all of you. We would be very happy to facilitate that for you. Please contact my SRC team and coordinator. We will facilitate this support, and we will connect you with the Atal Incubation Ahmedabad. They will help you to groom and realize and make sure that your thoughts and your innovation comes to reality, and you can commercialize it further. So in IIC, position number 3, which is a tie, is IIC 7 and IIC 9. IIC 7, Shivanshu Pandey, SVKM's Dr. Bhanuben, Nanavati College of Pharmacy, and IIC 9, Shraddha Sonavne, Baba Atomic Research Center, Homi Baba National Institute, Maharashtra. Congratulations, Shivanshu and Shraddha, for position 3. We have now position 2, that is IIC 8. That is position number 2 is Shreya Garze from SVKM's Bhanuben Nanavati College of Pharmacy. Congratulations, Shreya. And let's go on position number 1, which is IIC 6. IIC 6 stood first, Mr. Harsh Bokadia and Miss Aditi, Sunandan Divetia School of Science, STSOS, Mumbai. Congratulations to each one of you, and I wish you all the best, and I encourage you to work further to make sure that your innovation turns into reality as a product or services, and you can commercialize it. So congratulations all the winners of UG, PG, PhD, and IIC. Congratulations once again to all of you. Over to you, team.
Your achievements are a source of immense pride for all of us, and we eagerly look forward to seeing the lasting impact that your work has on the field of healthcare. Keep pushing the boundaries of research and continue to shape the future of healthcare with your passion and dedication.
As we conclude this remarkable event, let us carry forward the spirit of innovation and collaboration that has defined this Congress. The theme, Innovations for Better Health, is not just a tagline, but a call to action for all of us to strive for excellence and contribute meaningfully to society. We hope this Congress has inspired new ideas, forged valuable connections, and sparked a renewed passion for research that will drive transformative advantages in healthcare.
Once again, I extend my deepest gratitude to all our guests, sponsors, judges, faculty members, students, and volunteers who have made this event a grand success. Your support and dedication have been invaluable in bringing this Congress to life. Let us take a moment to celebrate our collective achievement and look forward to an even brighter future, filling with more groundbreaking research, innovation, and collaboration. Together, we are paving the way for a healthier tomorrow. Thank you once again, and here's to continued success.
Let us now proceed to the group photograph. May I request all participants, guests, and winners to kindly turn on the cameras. Let us capture this moment of celebration and togetherness.
Winners wish to say something?
Can I?
Yes, please. Okay.
Thank you so much for providing us the platform to show our work. It was a really great experience, and thank you to all the organizers for maintaining and for keeping it to the schedule. And it was really great listening to all of the research that is ongoing in the field of pharmaceuticals and innovation in health. Thank you so much.
Thank you so much, ma'am. Thank you so much, sir. Thank you, the organizing committee, for giving us such a great platform to represent our research and take the valuable insights from all the scientists, all the great scientists, and all the researchers. Thank you so much, ma'am. Thank you so much.
Can you introduce who are you? From which institute?
Ma'am, I am from the Department of Anthropology, Punjab University, Chandigarh, and myself, Nandini Chitara, ma'am. I'm pursuing my PhD, ma'am. Thank you.
Good, Nandini.
Thank you, ma'am. Thank you, ma'am.
We are very happy that we have a representation from all.
Great. Thank you, ma'am. Thank you, ma'am.
Thank you so
Dr.
much.
Dhanraj, thank you very much. I'm sure it was very tough for you to judge.
It was really tough, and it was hands-on, and everybody performed very well. It's been a pleasure taking part in this.
Great.
Thank you so much.
Thank you very much for spending your time, and this encourages the new generation, this PG, PhD, all these researchers. When they see dedication of seniors, they get encouraged and motivated. Thank you very much for providing that motivation. Any other winners? Do we have who would like to share? Do we have a Sacred Heart, Tamil Nadu? We have.
Yes, I'm here.
Yes.
I'm UG_08, and I would really love to say thank you so much. I didn't really expect the presentations were so enthusiastic, and actually I didn't really expect myself to find myself in any of the prizes, but yeah, God is good. I mean, thank you so much. I am so happy, and
congratulations.
If you keep on doing this, many of the youths are going to venture into this, and they are going to find their way to success. Thank you so much for giving us an opportunity to find ourselves, to search ourselves in whatever we're doing and coming back to the right thing. Thank you so much. I'm from Sacred Hearts College, and I'm Salome Piri.
And give my regards to Father Anthony.
Sure, thank you.
If I'm not wrong, correct?
Yes, yes.
Yeah, I just saw that.
Thank you so much, ma'am.
Spread this. Just see that next year more students from your college participate.
Sure, we are going to see to that. Sure.
Good, good. Do we have what was her name? I forgot.
Yes, he's there.
Yes.
Yes.
I'm sorry,
Hello.
I just could not pronounce your name.
Yes, first of all, I would like to thank the entire organizing team, starting from Munira ma'am and Afrin and Shidi. I have the truly honor, and this one always remain a special one. This is my first time I'm presenting my in vivo and in vitro study. So I was really scared and I was like very confused how to present my work. This is my first ever presentation of my cell culture study. So I was like tense also, but thank you for this opportunity. I will always remember and grateful to the entire team and to the student. They have done a fabulous, anchoring. Afrin and Shidi was always there online to help us in the WhatsApp group. And Munira ma'am, thankful for guiding everyone all of us so well. And thank you to the judges for asking questions that much. At least we got some input how we can improve our work and all. I am so thankful to the entire team, and I wish everyone all the best for your works, and congratulations to all the participants also. Thank you, ma'am.
That's great. Thank you. And this is what we are looking at. We want to spread the message of science. We want to promote the research at every level. And we are so happy that we are able to do it. And that too, our ACG team is supporting us. We have ACG technical advisor with us, Dr. Parizad. We are so happy that Madam started the event with a keynote speaker, and today she is again present with us. Madam, over to you.
I put my camera on mute. It was a fantastic event, and what presentations and like the very participation that was there. I think it's a real pleasure to be sponsoring such an event, and I mean, even the prizes, it must have been very difficult to judge because we are seeing, prizes, 2 first or 2 thirds or 2 second prize winners. I mean, it must have been so difficult to judge the presentations. A few of them I was honored to attend. Excellent work going on in India. I sincerely wish we can translate them to commercial. I sincerely wish that. And that would be the ultimate achievement of the total efforts that have gone into this type of a symposium. So anyone needs any help, any support, any mentorship, please feel free to contact. I had shared my contact details yesterday. So thank you so much. Thank you. It was an honor. Thank you.
Thank you very much, madam. That means a lot. That it motivates all students when they have an industry support. It encourages them, it motivates them to push the boundaries. So thank you very much, madam.
Most welcome. My pleasure.
Yes, over to you, team.
I'll just spot it.
All right.
But
Okay.
then you want to
Thank you, everyone. Officially close it.
Thank you.
Thank you, everybody.
फोटो बाद में आ जाएगा.
Keep researching.